and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner.
Good day, everyone, and welcome. Thank you for joining us today for day 2 of Advancing Generic Drug Development, Translating Science to Approval. My name is Ray Ford, and I'm with the Small Business and Industry Assistance Program, which operates within the Division of Drug Information, which in turn resides in FDA's Center for Drug Evaluation and Research, Office of Communications. I'll serve as your host and moderator for today's webinar. I'm pleased to announce that in addition to the certificate attendance, which supports CEs for RAPS, SOCRA, SQA, and ACRP, this webinar is offering a total of 13.25 contact hours to physicians, pharmacists, and nurses. Note, this is an entirely separate process from that of the certificate of attendance. The CEs are available to all real-time attendees who self-identified as healthcare professionals during the Adobe Connect registration process. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than that of only a single supporting company. CE faculty and speakers are required to disclose to attendees when products or procedures being discussed are off-label or unlabeled, not FDA approved, and any limitations on the information that is presented. Today's faculty has nothing to disclose. We have a spectacular lineup of speakers today for our webinar. The Planning Committee, Speaker, CE Consultation, and Accreditation team have nothing to disclose. Participants must attest to their attendance and complete the final activity evaluation each day via the CE portal at ceportal.fda.gov. Final activity evaluations must be completed within 2 weeks after the activity. No exceptions. So in this case, the deadline for today's webinar is September the 28th. I apologize in advance, but we are unable to make exceptions to that deadline. Pharmacists should note that the failure to provide your correct NABP and date of birth information in the required format may result in loss of credit for this activity. The NABP profile number should be the 6 to 7-digit number assigned by the CPE monitor, and your birth date should be in the month, day format, such as 0914. Do not provide your pharmacy license number. When logging into the CE portal, please click the My Account tab and then navigate to the Edit Contact Information to verify that your information is correct. Physicians and nurses may then view and print a statement of credit. Pharmacists should log into the CPE monitor 8 weeks after the last session of the activity to obtain their CE credit. To summarize, attendees have 14 days from the last day of the activity to log in, complete the required CE evaluations, and attest to attendance to claim credit. SBIA will issue a claim code to all attendees later today, so make certain that you complete the CE session evaluation on or before September the 28th. In addition to the above process, when an attendee completes the webinar survey evaluation, they can then download a certificate of attendance, which may be used in support of RAPS, SOCRA, SQA, and ACRP credits. Also, this survey link closes on 14 days, and that's the 28th of September. To simplify things, the email that we send to attendees later today will have information on both the CE claim code and the survey link. For your convenience, this slide provides the QR codes for our social media avenues. You can scan them now with your phones and be taken to the appropriate platform. In our ongoing environment, this event is 100% remote and 100% virtual. As such, there may be some internet issues, but please bear with us. And if in fact we encounter those, we'll try to resolve them as quickly as possible. Speaking of tech issues, please visit our technical issue support page for your answers to your technical questions, for downloading the files, and accessing the survey link to obtain the certificate of attendance. That's the same page you used for your online access to the event, so please bookmark that page. And if you have technical questions, please check there first, and you'll most likely find the answers. Before we begin, we'd like to cover some details. Slides for today's presentation are available from the right-hand side of your screen where the orange button says Download Slides. Please indicate the presenter that you wish to have answer your questions by identifying the speaker, for example, @Stephen. This activity is eligible for the attendance certificate, which supports continuing education by SOCRA, RAPS, SQA, and ACRP. Please refer to our website at fda.gov/SBIA for more details. You'll be able to obtain the attendance certificate only upon completion of the survey, which remains open for 2 weeks. Please remember to download the certificate immediately upon completing the survey. It cannot be retrieved after the fact. Again, that deadline is September the 28th. Even if you don't need the certificate, we would appreciate it if you let us know your comments and feedback via the survey. The webinar we posted in its entirety on our website in approximately 5 to 7 business days. We'd also like to remind you of some upcoming events on September the 18th and 19th.
Within my talk, I will be discussing spray velocity characterization. Spray velocity characterization provides an additional metric for emitted spray plume characterization. There are three main categories of OIDPs that are the focus of generic drug user fee amendments, or GDUFA funded research. These products are metered dose inhalers, otherwise known as MDIs, dry powder inhalers, otherwise known as DPIs, and inhalation spray inhalers. GDUFA-funded regulatory science research on spray velocity characterization indicates that spray velocity measurements have established regulatory significance for inhalation spray products. For certain MDIs and DPIs, there is potential for spray velocity measurements to support regulatory decision-making, but further research is needed. This presentation will focus on spray velocity characterization for inhalation spray products. So what is an inhalation spray product? The FDA guidance for industry on nasal spray and inhalation solution suspension and spray drug products, chemistry, manufacturing, and controls documentation recommended in July 2002 defines inhalation sprays as follows. An inhalation spray drug product consists of the formulation and the container closure system. The formulations are typically aqueous-based and by definition do not contain any propellant. Aqueous-based oral inhalation sprays must be sterile. Inhalation sprays are intended for delivery to the lungs by oral inhalation for local and/or systemic effects. The products contain therapeutically active ingredients and can also contain additional excipients. The formulation can be in unit dose or multi-dose presentations. The dose is delivered by the integral pump components of the container closure system to the lungs by oral inhalation for local and/or systemic effects. To date, there are 4 inhalation spray drug products approved on the market. According to their approved labeling, these 4 inhalation spray products utilize the Respimat soft mist inhaler device to produce a metered slow-moving aerosol cloud following actuation. There are no generic products available. The draft product-specific guidance recommendations for each inhalation spray product in the table is available on the FDA website for generic drugs. Inhalation sprays exhibit many similar features to aqueous-based solutions for nebulization, aqueous-based solution nasal sprays, and propellant-based solution MDIs. The sprays from inhalation spray products that are currently marketed have the following characteristics. Aqueous drug solution droplets resembling a nebulized aerosol, a longer spray duration that is approximately 10 times that of an MDI product, and a slower moving aerosol with a velocity that is approximately 1/10 that of an MDI product. These characteristics may impact how the inhalation spray is used as well as its performance. Development of bioequivalence recommendations for inhalation spray products has been supported by GDUFA-funded research. To address the unique features of inhalation spray products, the current method being used is based on the aggregated weight of evidence approach. The weight of evidence approach is comprised of in vitro BE studies and pharmacokinetic or PK/BE studies. The in vitro BE studies are single actuation content, aerodynamic particle size distribution, spray pattern, plume geometry, priming and repriming, spray duration, and spray velocity. While most of the in vitro BE studies shown in the list have been recommended for other types of orally inhaled drug products, the spray duration and spray velocity studies are specific to inhalation spray drug products. The in vivo BE studies are a comparative PK study with fasting single-dose two-way crossover design in the general population. All strengths of the product are to be tested where applicable. It is recommended for a generic product to have formulation sameness based on the qualitative and quantitative comparison to the reference listed drug product, as well as device similarity. Each component must be satisfied for BE to be established. No comparative clinical endpoint or pharmacodynamic BE study are recommended for inhalation spray drug products, although these studies are recommended for other types of orally inhaled drug products. Spray velocity studies require some unique considerations, which we will be discussing next. The draft product-specific guidance recommendations for inhalation spray products are available on the FDA website for generic drugs. The design of the spray velocity study recommended in the product-specific guidance is as follows. The spray velocity test should be performed at the beginning and end life stages of the product. High-speed imaging, particle individual symmetry, phase Doppler anemometry, or other suitable methods may be used to determine spray velocity. The equivalence is based on population bioequivalence, or PBE, or other statistical analysis of plume front velocity. Plume front velocity is the velocity at the front edge of the aerosol cloud. Since the velocity is dependent on the distance from the nozzle, one distance between 8 to 12 centimeters from the nozzle is recommended for this assessment. If other statistical analysis is used, it should be adequate considering the purpose of the study and scientifically justified. Full plume front velocity versus distance data should be submitted as supportive evidence for equivalent spray velocity, which may be used to confirm the appropriate distance from the nozzle used for collecting recommended plume front velocity data. The current labeling for marketed inhalation spray products states that the device uses mechanical energy to generate a slow-moving aerosol cloud of medication. Spray velocity is expected to influence drug deposition in the mouth and throat regions and in the lungs. Spray velocity may be determined by utilizing high-speed video recording, or HSV, as may be found in the literature in this study by Hottrainer et al. A high-speed camera was used to predict the plume front velocity by photographing the developing aerosol cloud using the dark field technique to illuminate the spray. These images captured the time evolution of the spray. Data were collected from the moment the spray begins to develop to the last moment when the spray is formed at the nozzle outlet. The plume tip displacement relative to the inhaler nozzle was measured for each frame using a measuring scale. Plume front velocity was determined using a curve fit of the distance versus time data and taking the time derivative of the resulting polynomial equation. The plume front velocity was determined for the 10 centimeter distance as measured from the nozzle outlet using interpolation. Spray velocity may be determined by utilizing particle individual symmetry, or PIV, as may be found in the literature in the study by Tamura. The laser light sheet illuminates the spray. Data were collected from the moment the spray begins to develop to the last moment when a spray was formed at the nozzle outlet. The plume tip displacement relative to the inhaler nozzle was measured for each frame using the image calibration scale. Plume front velocity was determined using a curve fit of the distance versus time data and calculating the time derivatives at distances of 8 and 10 centimeters from the nozzle outlet. We will continue discussing the use of PIV to determine spray velocity using a case study. For a case study, we will be discussing the evaluation of spray velocity from tiotropium bromide inhalation sprayed metered inhalers. The FDA is currently executing a spray velocity study for tiotropium bromide inhalation metered spray as part of GDUFA-funded regulatory science research on spray velocity characterization. Plume front velocity, or the velocity at the front edge of the aerosol cloud, was assessed using PIV or HSV recordings of a tiotropium bromide inhaler. An example of the experimental setup for the PIV studies is shown in the image at right. The PIV image sequence provides images captured from the moment when the spray begins to develop to the last moment when the spray is formed at the nozzle outlet. These images are shown below. The methods for determining plume front velocity are similar to the approach of Tamura. The PIV image sequence was used to visually determine the time duration between the beginning of the spray at the nozzle outlet and when the plume tip reaches the edge of the camera frame. Software was used to track the movement of the front edge of the plume. The elapsed time and the distance traversed by the plume front is collected and plotted. The plot shows the distance traveled by the plume front as a function of time. The velocity is determined by calculating the slope of the distance versus time curve using a backward differencing scheme. Plume front velocity was determined at 6, 8, 10, 12, and 14 centimeters downstream of the nozzle outlet. To determine the plume front velocity at a specific distance, linear interpolation was used. The distance versus time curves were assessed with HSV and PIV and compared with literature data for tiotropium bromide inhalation spray metered inhaler. The distance versus time curve is consistent with the work of Trainer et al. For an inhalation spray using the Respimat inhaler. The plume front velocity measurements were consistent with findings by Tamura. A comparison cannot be made for plume front velocity data in Trainer et al. Because a tiotropium bromide inhalation spray metered inhaler was not tested in the study. The selected distances of 6, 8, 10, 12, and 14 centimeters were assessed on the velocity versus distance curve to find the most appropriate distances for assessment of spray velocity. An 8 to 12 centimeter distance range was selected due to relatively low variability in the region beyond 6 centimeters and comparison to literature data. In addition to plume front velocity data at the selected distance used for PB analysis, the draft product-specific guidance on tiotropium bromide inhalation spray also recommends prospective applicants consider submitting full plume front velocity versus distance data as additional supportive information. The design of the spray velocity study recommended in the product-specific guidance permits other methods of determining spray velocity. Any suitable method may be used provided the methods are appropriate for assessing the spray velocity and adequately sensitive in detecting spray velocity differences between the test and reference inhalation sprays. It is encouraged that prospective applicants submit a pre-ANDA product development meeting request to discuss their method development and statistical analysis plan for spray velocity study designs. We refer you to the FDA guidance for formal meetings between FDA and ANDA applicants of complex products under GDUFA. The meeting package should include details on the method development and validation, preliminary studies, if any, and a proposed statistical analysis plan. In conclusion, inhalation sprays are propellant-free, typically aqueous-based formulations containing drug products intended for delivery to the lungs by oral inhalation for local and/or systemic effects. Establishment of bioequivalence for inhalation sprays is based on the weight of evidence approach consisting of in vitro and in vivo studies. For spray velocity studies, the plume front velocity at one selected distance between 8 to 12 centimeters from the nozzle should be measured using a suitable and sensitive technique. Full plume front velocity versus distance data also serve as additional supportive information. Prospective applicants are encouraged to submit a pre-ANDA product development meeting request for discussing with the agency their development program for inhalation spray products. For future work, the FDA is currently working on completing the RLD arm of the spray velocity study for tiotropium bromide inhalation metered spray. The data from this study will be published when available. Here's a challenge question for you. Plume front velocity is defined at what specific location? A. Velocity at the front edge of the aerosol cloud. B. Average velocity along the center line of the fully developed plume profile. C. Maximum velocity measured at a central location downstream from the inhaler nozzle. Or D. Average velocity measured only at the inhaler nozzle. The correct answer is A, velocity at the front edge of the aerosol cloud. Here's a challenge question for you. Plume front velocity may be determined from which of the following data sets? A, PIV data, B, high-speed imaging data, C, both A and B, or D, neither A or B. The correct answer is C, both A and B. For our call to action, if you have questions regarding a spray velocity study, consider requesting a pre-ANDA product development meeting. I would like to thank my colleagues who helped contribute to this slide presentation. This is a list of applicable resources.
Thank you. I'm glad to be here and talk to you about the first generic drug approval, and this is a case of budesonide and formaldehyde fumarate dihydrate inhalation aerosols, a generic version of Symbicort pressurized metered dose inhaler from the bioequivalence perspective. First, I'll introduce you some background information, give you an overview of locally acting orally inhaled or nasal drug product, OINDPs, and FDA's weight of evidence approach and equivalence assessment for one category of OINDPs, a suspension metered-dose inhaler. Then I'll discuss this case study that used alternative approach, a sensitivity analysis to support a non-quantitative or non-Q2 formulation of generic MDI product. Following this presentation, I hope you will be familiar with the bioequivalence studies recommended in the product-specific guidance for this MDI product. The OINDPs on the market can be classified into 5 categories: MDIs, DPIs, nebulizers, soft mist inhalers, and nasal spray or nasal aerosols. Each category has its distinctive features, and they are complex drug and device combination products. Their aerosol performance depends on the interaction between the formulations and delivery devices. In turn, a successful drug administration depends on the user interface and patient's inhalation maneuver. Most OINDPs are locally acting drugs for the treatment, for example, asthma and COPD. Locally acting means that the target efficacy does not rely on systemic circulation. Based on the drug mechanism of action, the OINDPs can also be roughly classified as beta-2 agonist, anticholinergics, corticosteroids, and a combination of either beta-2 agonist with anticholinergics or corticosteroids. Here, we will focus on MDIs. Among them, MDIs are drug formulations either suspended or dissolved in an inert propellant, typically hydrofluoroalkane, HFA, and sealed in a canister. MDIs have a metering chamber and an actuation mechanism. When actuated, the vapor pressure forces the pre-metered dose out of the actuator nozzle to form a respirable plume. Then an actuation-inspiration coordination is required to get correct aerosol dose into the respiratory tract or lungs. Because there are numerous uncertainties in the complex drug-device combination products, including MDIs, we recommend aggregate weight of evidence approach for the generic drug bioequivalence. They include 4 coherent elements: equivalent in vitro performance testing, including single actuation content, aerodynamic particle size distribution study, spirit pattern, plume geometry study, priming and repriming studies, and equivalence systemic exposure based on in vivo pharmacokinetic data, and equivalent local delivery based on a clinical endpoint study or alternative approach, and formulation sameness and device similarity between proposed test product and reference product, including device component sameness and formulation qualitative and quantitative or Q1, Q2 sameness. These 4 elements are evidence to support both equivalent efficacy and equivalent safety. None of them, when standing alone, would provide adequate assurance of the equivalence in both safety and efficacy. With these background information in mind, I will discuss a special case on approval of a non-Q2 formulation generic version of Symbicort. The reference listed drug Symbicort was approved in July 2006. This suspension MDI is indicated for the treatment of asthma in patients 6 years of age and older and maintenance treatment for COPD. The IOD has 2 strengths, 80 or 160 microgram of budesonide per actuation and a fixed strength of 4.5 microgram of formoterol fumarate per actuation. The product-specific guidance for this drug product was published in 2015. The agency recommends 5 in vitro studies on both strengths, including SAC, APSD, spirit pattern, plume geometry, priming and repriming. in vivo PK studies on the fasting condition on both strengths, and one comparative clinical endpoint study on the lowest strength in asthma patients. In addition, the formulation sameness and device similarity is also recommended. However, during the review of this application, it was found that the proposed formulation was Q1 the same, but not Q2 the same as the RD, because one of the excipients, here we designate it as excipient A, has percent difference greater than 5% between test and reference product. The excipient A did not exceed previously approved drug product intended for inhalation. This means there's no safety concern. The applicant submitted all 5 in vitro studies, in vivo PK studies, and comparative clinical endpoint studies as recommended in the PSG. In order to support non-Q2 formulation, the applicant conducted additional in vitro sensitivity studies to evaluate the impact of varying amount of excipient A on aerosol performance. The study report demonstrated the formulation understanding based on pharmaceutical development data and elaborate on how the proposed test formulation was selected. The sensitivity studies include the above-mentioned 5 in vitro studies, but conducted on 3 levels of the excipient A, lower than test, equal to, and higher than ROD. These 5 studies were designed to be 3 batches with 3 levels of excipient A for each strength, and 10 samples in each batch. PBE analysis were conducted to evaluate the equivalence between test and reference. In addition, 2 suspension formulation characterization, including morphology and stability studies, were also conducted. The result of all in vitro studies and suspension formulation characterizations indicated that varying amount of excipient A did not affect aerosol performance. The morphology characterization and suspension stability show similar results among test formulation with varying amount of excipient A and ROD. This slide shows a hypothetical data set for SAC results. The study was conducted at the begin, middle, and end life stages, and a mean delivered amount was presented as a percent label claim. You can see that the mean delivered budesonide and formaldehyde fumarate from the test product at 3 life stages was comparable to that of the RD product, and the mean delivered amount was roughly within 95 to 105% of the percent label claim in all cases. The study passed PBE analysis. Similar outcomes were also observed for priming and repriming after non-use and dropping. Eventually, the first generic of Symbicort to treat asthma and COPD was approved on March 15, 2022. So, a non-Q2 application may be submitted to FDA for MDI, and this generalized to all OINDPs. However, sufficient data and comprehensive information are needed to justify the impact of the formulation differences on bioequivalence and safety. For example, if SAC study shows consistently lower percent label claim for the test product than the RD product, as the graph shows here, it would likely fail to meet the PBE criteria. Then, an investigation should be conducted to determine whether this lower delivered dose is caused by the differences of the formulations between the test and the RD products, after excluding numerous confounding factors that may also affect aerosol performance. In summary, FDA recommended the weight of evidence approach to evaluate bioequivalence of locally acting OINDPs. A case study for a non-Q2 MDI application is briefly touched. Typically, a Q1, Q2 same formulation is recommended. Scientific justification with appropriate data and scientific explanation are recommended for the development of non-Q2 formulations. FDA recommends the applicant to follow product-specific guidance for efficient generic ONDP product review. If the applicant wants to use novel B approach, for example, non-Q2 formulations, in this case, FDA provides multiple communication channels, such as control correspondences, product development meeting, pre-submission meetings, mid-cycle review meeting, and enhanced mid-cycle review meetings, and post-complete response scientific meetings at various stages to address applicants' questions. We strongly encourage early communication with FDA, which could help to clarify the FDA's expectation for prospective applications early in the product development and assisting in submitting a comprehensive application. I'd like to thank you for the opportunity to present this talk to you. Special thanks to our Office of Bioequivalence Director, Dr. Partha Roy, Division of Bioequivalence III Director, Dr. April Brady, Deputy Director, Dr. Kiran, Dr. Li Gong, and my colleagues and inhalation experts from different disciplines in the FDA, including Dr. Fan Yuan from the Office of Pharmaceutical Quality, Dr. Brian Newman, and Dr. Elizabeth Besky from Office of Research and Standards, and Dr. Channing Gore from the Office of Testing and Research. Poll question number 1. What are included in the FDA's weight of evidence approach for orally inhaled or nasal drug products? A. Equivalent in vitro performance. B. Equivalent systemic exposure. C. Equivalent local delivery. D. Formulation and device sameness. E. All the above. Take a second to consider which answer you think is correct. If you choose E, all of the above, that's the correct answer. Poll question number 2. How many types of ONDPs are available on the market? A. Dry powder inhaler. B. Pressurized metered-dose inhaler. C. Nebulizer. D. Soft mist inhaler. E. Nasal spray, nasal aerosol. F. All the above. Take a second to consider which answer you think is correct. If you choose F, all the above, that's the correct answer. That concludes my presentation. I will be back shortly to answer questions. Thank you for your attention.
Good morning, everyone. I'm Feng Yuan, a senior chemist in the Office of Life Cycle Drug Products under the Office of Pharmaceutical Quality. Dr. Zhen Xu just went through the agency's bioequivalence approach for metered-dose inhaler and took the first approved generic version of Symbicort for a case study. Let's switch gears and discuss some recommended considerations from quality perspective. Speaking of quality, everyone deserves confidence in their next dose of medicine. And our day-to-day work in the Office of Pharmaceutical Quality is to ensure every single marketed pharmaceutical product's quality, so to assure the availability, safety, and efficacy of every dose taken by our patients. In this 15-minute presentation, I will discuss potentially overlooked issues in formulation development of generic suspension-based MDI products and highlight a few product characterization studies helpful to verify product design providing consistent delivered dose or identify the risk in product design causing inconsistent delivered dose. Budesonide and formoterol fumarate dehydrate inhalation aerosol has two strengths, and the reference product Symbicort was approved in 2006, and the first approved generic version, Brina, was approved in 2022. A few weeks ago, Viatris just announced the launch of the product. It is a suspension formulation consisting of two micronized drug substances co-suspended in the propellant-based formulation. The formulation also contains a small amount of suspending agent and lubricant. And this table here provides the metered dose by the valve and the delivered dose from an actuator for both strands. The co-suspension-based MDI product is a perfect representative of the challenging dosage form that I will discuss in this presentation. This is a cross-sectional diagram of a metered dose inhaler, and the canister contains the bulk formulation, where the concentration of the active ingredient is the assay of the product, and the total quantity of the bulk formulation filled inside the canister is the net content. And the valve outline is in green color, and in the middle of the valve, the small chamber here is the valve metering chamber. The size of the metering chamber determines the amount of the bulk formulation metered by each actuation, where the quantity of the active ingredient contained in the metered formulation is the metered dose. And the actuator outline is with the brownish red color. And at the bottom of the actuator, the extended part is the actuator mouthpiece. And inside the actuator here, the little space here is the actuator sump, which provides the moving chamber or moving space for the valve stem during the actuation. And close to the end of the actuator sump, the tiny opening here is the actuator orifice. So when a metered dose inhaler is in rest position, the metering chamber is open towards the formulation reservoir, and upon actuation, the metering chamber became closed towards the formulation reservoir and open to the atmosphere, which leads to the expansion of the metered formulation moving through the valve stem and subsequent atomization from the actuator orifice. So the quantity of the active ingredient discharged from the mouthpiece is the delivered dose, and the target delivered dose defines the strength of the product. So one of the major challenges in developing suspension-based MDI formulation is to maintain the consistent delivery dose through unit life over shelf life. And the CQAs, including assay, net content, metered dose, all can impact the delivery dose uniformity. Here's an example of typical generic MDI product composition table. And the composition table captures a number of CQAs we just discussed. The quantity of the active ingredients is the target delivery dose, and also the strength of the product, which is always same as the RODs. And the quantity of the active ingredients metered by the valve is the metered dose. And the total quantity of the formulation metered by the valve is the valve delivery, and also called the shot weight. The metered dose and valve delivery may or may not be same as the RODs, because they're also impacted by the size of the valve metering chamber and the density of the proposed formulation. And the metered dose is also impacted by the assay, or the canister concentration of the active ingredient. And the total quantity of the formulation filled in the canister is the net content. And that content also may not be same as the RODs because it's impacted by the size of the selected canister in the product design. The tail-off characterization studies should be used to justify the proposed minimum net content. And all of these 4 CQAs are specified in product label, but the quantitative values in the rest of the cells in this table are not public available information. So during formulation development, one of the big challenges is to determine the target canister concentration of active ingredients, or the assay of MDI. And the target canister concentration of inactive ingredients are subject to Q1, Q2 sameness evaluation for generic product. During formulation development for MDI product, most generic applicants choose to develop a formulation that is Q1, Q2 same as the ROD. But in the case where the proposed formulation is not Q1, Q2 same as the ROD, then additional supporting data will be needed to demonstrate that the deviation does not impact the product quality and performance. You may refer to Dr. Genshi's presentation regarding the required supporting data from bioequivalence perspective. And from the quality perspective, the type and amount of supporting data depends on the non-Q1, Q2 same inactive ingredients function in the formulation. I will give a brief example when we discuss the product characterization studies. The target canister concentration of the active ingredient is determined based on the reverse engineering study results from the ROD formulation. Same as other dosage forms, the agency discouraged use of overage as per ICH Q8 guidance. But if the development study suggests that an overage is necessary, then applicants need to provide adequate justification and supporting data. For example, unavoidable drug loss during manufacturing process that may compromise the delivery dose uniformity. But here, I also would like to recommend our applicants carefully investigate the potential drug loss due to drug deposition on device constituent parts during use. If you recall, when we discussed the operating mechanism of MDI, upon its actuation, the formulation is metered by the metering chamber and then passing through the valve stem, actuator sump, actuator orifice, and mouthpiece before inhaled by the patient. So the drug particles may deposit at the inner surface of those device constituent parts, and the level of deposition vary from dose to dose through the unit life, usually higher at the beginning of the unit life because all the surfaces are clean, not occupied by the drug particle residue. And depending on the geometric dimension, materials of construction, and coating treatment of the selected device constituent parts, the level of deposition may vary as well from product to product. We're halfway through this presentation. Let's take our first challenge question. Which of the following critical quality attributes can impact the delivered dose uniformity of MDI products? A, assay, B, leachables, C, net content, and D, metered dose. The correct answer is assay, net content, and metered dose. The 2018 Draft Quality Guidance recommends a list of product characterization studies for the purpose of supporting robustness of to-be-marketed products' performance and labeling statements. We recommend applicants to conduct these studies using at least 3 exhibit batches that represent the to-be-marketed product, and you may refer to the draft guidance for additional details on the purpose and recommended design for individual study. But here, I'd like to emphasize that these product characterization studies can be more than a tool verifying the final or close to final design of your proposed product. And I will highlight a few characterization studies as examples to further elaborate how we can leverage them to identify the risk in product design at early stage of your development program. So drug deposition, mouthpiece, and/or accessories characterization study in conjugation with cleaning instruction study can be helpful to assess the trending of delivered dose through in-use period due to drug deposition on the internal and external surfaces of individual device constituent parts. To serve this purpose, we strongly recommend applicants to assess the trend of drug deposition. Following the label instructed cleaning intervals through unit life and include all critical device constituent parts involved in the flow pathway of the metered drug formulation during the actuation. And for the cleaning instruction study, we recommend applicants to strictly follow the cleaning intervals through unit life and cleaning procedure described in the label. And based on the study results, carefully select critical device constituent parts and optimize the product formulation to minimize the impact of drug deposition on device constituent parts on the delivered dose uniformity through unit life. The profiling of actuations near device exhaustion study, also called tail-off study. The study can be used to justify the proposed minimum net content of the product to ensure consistent and adequate product performance through the label claimed number of doses. And the effect of storage and shaking study is to demonstrate the adequate product performance according to the label instructed shaking procedure before each actuation. And in addition, if an applicant proposed a generic formulation contains surfactants not Q1, Q2 same as the ROD, you may use the study results as part of your justification for the deviation in the proposed formulation. Now, it's time to take our second challenge question. Which of the following statements regarding product characterization studies are correct? A, to support labeling instructions, and B, to verify robustness of product design. C, one-time studies never needs to repeat. D, either using development badges or exhibit badges. The correct answer is A and B. It's not routine studies. However, if you make changes to your product design, you may need to repeat the product characterization studies depending on the level of the proposed changes. Here's a list of the related guidance and compendial standards for your reference. Take-home messages. To minimize the risk of inconsistent delivered dose through unit life over shelf life of generic suspension-based MDI products, we recommend applicants cautiously select device constituent parts to minimize the drug particles built up during use, and do not overlook potential impact of drug deposition in device constituent parts on the delivered dose uniformity. And we encourage applicants to leverage certain product characterization studies to quickly identify the risk in product design that causing inconsistent delivered dose and optimize the product design at early phase of development if possible. That brings us to the end of my presentation, and thank you all for your attention. Let's reconnect during the panel discussion.
Hello, my name is Andrew Clareman, and I am a senior physician in the Division of Therapeutic Performance 1 within the Office of Generic Drugs. Today I'm going to discuss the post-approval impact of generic fluticasone propionate and salmeterol inhalation powder. The learning objectives for my talk include describing the high costs of inhalers in the United States and the effect on patient outcomes, identifying the difference between an approved generic through the ANDA pathway and an authorized generic, and describing the post-market impact of ANDA-approved generics and authorized generic inhalers. The U.S. per capita prescription spending is higher than comparable countries. As can be seen in this figure from a Peterson KFF study, comparable countries have spent significantly less money on prescription drugs since 2004, and the difference appears to be accelerating. Looking specifically at inhalers, these are a substantial cost to the U.S. healthcare system. Between 2012 and 2018, Medicaid spent over $26 billion on inhalers, and this annual spending has increased over $2 billion, or 120%, during this time. Inhaled corticosteroid long-acting beta agonist, or ICS-LABA, combination inhalers represented 53% of that spending and increased over 600%. Finally, almost 7% of the $168 billion spent by Medicare Part D in 2018 went toward inhalers for asthma or COPD. In U.S. patients who rely on inhalers, cost barriers are associated with uncontrolled asthma in both adults and children, at least one asthma-related hospitalization in elderly patients, and in COPD patients, a higher likelihood of abandoning inhaler therapy. The FDA conducted an investigation looking at the impact of generic inhaler approval on uptake and costs. This investigation included looking at Wixela Inhub, a generic dry powder inhaler that references Advair Diskus and was approved in January 2019 through submission of an ANDA. Additionally, an authorized generic of Advair Diskus entered the market in February 2019. FDA investigators published this research that investigated the medication cost savings as well as the utilization of generic ICS-LABA products in the United States using IQVIA data. IQVIA provides information about invoice transaction pricing of pharmaceutical sales as well as dispensing. In this study, the investigators hypothesized that introduction of the first approved generic would lead to cost savings compared to an authorized generic. Let's take a moment to look at the difference between authorized generics and approved generics. Authorized generics are identical to a brand name drug. They are marketed under the same new drug application as the brand name, but do not display the brand name on the product. Comparatively, approved generics may have differences from the brand name drug, but are required to demonstrate bioequivalence through an ANDA submission. So how do authorized generics impact the marketplace? In 2011, the US FTC issued a report that found that authorized generics impact the marketplace in several ways. This included that authorized generic competition during the 180-day exclusivity period for an approved generic led to somewhat modestly lower prices of 4 to 8%. However, they also found that authorized generics have a substantial negative effect on the revenues of competing generic firms. Finally, they found that brand companies may use the threat of issuing authorized generics to prompt generic companies to delay their products, and this may lead to limited market competition and higher prices for consumers. Moving back to the FDA's investigation, they looked at a few key products within this drug class. First, they looked at the approved generic referencing Advair Diskus, which was introduced in 2019 following ANDA approval. And around the same time, in February 2019, an authorized generic of Advair Diskus was also introduced. In January of 2020, an authorized generic of Symbicort was introduced to the marketplace, providing a useful comparison. Let's first look at the wholesale units of ICS/LABAs between 2015 and 2020, which was the FDA's study period. This graph shows the wholesale units over time, with the red line on top representing the total units. The first dotted line represents when the Advair generics were introduced in early 2019, and the second dotted line represents when the Symbicort authorized generic was introduced. Now let's take a closer look at the time before generic approval. This graph shows the wholesale units between 2015 and 2018 prior to the release of the generic Symbicort and Advair products. The red line represents the total units of the ICS/LABA class, which you can see is gradually increasing over this time. The Advair brand name product is gradually decreasing, and this is likely driven by a gradual increase in the uptake of a competitor brand name product, Bioequivalence. Now let's look at what happened after generic approval. This graph shows the wholesale units between 2019 and 2020 for the Advair Diskus and its associated generics. As you can see, in early 2019, when the generics were approved, there was a rapid decrease in the wholesale units of Advair Diskus brand name, while there were increases in the wholesale units of the authorized generic, as well as the approved generic. Notably, the total wholesale units stayed relatively stable. Similarly, looking at the Symbicort brand name as well as authorized generic, the total units stayed relatively stable, but in early 2020, when the authorized generic was released, you saw a decrease in the wholesale units of the brand name and a corresponding increase in the authorized generic. Now let's look at cost. Here's a graph of the wholesale cost of ICS/LABAs between 2015 and 2020. Again, the first dotted line is when the Advair generics were introduced, and the second dotted line is when the Symbicort authorized generic was introduced. Again, we're going to look at the time period before generic approval. So this graph shows wholesale costs between 2015 and 2018. As you can see, the total wholesale cost was increasing relatively quickly. However, the wholesale cost of Advair brand name was decreasing. So the increase in wholesale cost was likely driven primarily by the uptake of the competitor brand name, Bioequivalence, as well as Symbicort to a lesser extent. Now let's look at after generic approval. Here's wholesale cost of Advair Diskus and its corresponding generics between 2018 and 2020. As you can see, in early 2019, there was a rapid decrease in the total wholesale cost of this product family. This was primarily driven by a rapid decrease in the wholesale cost of Advair Diskus brand name, with a corresponding small increase in the wholesale cost of the authorized generic as well as the approved generic. Similarly, we look at the wholesale cost for the Symbicort family of products. In early 2020, when the authorized generic was introduced, we see a corresponding decrease in the wholesale cost of the brand name Symbicort, along with an increase in the wholesale cost of the authorized generic. However, there is not much change in the total wholesale cost for this product family. Now that we've looked at wholesale units and cost independently, let's put the data together. This graph shows the wholesale units of the Advair Diskus family of products. As we noted before, the total wholesale units stayed relatively stable. However, we can see that beginning in early 2019, there was a rapid decrease in the total wholesale cost, as seen on the red line in the graph on the right. Therefore, despite relatively stable units being sold, there was a significant decrease in the total amount being paid for these products. Now for comparison, let's look at the Symbicort family of products. From 2018 through 2020, the wholesale units of the Symbicort family stayed relatively stable. And again, in early 2020, with the introduction of the authorized generic, there was a small decrease in the wholesale units of the Symbicort brand name. Looking at costs, the wholesale cost through the same time period stayed relatively stable. And despite the introduction of the authorized generic, there was no corresponding decrease in the total wholesale cost like we saw for the Advair family of products. Now let's put some numbers to the cost savings associated with generic Advair approval. The total cost for Advair and its generics decreased from $337 million in January 2019 to $233 million in January 2020, despite stable wholesale units. A similar corresponding decrease did not occur for Symbicort, despite the release of an authorized generic. The average unit cost for the approved generic of Advair was $115 compared to $169 for the authorized generic and $334 for the brand name. To put these cost savings into a more real-world context, let's look at GoodRx prices for zip code 20903, which is where the FDA headquarters is located in White Oak, Maryland. This was accessed on June 27, 2023. As you can see on the left, for the Advair approved generic, Wixela InHub, the lowest price available was around $75. By comparison, the Advair Discus brand name for the same dose, the lowest price was about $385. To summarize, the U.S. spends a lot of money on inhalers, and high inhaler costs are associated with worse patient outcomes. The FDA investigation described in this talk found reduced costs with the ANDA approval of a generic referencing Advair. However, introduction of an authorized generic for Symbicort had limited impact on costs compared to the release of the approved generic for Advair. Now let's move to the challenge questions. Challenge question 1. According to a CDC study, U.S. asthma patients 65 years of age and older who had at least one asthma-related hospitalization in the past year were more likely to report A. Pets in the bedroom, B. Cost barriers to obtaining medication, C. Secondhand smoke exposure, or D. Obesity. The answer is B. Cost barriers to obtaining medication. Challenge question number 2. According to an FDA investigation, introduction of an approved generic ICS/LABA through the ANDA pathway resulted in significant cost savings, whereas introduction of a blank did not. A. Nebulized formulation. B. Companion app to monitor inhaler adherence. C. Authorized generic. Or D. Leukotriene receptor antagonist. The answer is C. Authorized generic. Here's a list of my sources for reference. If you have any questions, please submit them for the question and answer session that will follow. As a final thought, I want to leave you with this quote from one of my OGD colleagues. On behalf of OGD, I want to thank the audience and our stakeholders for their hard work bringing safe and effective generics to the marketplace. Thank you.
Thank you all for the great presentations. As another reminder, if you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. In addition to our presenters, we welcome the following panelists: Dr. Srinivas Behera, Chemist, Division of Immediate and Modified Release Products 3 and OPQ; Dr. Tien Ma, Senior Staff Fellow, Division of Bioequivalence 1, Office of Bioequivalence and OGD; and Dr. Elizabeth Bielski, Senior Pharmacologist, Office of Research and Standards, and OGD. Looks like we have some questions coming in right now. The first couple of questions will be directed to Dr. Stephen Chopsky, and here is the first question for Dr. Chopsky. Why is the current thinking on regulatory significance of spray velocity for orally inhaled drug products limited to only the plume front velocity rather than the temporal and spatial development of the spray plume velocity?
Yes, thank you, Ray. That's a very good question. So plume front velocity is the velocity at the tip of the plume, which I discussed in my talk. So plume front velocity provides a more appropriate metric for velocity with minimal variability as compared to other velocity measurements of the plume. With regard to temporal and spatial development of the plume, there can be a lot of variability in the internal, basically the internal velocities of the plume. If you were to look at local velocities, the velocity at the tip of the plume provides a very regular decline in velocity and can be effectively measured. Thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Chopsky, and here is the next question. Can you elaborate on what is the role for spray velocity measurements for demonstrating bioequivalence of meter dose inhalers?
Yes, thank you, Ray. That's a very good question. So the product-specific guidance documents, or PSGs, for all solution-based MDI products provide for an alternative bioequivalence approach to the comparative clinical endpoint study. So these additional supportive in vitro studies may include studies that support the characterization of emitted aerosol sprays with respect to velocity profiles and also evaporation rates, which would include spray velocity measurements. So I would note that spray velocity measurements, if they're used in an alternative BE approach, would be best used as in a supporting role rather than as pivotal evidence for establishing BE. I'd also like to point out that the Center for Complex Generics actually conducted a workshop back in April, so earlier this year, and highlighted the use of spray velocity measurements in a supportive role for BE. If anyone's interested, you can find the proceedings for the workshop on the CRG website. Thank you.
Thank you for responding to that question, Dr. Chopsky. And we have one more question for you in this round, and here's the question. What is the purpose of the FDA working on the reference-listed drug arm of the spray velocity study as described in the product-specific guidances for all inhalation spray products?
Yes, thank you for the question, Ray. So the FDA is working to produce a spray velocity reference data set based on the RLD product for spray inhalation spray products. The study results may provide a very useful reference for potential ANDA applicants who are considering conducting a spray velocity study. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few questions that just came in for Dr. Zhen Zhu. And here's the first question for Dr. Zhu. What should be submitted to meet the standards for the proposed non-Q2 formulation product? I'm sorry, Dr. Zhu, we're not able to hear you at this time. We're going to have our tech team check in with you and check your volume. We're going to move on to our next panelist, Dr. Fang Yuan. And here is the question for Dr. Fang Yuan. Does the agency expect that the product characterization studies to be performed on at least 3 batches of each strength or a total of 3 batches across the proposed strengths?
Thank you. This is a great question. So it's a very common question that we encounter during the pre-NDA phase as well. So in general, we do expect that the applicant should submit product characterization studies on at least 3 batches of each strength. the reason is that we recommend at least 3 batches is to evaluate the batch-to-batch variation and the difference in strengths between strengths. The level of the variability across strengths may not be the same, and the batch-to-batch variability from the product components may have different level of impact on the end product batches. Thank you.
Thank you for responding to that question. We have another question that came in for Dr. Yuan, and here's the question. If changes have been made to the product design after all product characterization studies have been completed, does the applicant need to repeat all product characterization studies?
Thank you. Great question. There is no unique rule for that. In general, we do recommend the applicant to submit a risk assessment for the changes, and also and justification for the selected product characterization studies if they do not wish to conduct all product characterization studies. So it really depends on the level of changes. Thank you.
Thank you, Dr. Yuan. We have one more question for you in this round, and here is the question. Where in the ANDA submission should we include the device constituent parts, characterization studies, and cleaning instruction study?
Thank you. I'm not sure what the device constituent parts characterization studies meant for. If you are referring to the product characterization studies, then we do, in general, we recommend applicants to submit the product characterization studies, including cleaning instruction study in P2. But if you're talking about the information and related functionality studies on device constituent parts, then we recommend applicant to submit such information in P7. Thank you.
Thank you, Dr. Yuan, for responding to that group of questions. We're going to check back in with Dr. Zhen Zhu to see if we have connectivity with her mic. And here is the next question for Dr. Zhu. What should be submitted to meet the standards for the proposed non-Q2 formulation product?
Thank you for the question. So if a sponsor is interested in submitting a non-Q2 formulation, the sponsor should first provide an explanation why a Q1, Q2 formulation cannot be used and what is the rationale for selecting a non-Q2 formulation. Then a sponsor should demonstrate that non-Q2 formulation has no safety concern. And then after that, the sponsor should provide pharmaceutical development data, including in vitro testing of multiple drug to excipient ratio that encompass combinations below and above the ratios used in the TNR product. So this is to ensure that non-Q2 formulation does not impact the drug product performance. Thank you.
Thank you, Dr. Zhu. We hear you loud and clear. We do have a few more questions for you in this round, and here is the next question. Does the non-Q2 generic drug development strategy apply to all and orally inhaled drug products? And how about nasal non-Q2 generic drug products?
Yeah, thank you for the question. It depends. Orally inhaled products are complex and they have vast differences in formulation and device technology. So they may be passive inhalers that are highly dependent on the inspiration maneuver, or contain a propellant that requires hand-lung coordination. So the inhalation products have different indications that may target the locally or systemically. Or there are new formulation technologies used in getting respirable particles, or need to be considered, and they will be evaluated case by case. So scientific justifications with appropriate data and the explanations are recommended for the development of non-Q2 formulation, for example, those MDIs or DPI products. On the other hand, for the nasal product, for example, solution nasal product, the product-specific guidance generally have two options. The bioequivalence can be established from in vitro or in vivo options. If they are Q1, Q2, they can choose the in vitro options, and typically the SAC, priming, repriming, DSD, small particle by cascade impaction, spray pattern, and plume geometry studies. If non-Q2, an in vivo PK study is generally recommended to establish bioequivalence. Thank you.
Thank you, Dr. Zhu. We do have one more question in this round, and here's the question. Why does the applicant want to overcome the hurdles and develop a non-Q2 generic drug while the easier Q1, Q2 route is accessible?
Yeah, that's a good question. So following Q1, Q2 route based on the PSG recommendation, it should be straightforward, but there might be some issues like patent issues or some unknown issues. So that drive the applicant to choose non-Q2 route. However, applying a non-Q2 generic drug product would face many more scientific and regulatory challenges for sure. This is why it's important to communicate with the agency earlier, for example, through control correspondences or pre-ENDA meetings to discuss the matter like those proposed non-Q2 formulation in advance. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist. A few questions came in for Dr. Andrew Klurman, and here's the first question for Dr. Klurman. Do you have any data on how the marketplace has evolved since 2020 when the FDA study was completed?
Thank you for that question. So since the study was completed in 2020, we haven't done any formal analyses, but from what we can tell, the trends that we saw in the FDA study have stayed relatively stable. Thank you.
Thank you for responding to that question, Dr. Klurman. We've got a couple more questions, and here's the next question. Do you have any data on how the marketplace, sorry, I read that question. Go back to the first question just came in. What do you anticipate the impact of a second, third, etc. generic approval would be on the cost and uptake within a product family?
Thank you for that question. So what we anticipate is for subsequent generic approvals to continue to lower the cost of that product family. We do have data, and I believe there's a FDA publication from a few years ago that shows that we have increasing cost savings with second, third, and fourth generics compared to just that first generic. So within the past few years, we've had some additional approvals, and we intend to continue this research to continue to monitor the cost impact and the uptake impact that these generic approvals are having. Thank you.
Thank you for responding to that question. We have one more question for Dr. Klurman, and it is the following. How difficult would it be to do a similar cost impact analysis for other generics?
Great question. question. So similar to the last question, I think it's not necessarily a matter of difficulty. I think we have the data. It's just a matter of having additional products to do the research on. And now that we've had a few additional approvals of various orally inhaled drug products over the past few years, we do intend to continue this research and evaluate the continued cost savings. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a couple questions that came in for Dr. Srinivas Behara. And here is the first question for Dr. Behara. For an A&D submission, is it acceptable to manufacture one exhibit batch at commercial scale and the remaining 2 exhibit batches of the product to be manufactured at 1/3 of the proposed commercial scale?
Good morning, everyone. That's a very good question. 3 batches of drug product to be manufactured from 3 discrete lots of drug substance and critical excipients, and also device constituent part components. Among the 3 batches, at least 1 batch should be manufactured at the proposed commercial scale, and the other 2 batches at least at 1/3 of the commercial scale. All the batches of the drug products to be fully filled. For example, if we are talking about soft mist inhalers, drug product to be fully filled into cartridges. Therefore, the approach you have appears to be reasonable. If applicants have different strategies, we recommend requesting a pre-ANDA product development meeting. Hope this clarifies your question. Thank you.
Thank you, Dr. Behara. We do have one other question that just came in for you, and here is the question. From a chemistry standpoint, what are the resources for the product development of inhalation spray products?
That's another good question. The resources for the development of inhalation spray products would be MDI, DPI products quality considerations, which is April 2018 draft guidance. Product-specific guidance, if any, I would say USP 5, USP 601, and nasal spray and inhalation solution suspension, and spray drug products CMC documentation, which is July 2002 guidance. If you have any specific development question, we highly encourage you to request a pre-ANDA development meeting. Thank you.
Thank you for responding to both of those questions. Moving on to our next panelist. Got a few questions that just came in for Dr. Tien Ma. And here's the first question for Dr. Ma. What studies should be submitted to support the non-Q2 formulation for each of the solution meter-dosed inhaler, suspension meter-dose inhaler, dry powder inhaler, and soft mist inhaler?
Thank you for the question. So actually, Dr. Zhen Xu answered a very similar question earlier, and he provided the answer very nicely. So here I just want to emphasize that we still highly recommend Q1, Q2 formulations to be provided. But if it cannot be accomplished and you have to use a non-Q2 formulation, we recommend you to communicate to the agency earlier so that to have a better understanding and discussion about what studies or information is needed for your submission. And this can be done through the control correspondence, product development meetings during the pre-ANDA phase, and also meetings such as the mid-cycle review meetings or enhanced mid-cycle review meetings or post-CR scientific meetings. Thank you.
Thank you for responding to that question. We do have one more question from Dr. Ma. Here's the question. If the RLD product changes after the completion of the bioequivalence studies, do we need to repeat all BE studies?
Thank you for the question. This is a very nice question. So it really depends on the changes that the RLD product has. So it may be so the best way to address this is, so sometimes it is, you don't have to repeat any B studies if the change is very, very minor. And sometimes you need to repeat the in vitro B studies, or sometimes in vitro and in vivo study. It really depended on the changes. And based on the data you provided, additional B studies may be recommended. And again, we encourage you, once you identify that the RLD product has changed, you contact the agency as soon as possible. And you can propose, you can delineate what changes the RLD have, and also propose the studies that you think may address these changes. And also, if you think no changes need, no B study need to be conducted, we recommend you to also provide justifications for that. And we encourage you to contact us as soon as possible during the pre-ANDA phase through the control correspondence or product development meetings, or if you identify this after your ANDA has been submitted, you can contact the regulatory project manager regarding how to proceed in this case. Thank you.
Thank you for responding to those questions. Moving on to our next panelist. A few questions came in for Dr. Elizabeth Bielski, and here's the first question for Dr. Bielski. Will the B studies recommend change in the case where an approved MDI's generic propellant must be changed to a new and quote green propellant? Would this depend on whether the RLD has adopted a new green propellant, or said another way, should the generic sponsor plan to adopt green propellants be without regard to the RLD?
Thank you for that question. That's a wonderful question and a really good hot topic currently in the space. I'd say right now, as it currently stands, a lot of the green propellants, we would require safety data in order to assess them. So until a new drug product comes onto the market, we really have no pathway to approve generic versions. However, once we do get that safety data from the new propellants and a new drug, a product is approved using that type of propellant, then we may be open to really looking towards approvals of generic products. But we encourage folks to actually start adopting their bioequivalence pathway, even if the RLD doesn't necessarily transition. So it won't necessarily depend on the RLD. We look at both pathways, and if at that point we are open to kind of looking at the product-specific guidances for those products. So the bioequivalence studies we may recommend will most likely follow those, but we may also ask for additional data or information at that time, depending on if the generic, what the generic product of interest is, what the RLD is, and all the specifics. So at that point in time, I think we highly encourage folks who are interested in this area to come with us through our product development meeting process through the pre-ANDA pathway to have those discussions. Thank you.
Thank you for responding to that question. The next question for Dr. Bielski is the following. How does FDA determine which products to target for GDUFA-funded research to generate PSGs? Is it based on demand or interest from the generics industry, pricing concerns from payers and patients, or lack of competition based on Orange Book listings?
Thank you for that question. That's a really great question. And I think the way we can address this is we really look at all of those factors combined. I'd say first we consider the complexity. So if the generic product is concerned to be complex, we focus our priorities towards those complex generics. Then we also take into consideration how much interaction we had with industry in terms of interest in developing those products. We also look at the lack of competition as well as what the marketplace looks like. So all these factors combined kind of lead or gear us towards which products or which PSGs, which type of research we will fund and use to help us develop those guidances. Thank you.
Thank you, Dr. Bielski. This is referring to a question, our next question for you, that was responded to by Dr. Zhen Zhu, and it's asking, Q1 and Q2, is it not mandated for these products? Is this a correct assumption?
Thanks for that follow-up question. And I would like to clarify, yes, our regulations don't mandate us or require us to have a Q1, Q2 formulation sameness for inhalation products. However, we do recommend them most of the time in a lot of our guidances for inhalation products. So again, it's not a requirement, but we do recommend it. And then, of course, as you've seen with the budesonide formoterol fumarate MDI case for that generic, as well as other cases, if you don't want to use a Q1, Q2 formulation sameness, you can come to us and we can discuss what additional data or information we may need for that product. Again, I think we did touch base on this earlier in our panel discussion. So if you do have questions about this, we are always welcome to hear your thoughts through our controlled correspondence pathway or a product development meeting. Thank you.
Thank you for responding to that question, Dr. Bielski. And we do have one really long question for you that just came in. I can repeat it if needed, and here is the question. A clinical endpoint bioequivalence study was used to establish bioequivalence for the recent generic approval of budesonide and formoterol fumarate dihydrate inhalation aerosol as recommended in the product-specific guidance. A few other PSGs for solution-based metered inhalation aerosol products provide recommendation for alternatives to the clinical endpoint studies. Can those possible solution MDI alternative approaches to the clinical endpoint study mentioned in those PSGs be applicable to development of generic budesonide and formoterol fumarate dihydrate inhalation solution, a suspension-based MDI product?
Thank you so much for that question. It's a really good one and probably complex one. I think the agency generally is open to alternative approaches to these cases. The PSGs that mention possible alternative in vitro, in vivo, and in silico methods can serve as a guide to building proposed alternative approach for a budesonide formoterol fumarate MDI case, even though those alternative approaches is not mentioned in the PSG. However, recommendations are product specific and are dependent on the dosage form and formulation. So other inhalation products like suspension-based MDIs and DPIs can have some influencing factors as well as additional factors that may add to their product complexity. Thus, additional factors may necessitate additional information and or other studies. Therefore, you should really evaluate the critical quality attributes of your proposed generic inhalation product and choose those alternative in vitro, in vivo, and or in silico methods that support characterization of such specific product interest. Also help maybe establish your in vitro and vivo correlations or help establish overall equivalence at the local site of action. In this case, it would be the lungs. The proposed studies should always work together to provide a comprehensive evaluation of local drug delivery for establishing equivalence. We really encourage companies interested in development of alternative approaches to what is recommended in the PSG for budesonide, formoterol fumarate MDIs case or other suspension MDIs or DPIs. You come to the FDA with a pre-ANDA product development meeting with your proposed alternative approach. Within that meeting, we really want to see your scientific justifications, your critical questions of interest, and that will allow us to have that proper discussion to see how that alternative approach may fit for your specific product of interest. Thank you.
Thank you for responding to that group of questions. Moving back to our previous panelists, we do have a couple of questions that came in from Dr. Fang Yuan. And here is the first question for Dr. Yuan. Dr. Yuan, can you bracket?
Thank you. It's a good question. We have encountered that during pre-NDA meetings and control correspondence as well. So in general, for complex products, the orally inhaled drug products, meter dose inhaler or dry powder inhaler are complex products. We generally do not recommend bracketing study design. Thank you.
Thank you for responding to that question, Dr. Yuan. We do have another question that just came in for you. Which specified microorganisms does FDA think needs to be controlled in the drug substance specification for MDI DPI products?
Thank you. Thank you for the great question. So I recommend you to refer to USP General Chapter 1111, Microbiology Examination of Non-Sterile Products, Acceptance Criteria for Pharmaceutical Preparations and Substances for Pharmaceutical Use. There are detailed recommendations. Thank you.
Thank you for responding to that question. Now, moving on to our next panelist, we do have a question that came in for Dr. Andrew Klerman, and here's the question for Dr. Klerman. Why doesn't the U.S. legislate for or incentivize the take-up of generics and thus bring the branded prices down?
Great question. So here at the FDA, I can't necessarily speak specifically to legislation. However, I will say that the American public does already have a pretty robust cost savings from approved generic drugs. There was a study by the Association for Accessible Medicines back in 2020 that found that 90% of prescriptions in the U.S. were for generic drugs, but that represented only 22% of the cost. However, in this case, for orally inhaled drug products, I think the issue is just lack of generic competition. But that is something we are certainly working very hard at and utilizing regulatory science to help push that field forward and bring more generic OIDPs to the marketplace. Thank you.
Thank you, Dr. Klerman. And just giving our audience a moment, if you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We're trying to take a look at a few more questions that are coming in to see if they're relevant to the presentations that have been provided thus far. All right, looks like we have a question that came in for Dr. Srinivas Bahara. And here is the question. How does the agency recommend sponsors for generic handle a change in formulation to a new green propellant from a quality perspective? And what issues should be considered?
That's a great question. It's in fact not a simple switch. There are a lot of challenges that one needs to be considered as we transition to low GWP propellants. Some of the considerations should be the drug solubility, potential for off-fall ripening for suspension-based formulations, for example, and drug substance stability profiles. We may have to rethink about, what are the studies that are required, but if you have any specific questions, we highly encourage you to request a pre-NDA development meeting. Thank you.
Okay, thank you for responding to that question. We're just surveying the remaining questions to see if there are any relevant to the presentations that have been provided thus far. Just a momentary pause for that to give our panelists a chance to review some more things that are coming in right now. So be right back in just a moment. Looks like we do have another question and it's for Dr. Andrew Klerman. And here's the question for Dr. Klerman. As a physician, what is your viewpoint regarding the therapeutic substitution of a lower cost product within the same category, such as ICS-LABA?
Thank you for that question. So speaking clinically, I mean, I think I would be inclined to first and foremost provide the patient with a drug that I think is going to treat their condition to the best possible ability. But I think I would also consider what's the financial cost of that treatment. And I would certainly take that into consideration and work with the patient to provide the best kind of value for their treatment. And interestingly, I think obviously I work for the Office of Generic Drugs. I have a great deal of confidence in our evaluation of bioequivalence, but there was actually just a paper published in the Annals of Internal Medicine by Dr. William Feldman from Harvard, and he did a real-world evidence analysis looking at the Advair Discus brand name versus the Advair Discus approved generic or Wixela Inhub, and compared a wide variety of patient outcomes between those two, and found they were essentially identical in terms of clinical outcomes. So I'd encourage you to look up that study. It's by Dr. William Feldman in the Annals of Internal Medicine, published last month. And I think studies like that should provide a great deal of additional confidence in the therapeutic equivalence of these products. So I would have no hesitation in prescribing these products. Thank you.
I want to thank all the panelists and the presenters for answering a numerous amount of questions that came in from our audience. We'll now enter into our morning break and return at 11:05 a.m. Eastern Time. Enjoy your break. Welcome back from the break. We hope you're able to warm your coffee or tea to enjoy during the next few presentations in session 6. Noteworthy complex generic drug approvals, oral locally acting, and oral suspension drug products. Our first presenter will be Dr. Weijie Sun, Senior Staff Fellow, Division of Therapeutic Performance 2 in the Office of Research and Standards OGD. Next, we'll welcome Dr. Manar Algavish, Senior Staff Fellow, Division of Therapeutic Performance 2, Office of Research and Standards in OGD. And our final presentation will be provided by Dr. Suman Dandamunde, Senior Pharmacologist, Division of Bioequivalence 3, Office of Bioequivalence in OGD. Please join me to welcome our first presenter, Dr. Weijie Sun.
Thank you for the introduction. This section is noteworthy complex generic drug approvals, oral locally acting, and oral suspension drug products. My presentation title is Bioequivalence for oral locally acting gastrointestinal drug products. For this presentation, I will give B recommendations for oral locally acting GI drug products and the rationales of B recommendations. For the learning objectives, the first one is to identify B approaches for oral locally acting GI drug products. The second one is to provide examples of oral locally acting GI drug products to establish BE. The third objective is to explain the rationales of B recommendations. For the locally acting GI drug product, drug substance may or may not be absorbed into the systemic circulation. So B recommendations are based on drug product properties and the product's mechanism of action. If a drug product is systemically absorbed, several in vivo study methods could be used to establish BE, including pharmacokinetic study, pharmacodynamic studies, comparative clinical endpoint B studies. Depends on the property of the drug substance or drug products. In vitro studies may be recommended. If a drug product is not systemically absorbed, or the drug absorption is very limited, it is not possible to measure drug concentration in plasma. So PK study cannot be used to establish BE between the test product and the reference standard. Instead, the PD study and the comparative clinical endpoint B study can be used. In vitro study may be recommended as well. As mentioned in the previous slide, because of different drug properties and the mechanism of action, methods to establish BE could be varied from products to products. For some of products, it may just recommend in vitro studies to establish BE, such as Survelamer products. For some products, they may have options of in vitro studies or in vivo studies. Vancomycin hydrochloride capsule is an example, or both in vitro study and in vivo studies are recommended, such as mesalamine products. Sometimes only in vivo studies are recommended for B establishment. Then this is the challenge question 1. If a drug product is not systemically absorbed, which of the following is not an adequate method to establish BE? A. PD study. B. In vitro study. C. PK study. D. Comparative clinical endpoint BE study. The correct answer is C. If a drug product is not systemically absorbed, it is not possible to determine PK parameters. So PK study cannot be used to establish BE between the test product and the reference standard. As mentioned before, the B recommendations are based on drug product properties and the product's mechanism of action. For the following slides, I will provide 4 cases for B establishment and the rationales of B recommendations due to the different properties and the mechanism of drug products. The first case is Cevimeline drug products. Cevimeline carbonate has two dosage forms, tablet and powder for suspension. Currently, there are 10 approved ANDAs for tablet and 5 approved ANDAs for powder for suspension. Cevimeline carbonate products are for the control of the serum phosphorus. Cevimeline carbonate is non-absorbent phosphate-binding cross-linking polymer. It contains multiple amines on the polymer backbones. These amines can bind to phosphate molecules through ionic and hydrogen bonding, and then forms insoluble complex. This insoluble complex will be eliminated in the feces. Therefore, Cevimeline carbonate can decrease phosphate concentration in the serum. Because of the mechanism of Cevimeline carbonate, the bioavailability of these products are very low, and the PK studies is not feasible to determine the BE for generic drug products. So instead of urine PK studies, in vitro studies are recommended to establish BE for Cevimeline carbonate products. In the product-specific guidance, it recommends API sameness, in vitro kinetics binding study, and in vitro equilibrium binding study. Cevimeline carbonate is a polymeric drug substance and is considered as a complex API. So the API sameness demonstration is recommended. API sameness determination is based on the totality of the evidence, such as manufacturing process of the API and the comparative physical-chemical characterizations. All of the factors will be taken into the consideration when developing B recommendations and reviewing ANDA products with complex API. For Cevimeline, the API sameness could be supported by API synthetic route and the comparative characterization results of the test and the reference APIs, including but not limited to degree of cross-linking, degree of the protonation, particle size, elemental analysis, Swirling index, etc. Because the mechanism of Cevimeline is to bind to phosphate molecules to have therapeutic efficacy, in vitro kinetic binding study and in vitro equilibrium binding studies are recommended to establish BE. For in vitro kinetic binding study, it can assess the rate of the binding and demonstrate how much time is required to reach the maximum binding where phosphate concentration is fixed in the binding solution. So it can identify that how much incubation duration is needed for equilibrium, which support the equilibrium binding study. The right figure is an example of kinetic binding profiles to compare test product and the reference standards. X-axis is the time, and the y-axis is percentage of phosphate binding. To conduct the in vitro kinetic binding study, at least at different lengths of the time should be used to incubate test product and the reference standard. The selecting time should show that the maximum binding is reached. So from the figure, you can see that the amount or the percentage of the phosphate binding increase when the binding time increase, and then it reaches to plateau, which is the maximum binding. For the test product and the reference standard comparison, the value of T to R ratio should be compared, but it is not for 90% confidence interval evaluation. For the in vitro equilibrium binding study, it can be used to calculate the affinity and the capacitive binding constants. In vitro equilibrium binding study is considered as the pivotal study and is used for biowaiver decision for the additional strengths. To conduct the in vitro equilibrium binding study, it is performed under the conditions of the specified time, which is usually equilibrium binding time, and the varying absorbate concentrations, which is phosphate concentration for Cevimeline carbonate products. The figure here is an example of equilibrium binding profiles. X-axis is the initial adsorbate concentration, and the y-axis is bound adsorbate. The selected concentration should be contain the increasing portion of the binding curve until the maximum binding is reached. To ensure the maximum binding, it should include at least 2 concentrations at plateau. In addition, because a sorbate may have different affinity to API under different pH conditions, different adsorbate conditions may be applied. This slide is the case example for ENDA review. The PSG for development components recommends 2 pH conditions for binding study, pH 4 and pH 7. In this ENDA, the applicant selected the same phosphate concentration range for both pH 4 and pH 7. Phosphate has higher affinity to development at pH 7 than pH 4 due to the different ionization state of phosphate species. Therefore, from the in vitro equilibrium binding study under these 2 conditions, it reached to equilibrium for pH 7 conditions, but did not reach to equilibrium under pH 4 conditions. This case shows adequate phosphate concentration should be applied under different conditions. The second case is mesalamine drug products. Mesalamine contains 2 delayed release dosage forms, including DR capsule and the DR tablet. It also has 1 extended release capsule. There are 1 to 6 approved ANDAs for each dosage forms. Mesalamine is for the treatment of active ulcerative colitis. It has a topical anti-inflammatory effect on chronic epithelial cells. Although mesalamine is for the local treatment, the bioavailability is about 20 to 35%, depends on the drug products. For B recommendations of mesalamine DR products and ER products, they are similar but still have some minor differences. For PK study, fasting and fat studies are recommended for both DR and ER products. However, the partial AUC is different. For DR products, partial AUC is recommended from 8 to 48 hours. For ER product, partial AUC is recommended from 0 to 3 hours and 3 hours to last measurable time point. Comparative dissolution testing under various pH conditions is recommended for both DR and ER products. Dissolution similarity factor, which is F2, is evaluated to compare test product and reference standards. However, because the dissolution of the DR dosage form shows high variability, the product-specific guidance recommends 24 units of the test product and at least 2 lots of the reference standards to compare the dissolution testing. This slide shows the review for highly variable dissolution data, which is mesalamine DR product. If the dissolution profile is highly variable, the bootstrapping method is used to calculate mean data and the lower bound of 90% confidence interval of the F2 values. To do this, first, it can compare test and the reference. If both mean data and the lower bound of 90% confidence interval are above the acceptable limit, then the dissolution result meet the acceptance criteria. If F2 does not pass the acceptable limit, we can further calculate the data between 2 lots of the reference standards, and then compare the value of the test versus reference to reference versus reference for the dissolution evaluation. Please note that methods other than bootstrapping method with sufficient justification are also acceptable for highly variable dissolution data. For the rationales of B recommendations, the PK study is recommended because the bioavailability of the mesalamine is about 20 to 35%, so the drug concentration in plasma is measurable. For PK parameters, in addition to AUC and Cmax, partial AUC is recommended. Partial AUC can reflect drug absorption in GI tract. In addition, it helps discriminate the formulation differences between the test and the reference standard. In vitro comparative dissolution study in different pH can ensure the test product to have similar dissolution behaviors as the reference standard. It serves as a surrogate of in vivo drug release in the GI tract. Mesalamine DR dosage forms have high dissolution variability. More units of products help to perform bootstrapping F2 analysis. The next case is vancomycin hydrochloride capsule. For vancomycin hydrochloride capsule, it has 5 approved ANDAs. Vancomycin acts locally in the lower GI tract. The indication is for the treatment of C. difficile associated diarrhea and staphylococcus enterocolitis. The mechanism is through the inhibition of the cell wall biosynthesis of bacteria. For the drug absorption, vancomycin is poorly absorbed, so the vancomycin concentration measurement is very limited in plasma after oral administration. For B establishment, there are 2 options for vancomycin hydrochloride capsule. For the first one, if test product and the ILD are Q1 and Q2 the same, B can be established during in vitro competitive dissolution testing under 0.1N hydrochloride, pH 4.5 and pH 6.8. And then F2 is evaluated to compare test and the reference. The F2 evaluation is similar to mesalamine drug product. If it is not Q1 and Q2 the same for the test product and the ILD, instead of in vitro testing, an in vivo comparative clinical endpoint B study should be conducted to demonstrate BE. For the rationales of B recommendations, because the absorption of vancomycin is very limited, it is not possible to use PK study to demonstrate BE, which is different from mesalamine drug products. For the API properties, vancomycin is highly soluble across pH in the GI tract, so in vitro dissolution testing can be used to predict drug in vivo release behavior. If test product and the ILD are Q1 and Q2 the same, the impact of excipients on absorption or other in vivo performance is minimized. In this case, in vitro competitive dissolution testing can be used to establish BE. However, if the test product and the ILD are not Q1 and Q2 the same, certain excipients may have impact on drug in vivo performance, which is unknown. Therefore, the competitive clinical endpoints B study is recommended to establish BE. The final case is fludarabinurate drug product. Fludarabinurate has 2 dosage forms, tablet and suspension. Currently, there is no approved ANDA for both dosage forms. Fludarabinurate is for the treatment of C. difficile associated diarrhea. It is an antibacterial drug by inhibiting RNA synthesis of bacteria. For API properties, the permeability and the absorption are poor, but small amount of API can still be absorbed into the body. A little similar to vancomycin hydrochloride capsule, there are 2 options for B establishment. If test product and the ILD are Q1 and Q2 the same, in vitro competitive dissolution testing is recommended to evaluate dissolution similarity between the test and the reference. The difference from the vancomycin hydrochloride capsule is that for fludarabinurate, PK studies, including fasting and fat studies, are recommended. If test product and the ILD are not Q1 and Q2 the same, a competitive clinical endpoint B study is recommended to demonstrate BE. For the rationales of B recommendations, if test product and the ILD are Q1 and Q2 the same, the impact of excipients on drug in vivo performance, such as absorption, can be minimized. Although fludarabinurate is poorly permeable and soluble, there is no analytical sensitivity issue to determine drug concentration in plasma, so PK study is recommended. However, the API is not stable during GI transit. equivalent systemic PK does not guarantee equivalent amount of the API at the site of action. So PK studies serve as a complementary measure. Comparative in vitro dissolution studies can simulate and evaluate comparative drug release behavior between the test product and the reference standard in different portions of the GI tract. It is critical to assess BE. If test product and the ILD are not Q1 and Q2 the same, different type and amount of the excipient may have different impact on in vivo performance at the site of action. Therefore, the comparative clinical endpoint B study is recommended to establish BE. Challenge question 2. Which of the following is the consideration to develop BE recommendations for oral locally acting GI drug? A. Mechanism of drug products. B. Systemic or non-systemic absorption of drug product. C. Physical property of drug products. D. All of the above. The correct answer is D. Mechanism of drug products, absorption of drug products, and the physical property of drug products are all considered for B recommendation development. There are 2 ongoing grants under the GDUFA. The purpose of these grants intends to develop a biopredictive dissolution method and establish modeling to support B demonstration for generic locally acting GI drug products. The ultimate goal is that it can establish alternative approach to demonstrate BE between the test product and the ILD. To summarize, for locally acting GI drug, systemic exposure may not reflect drug concentration at the site of action. In addition, drug plasma concentration may be limited, so it needs adequate method to establish BE. For BE recommendations of the oral locally acting GI drug product, we consider drug product properties and the mechanism of action. There is ongoing research. The purpose is to improve in vitro dissolution method and develop a predictive in silico model to support the BE determination. Finally, I would like to thank Myrna, Jasmine, Heather, Myungjen, Lei, and Rob for their input and suggestion for the slides and the presentation. That's all my presentation. Thank you.
Thank you for the introduction, and good morning, everyone. In the second presentation of this session, I will be talking about Q1, Q2 recommendations of locally acting gastrointestinal or GI drugs with a focus on Sucralfate oral suspension as a key case study. From this presentation, you will learn to define qualitative or Q1 and quantitative or Q2 sameness and employ best practices in determining Q1, Q2 sameness. You will recognize the different scenarios where Q1, Q2 sameness is recommended for locally acting GI drugs. You will describe a key case study of Q1, Q2 recommendations for locally acting GI drugs, which is Sucralfate. And finally, you will describe the rationale behind bioequivalence or BE recommendations of Sucralfate oral suspension. Starting with the first objective, what is Q1, Q2 sameness? Q1, Q2 is an assessment of the inactive ingredients in the proposed generic drug product compared to the inactive ingredients in the reference listed drug or RLD, where Q1 same means the inactive ingredients are the same, and Q2 same mean the concentration of the inactive ingredients are the same. Please note here that the current FDA's practice generally allows a plus or minus 5% difference in the amount of an active ingredient compared to that of the RLD. But why Q1, Q2? Generic oral drug products are not expected to be Q1, Q2 the same as their RLD, which is in contrast to non-oral drug products such as parenteral, otic, or ophthalmic drug products that are generally expected and in certain cases are required to be Q1, Q2 same as RLD. However, in certain cases, inactive ingredients may impact either the availability of the drug at the site of action or the product performance or both. An example will be a bile salt or a surfactant that acts as an absorption enhancer in products of low permeability drugs. And in such cases, Q1 is same and Q2 same or similar, depends on the case, are recommended. So we covered the what and the why, and now how to determine Q1 and Q2 sameness. While developing an oral drug product for an abbreviated new drug application, or ANDA, a firm can ensure that the product is Q1, Q2 the same as the RLD by checking the product qualitative composition in the approved labeling of the RLD, and the firm can also submit a controlled correspondence requesting to check if the proposed formulations are suitable for BE approach with the condition that the product-specific guidance of the product include a Q1, Q2 recommendation. To learn more about that, please refer to the general guidance linked in the slide. For locally acting GI drug, there are several PLG that include Q1, Q2 recommendations as listed on the right side, such as a carbose tablet, linaclotide capsule, and vancomycin hydrochloride capsule. The Q1 Q2 sameness in these PLGs is either a prerequisite to waive in vivo studies with pharmacokinetic endpoints, such as the PE recommendations for vancomycin hydrochloride capsule that Dr. Sun described earlier, a prerequisite to waive in vivo studies with clinical endpoint, such as the PE recommendation for rifaximin tablets and fidaxomicin products that also Dr. Sun discussed, a prerequisite to waive in vivo studies with pharmacodynamic endpoint, such as the PE recommendation for acarbose tablets, and combined with in vitro studies for BE determination, which is the only BE option for sucralfate products and barium sulfate products. And now we stop at our first challenge question. One reason a PLG of an oral drug product to recommend Q1, Q2 sameness is A, as a prerequisite to waive in vivo BE studies, B, to ensure the particle size of the excipients is the same, C, to encourage developing generic drug products that are the same as the RLD. I will give you a few seconds to think about the answer. And the right answer is A. Particle size are not part of a qualitative or quantitative sameness, and oral drug products are not expected to be Q1 and Q2 same, but in certain cases, if the products are Q1 and Q2, an alternative BE approach to that of the in vivo study can be used. Moving to our key case study of this talk, which is sucralfate oral suspension. To start, sucralfate is a locally acting agent that is indicated in the short term, up to 8 weeks, treatment of active duodenal ulcer. It is minimally absorbed from the GI tract, and structurally, sucralfate is an aluminum salt of sucrose octasulfate. Here, I want to bring an important point that sucralfate products, which are tablets or suspension, are classified as complex products because they work locally, complex route, and because the active pharmaceutical ingredient or the API itself is classified also as complex. In this table, I listed the approved sucralfate products in the United States, including the generics and their RLD, as well as the PSG of the products. From the table, you can see that the first PSG of sucralfate drug products were withdrawn in 2015. And I want to talk in the following slides about the history of the BE recommendations and the developments of the current BE recommendations specifically to sucralfate suspension. The first PSG of sucralfate suspension recommended an in vivo BE study with clinical endpoint that included patients with active duodenal ulcer. And this PSG was withdrawn because it was considered unethical to enroll patients who are H. pylori positive without providing standard therapy in the placebo arm. On the other hand, the recruitment of patients who are H. pylori negative would significantly limit the enrollment. And that resulted in the need for the development of an alternative approach for BE evaluation. The rationale behind this alternative approach is developing an in vitro method based on the understanding of the product-specific factors and the drug mechanism of action. The process of developing BE recommendation for sucralfate suspension, including collecting information about sucralfate mechanism of action, the administration of the suspension, the formulation, manufacturing process of the API and the suspension, conducted in vitro and in vivo studies from different resources, including RLD labeling and the submission, and literature, and as well as the lab research effort in developing in vitro methods related to the performance of sucralfate suspension. All this effort led to the development of the PSG and helped in the approval of the first generic of sucralfate suspension. Revealing the current BE recommendation of sucralfate suspension, which include first, sameness of the API, especially that, as we said earlier, sucralfate is a complex API. 2, comparative physiochemical characterization of product, since the product attributes can impact the quality, physical stability, and performance of sucralfate suspension. And finally, bioassays of the products, including binding study. Those in vitro bioassay studies are based on the mechanism of action of sucralfate. In addition to the sameness assessment of API and comparative in vitro testing of both the proposed test and reference standard, the test product should be Q1 and Q2 the same as RLD. The recommendation of sameness applies to all inactive ingredients except for flavor or color. Ready for our second challenge question? Which of the following is false regarding sucralfate suspension? A. It contains a complex active pharmaceutical ingredient. B. It is considered complex due to its route of administration. Its current BE recommendations include BE studies with pharmacokinetic endpoints. D. Its current BE recommendation include in vitro binding studies. Few seconds. And the right answer is C. BE of sucralfate products include in vitro studies, and sucralfate is minimally absorbed. So it's hard to link its systemic absorption and the pharmacokinetic endpoint to its action, to its local action in the GI. Sucralfate API is actually insoluble in water. So how adding inactive ingredients to make a suspension would impact sucralfate? Here is my evidence. As part of sucralfate mechanism of action, aluminium cations are released from sucralfate in acidic media, and sucralfate becomes charged and thus binds to proteins in the ulcer area. Performing acid titration to sucralfate API reveals that sucralfate aggregates and forms a paste with adhesive properties. However, the role of the adhesive paste is not fully understood. On the other hand, sucralfate suspension doesn't form the adhesive paste in the acidic media, indicating that viscosity modifiers and suspending agents could prevent the formation of the paste. However, it was observed that the apparent viscosity and the sedimentation rate of sucralfate suspension increased with acid addition, indicating that at least aggregation of sucralfate occurs. Here, I want to highlight that both the mechanism of action of sucralfate and the impact of the excipients on the performance of sucralfate still are not fully understood. Thus, the PE determination of sucralfate suspension is based on a totality of evidence approach, which involves characterization of both the drug substance and the formulation of the proposed generic product that has the same active and inactive ingredients of the RLD. In summary, Q1, Q2 recommendation for oral drug product is considered when the inactive ingredients may impact the availability of the drug either at the site of action or the product performance or both. For BE determination of some locally acting GI drugs, Q1 Q2 sameness can be a prerequisite to waive in vivo studies. The current BE in vitro recommendations of sucralfate suspension, the case study discussed, are based on the product attributes and the mechanism of action of sucralfate. Finally, demonstrating BE for sucralfate suspension is based on a totality of evidence approach of a proposed generic product that is Q1, Q2, same as the RLD. If you have any questions, please submit them in the chat box, and I will be glad to answer them in the panel discussion later today. Finally, on behalf of AGD, thank you.
Hello everyone, I am Suman Nandamudi, Assessor in Division of Bioequivalence 3 within Office of Bioequivalence in Office of Generic Drugs. Firstly, I would like to thank SBI staff for organizing this meeting. Today, I'll be speaking to you about approval of generic product for Sucralfate oral suspension, which is not quantitatively the same as the reference listed drug. I have 3 learning objectives for today's talk. First, I would like for you to understand the FDA's considerations in the development of the product-specific guidance for Sucralfate oral suspension. Next, I would like for you to understand the rationale for recommendation of qualitative and quantitative, that is Q1, Q2 sameness between the test and reference formulations when developing the generic versions for this drug product. The final objective is describe the potential alternate approaches that can be utilized for the demonstration of bioequivalence for generic Sucralfate oral suspension products. A little brief background about this drug product. The reference listed drug is Carafate oral suspension, which is available in 1 strength, 1 gram per 10 ml, and was approved in 1993. As you can see the structure of Sucralfate in this slide, it is a complex of sucrose, sulfate, and aluminum. It is an aluminum salt of sucrose octasulfate. The drug product is indicated in the short-term treatment of active duodenal ulcer. Per the RLD labeling, Sucralfate oral suspension should be taken on empty stomach. This is critical for Sucralfate to exert its action. It is a locally acting drug which is minimally absorbed from the GIT. Any small amount of sulfate disaccharide that is absorbed is excreted primarily in the urine. Before we go into the bioequivalence recommendations for this drug product, we will first understand the mechanism of action of Sucralfate. The exact mechanism of action of Sucralfate is unknown, but within the gastric environment, it is first activated to exert its action. When taken on an empty stomach, Sucralfate is dissociated into sulfated sucrose and aluminum salt. Within the acidic environment, the sulfated sucrose forms polymer, and now this negatively charged polymer can interact with the positively charged proteins within the mucus such that it increases the viscosity and increases the thickness of the mucosal layer. In this way, Sucralfate can increase the mucus secretion, which then reduces the diffusion of H+ ions to the ulcer base. Similarly, Sucralfate polymer can also form a coat on the ulcer base so that it is protected from further attack by the H+ ions. Also, another effect is on pepsin and bile salts. Sucralfate forms stable complexes with proteins and inhibits their hydrolysis by preventing the pepsin-substrate interaction. Pepsin is a cleavage enzyme which is responsible for the cleavage of proteins. Sucralfate also inhibits peptic activities by binding directly to the pepsin. Similarly, it is postulated that Sucralfate also binds to the bile salts. Sucralfate is also known to stimulate the mucosal prostaglandin synthesis and bicarbonate secretion. This increase in the bicarbonate secretion can further interact with the gastric acid to reduce the gastric acidity. So all these effects can reduce the ulcer progression and increase the ulcer healing. The product-specific guidance for Sucralfate oral suspension provides bioequivalence recommendations with an in vitro option. However, to qualify for this in vitro option, the following criteria should be met. The test and RLD formulations have the same API. Since there are no clinical studies recommended and that the drug product is a locally acting, the agency would like to reduce any uncertainty that might arise due to the differences in the formulations between test and RLD products. Therefore, the PSG recommendations include qualitative and quantitative sameness between the test and RLD formulations. That is, the difference in the amount of each excipient should be within 5%. Comparative physicochemical characterization between the test and RLD products should be demonstrated. Lastly, acceptable bioassays. In the next slide, I'll go over the details of the in vitro bioassays that are recommended in this PSG. The text that is highlighted in blue, that is the Q1, Q2 sameness, and the bioassays are evaluated by the Office of Bioequivalence. However, the API sameness and comparative chemical characterization evaluation is done by Office of Pharmaceutical Quality. All the above recommendations in the PSG, they reflect the totality of the evidence approach for the demonstration of bioequivalence for the generic products of this drug product. Based on the current FDA scientific thinking, in vitro testing is considered appropriate for the generic version. However, if any applicant intends to submit alternate methods to demonstrate bioequivalence, then the agency will evaluate these methods on the case-by-case basis. Given the complexity of the mechanism of action of this drug, it is not practical to develop a bioassay that can incorporate all the aspects on how sucralfate elicits its action. Therefore, based on the postulated mechanism of actions of sucralfate, the PSG recommends 4 bioassays. As I have discussed in the previous slide in detail, sucralfate releases aluminum ions in the acidic environment and produces a negatively charged complex which can tie bindingly to the positively charged proteins such as albumin, on the ulcer side. So based on this, the PSG recommends in vitro equilibrium binding study with human or bovine serum albumin. The bioequivalence is based on 90% confidence intervals of the Langmuir constant K2 value being within 80 to 120%. The other mechanism of action by which sucralfate acts are inhibits the pepsin activity and binds to the bile salt. Therefore, based on this, the PSG recommends in vitro equilibrium and kinetic binding studies with the bile salt. And similar to the HSA studies, the bioequivalence is based on the 90% confidence intervals of K2 within 80 to 120%. The PSG also recommends the in vitro enzyme activity assay, and the equivalence is based on the quantitative comparability of percent decrease in the pepsin activity. In order to use these bioassays for bioequivalence establishment, the study conditions should be sensitive enough to detect the relevant differences between the test and reference standard products. Physiologically relevant conditions should be taken into consideration in conducting these in vitro binding studies. Study conditions for each bioassay should be optimized to obtain an optimal and sensitive biorelevant in vitro condition. The PSG for this drug product recommends providing optimization report for the selection of all the bioassay conditions such as the negatively charged complex formation process, the adsorbate and adsorbent concentrations, the assay duration, and the pH condition. For example, pH condition. Sucralfate activity is pH dependent. Therefore, it is critical that optimal pH is selected. A physiological pH condition that allows for maximal binding should be selected. In case of assay duration, incubation duration should be optimized to maximize the adsorption of sucralfate to either albumin or bile salt. Now that we understand the rationale for Q1, Q2 sameness recommendation for this complex drug product, can a generic product be approved if the test formulation is not qualitatively and quantitatively same as the reference listed drug? So for the remainder of my talk, I'll go over a case study for which the proposed test formulation is qualitatively the same but differ in the amount of excipient, that is, quantitatively not same as the RLD formulation. The PSG should be viewed only as recommendations unless specific regulatory or statutory requirements are cited. In case of oral suspension products, there is no regulatory requirement for Q1, Q2 sameness. Therefore, potential Q2 differences may be justified by providing comprehensive scientific rationale with multiple meaningful measures showing the lack of impact of the formulation difference on the product performance. Any alternate BE approaches can be utilized for the bioequivalence demonstration. So for this case study, the test formulation is Q1 the same but not Q2. As shown in the table in this slide, the difference in the amount of excipient A between the test and reference formulations is greater than 5%. Therefore, in order to justify the Q2 difference, the applicant provided the results of 6 bioassays as listed in this slide, in addition to the studies that were recommended in the PSG for this drug product. I will not be going over the details of these studies. However, I want to make a note that all the proposed additional studies were based on the mechanism of action of the sucralfate, which I have gone over in detail in the previous slide. In order to show that the difference in the amount of excipient A between the test and reference formulations do not have any impact on the product performance, the applicant manufactured the altered test formulations that included changes in the multiple excipients along with the excipient A. All the bioassays that were listed in the previous slide were conducted using test product, reference standard product, and altered test formulations. However, the in vitro bioassay results obtained using these altered test formulations were insufficient to mitigate the BE concerns of Q2 differences due to the confounding factor of varying other excipients simultaneously. In addition to this, Office of Bioequivalence also has a concern on the study design for the HSA binding assay. The PSG recommends that a fixed concentration of sucralfate with varying concentrations of HSA be used to conduct the study and the Langmuir constants K1, K2 be calculated. However, in this case, the applicant used a fixed concentration of HSA with the varying concentrations of sucralfate, and the constant Kd value based on the Hill equation was calculated. The K2 values are not dependent on the sucralfate or HSA concentrations. However, the Kd values are variable and differ when varying concentrations of HSA were used. Therefore, the applicant did not provide sufficient supporting evidence to demonstrate that its approach is more sensitive to detect the binding differences of sucralfate with HSA between the test and reference products than that recommended in the PSG. Based on this, the applicant was asked to repeat the in vitro bioassay studies. Based on the agency's recommendations, new altered test formulations with specific changes made only to excipient A were manufactured. The concentrations were bracketed, covering the proposed test and RLD amounts. The test LC contains lower amount of excipient A than proposed in the test product, whereas test HC contains higher amount of excipient A than the amount proposed in the test product. Additionally, all the bioassays were reperformed using the test product, RLD product, and these altered test formulations. These bioassays included the studies that were recommended in the PSG and some additional studies like mucoadhesion assay, delay in bile salt diffusion, and delay in acid diffusion. Additionally, the applicant also repeated the HSA binding study as per the study design recommended in the PSG of this drug product. In the next few slides, I will go over the results of the in vitro bioassays that were conducted to demonstrate that the Q2 differences do not have any impact on the bioequivalence demonstration. I would like to make a note here that all the results that are shown in the slides today are the hypothetical and not taken from any applications. To begin with the studies that were recommended in the PSG. This slide shows the equilibrium binding study results with HSA and bile salts. The binding profiles clearly shows that the maximal binding is established under the concentrations tested. The Langmuir constants K2 values were calculated for both the studies. As per the PSG, the 90% confidence intervals of the T/R ratios of the K2 values were calculated, and these values are within 80 to 120%, thus demonstrating the capacity of protein and bile salt binding is similar between the test and reference products. This slide shows the results of the pepsin activity study. The PSC recommends that the equivalence is based on quantitative comparison between the test and RLD products with respect to decrease in the pepsin activity. Currently, there is no recommendation for statistical analysis for this test. The study was conducted using the test product, reference product, and the two altered test formulations with low and high amounts of the excipient A. A predefined comparative criteria was applied to demonstrate the equivalence of the test formulations to the RLD. As you can see in this slide, the inhibition profiles of the pepsin activity are comparable between the proposed test product, altered formulations, and the RLD. This shows that the difference in the excipient A amount has no effect on the inhibition of the pepsin activity. The slide here shows the results of the mucoadhesion study. Sucralfate has preferential binding to the ulcer tissues as compared to the normal tissues. The amount of sucralfate bound to the ulcer tissue was compared across different treatments. For the statistical comparisons, equivalence acceptance criteria was proposed. Based on the scientific rationale with supporting data, the agency considered the proposal to be adequate. As shown here, the sucralfate bound to the ulcerated tissue was comparable across the different treatments tested. Thus, the results indicate that the two test formulations with the varying in the excipient amounts are equivalent to the reference product. Further, to demonstrate the bioequivalence for non-Q2 test formulation, additional bioassays such as delay in bile salt diffusion study and delay in acid diffusion study were also performed. These studies were conducted to demonstrate the equivalence of the test formulations with low and high excipient amounts against the reference standard. The assessment of delay in bile salt diffusion was performed by measuring the permeability values of the bile salts across a semi-permeable membrane. As shown in the results here, the pH values are similar among all the formulations tested. Based on the statistical criteria proposed by the applicant, the 90% confidence intervals of the T/R ratios of pH values are within 80 to 120% when the test product is compared with the reference product, additionally when the altered formulations were compared with the RLD product. The ability to delay the diffusion of acid provides important information about the bioequivalence of the test formulation to the RLD. The pH values were recorded initially and also at the end of the study duration. The pH delta values as shown in the result here are similar between all the formulations tested. Based on the statistical criteria, the 90% confidence intervals of the T/R ratios of the pH delta values are within 80 to 120% when the test product is compared with the reference product and the altered formulations are compared with the reference product. Therefore, based on the results of all these bioassays that were submitted, provides the totality of evidence approach to demonstrate bioequivalence for non-Q2 test formulation. I have two challenge questions. Challenge question 1. Which statements is not true? A. In vitro bioassay recommendations are based on the mechanism of action of the drug. B. The PSC recommendations for demonstration of bioequivalence for sucralfate oral suspension include both in vitro and in vivo PK studies. C. Physiologically relevant conditions should be taken into consideration in designing and conducting the in vitro binding studies. D. The totality of the evidence approach to assess demonstration of bioequivalence. If you have selected the answer B, you are correct. The PSC recommendations for the demonstration of bioequivalence for sucralfate oral suspension currently includes only in vitro option. Now challenge question 2. What statement is not true? A. Sucralfate binds to potassium. B. An in vitro binding study with bile salts is recommended in the PSC for sucralfate oral suspension. C. The BE assessment for the in vitro binding study is based on the 90% confidence intervals of the Langmuir constant K2. D. API sameness is recommended in the PSG for sucralfate oral suspension? The answer is A. Sucralfates bind to proteins, bile salts, and inhibits the pepsin activity. However, it does not bind to potassium. To summarize my talk, the product-specific guidance recommendations for the bioequivalence demonstration of the drug product sucralfate oral suspension is based on totality of evidence approach. Any studies that deviate from the PSG recommendation or alternate approaches can be submitted, however, with sufficient scientific evidence in order to demonstrate bioequivalence. In the case study that I have presented today, the combined findings from all the in vitro bioassays provided the totality of evidence for the demonstration of bioequivalence of a non-Q2 generic drug product. For a complex product such as sucralfate oral suspension, applicant can seek agency's feedback through multiple communications channels such as control correspondences, product development meetings, pre-submission meetings, mid-cycle review meetings, post-complete response letter meetings at various stages for any proposed alternate BE approaches. That concludes my talk, and I would like to acknowledge my colleagues who have contributed for this talk. I would like to thank you all for listening. The Office of Generic Drugs ensures high-quality, safe, affordable generic drugs are available to the American public. As an assessor in the Office of Bioequivalence, while performing thorough evaluation to ensure that these products meet the rigorous standards for safety and efficacy, I have always in my mind that I am part of that American public and I am a patient too. So on behalf of OGD, thank you all. I'm happy to address any questions that you have in the next Q&A session.
Thank you all for the great presentations. As a reminder to our audience, if you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We'd like to introduce and welcome additional panelists. First, Dr. Lisa Hoover is a supervisory chemist, Division of Pharmaceutical Analysis in the Office of Testing Research, OPQ. I'd also like to welcome Dr. Fang Wu, Senior Pharmacologist, Division of Quantitative Methods and Modeling, Office of Research and Standards in OGD, and Dr. Hongfei Zhou, Senior Pharmacologist in Division of Bioequivalence III, Office of Bioequivalence, and OGD. Looks like we have some questions coming in right now. The first few questions are addressed to Dr. Weijie Sun, and here is the first question for Dr. Sun. Why are some products just need the PK and the in vitro dissolution studies to establish BE, but some products need Q1 and Q2, the same in addition to PK and in vitro dissolution studies?
Okay, thank you for the questions. So several factors are considered for the BE recommendations. So for example, in the case of the fludarabine, it requires the Q1 and Q2 the same in vitro dissolution study and the in vivo PK study. And this is because the API is not stable in the GI tract. So the equivalent PK does not guarantee the equivalent amount of the API at the site of the action. So the PK study cannot be used solely to establish BE. It needs the Q1 and Q2 the same together with the in vitro and the in vivo PK study to establish BE. And in the case of the methylamine drug product, and there's no API stability issue in the GI tract. So only the dissolution study and the PK study are recommended to establish BE. Thank you.
Thank you for responding to that question. We've got a couple more questions, and here's the next question for Dr. Sun. Why are PK studies recommended for fidaxomicin with Q1, Q2 to the RLD, and it seems to be, is it very similar to the absorption of vancomycin?
Okay, thank you for the questions. So this question is a little related to the previous question. So again, several factors are considered for the B recommendations. So in the case of the vancomycin, because the absorption is very limited, so it is not feasible to have an in vivo PK study because the bioanalytical sensitivity issue. And vancomycin is also highly soluble in the entire GI tract. So the API is expected in solution when it reaches to the site of the action. So only in vitro dissolution study is recommended if the generic product and the RLD are Q1 and Q2 the same. And in the case of the fludarabinamycin, partial of the API is absorbed into the systemic circulation. So the PK study is feasible. But because the fludarabinamycin is not stable in the GI tract, so the PK cannot be used solely to establish BE. In addition, the solubility of the fludarabinamycin is not that high in the entire GI tract. So it needs both dissolution and the PK studies to establish BE when it is Q1 and Q2 the same. Thank you.
Thank you, Dr. Sun, for responding to that question. We've got a couple more questions for you, and here's the next question. This is referring to the bioavailability being low in GI locally acting drugs. Is it possible to use multiple units of the drug unit to increase drug plasma concentrations for the PK study?
Yes, so if the P product specific guidance recommends the in vivo PK study, and it is acceptable to use the multiple units of the drug product to increase the drug plasma concentrations and achieve the sufficient bioanalytical sensitivities. But the total single dose should be within the labeled dose range, and the dose range is safe to the study subject. Thank you.
Thank you, Dr. Sun. We just have one more question in this round for you, and here's the question. You mentioned in vitro equilibrium binding study is considered as a pivotal study. How many lots are needed for the in vitro equilibrium binding study?
Okay, thank you for the questions. So if there is no further recommendation in the product specific guidance, then one lot for the in vitro binding study is sufficient and is fine. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few questions that came in from Dr. Manar Algabish, and here's the first question for Dr. Algabish. For products such as vancomycin that require multi-pH in vitro studies, would it require similar multi-pH comparative dissolution studies for post-approval changes, such as site change or dissolution profile comparison in QC media? Is that sufficient?
Thank you, Ray, and that's a very good question. To summarize it, I will start that it all depends what are those changes and also how significant they are. So talking about the multi-pH dissolution, which is a recommendation, it's a BE recommendation, and that's this recommendation is based on Q1, Q2 sameness demonstration for the vancomycin product. So if that changes, it did not include a Q1, Q2, meaning the excipients are not there. However, the changes could impact the performance, and that's a BE demonstration is needed. Then the competitive multi-pH solution should be conducted. That's the first part. The other part about the QC dissolution, this recommendation is usually there. It's for the quality. So usually part of the release of this new batch should be there anyway. So it's a two different things about would that change affect quality, which is probably could impact that, and would that also impact the performance of the product. Thank you.
Thank you for responding to that question. We do have a few more questions for Dr. Algabish, and here is the next question. If the RLD is withdrawn, can Q1, Q2 sameness be determined using the RS product?
Another interesting question was, so usually would say that Q1, Q2 sameness is based on the RLD. However, I would understand that, it could be a company may need to do some reverse engineering to figure out the composition quantity and so on. If the RLD is not there, how to figure that out? I would always say, generally speaking, if could be that our product to start with is Q1, Q2 same as RLD, so you're not far off. And the second, there is always the option to submit a control correspondence for FDA to ask for, is the biowaiver option based on the Q1, Q2 sameness. So hopefully that helped answer the question. Thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Algabesh, and here's the next question. For complex APIs that require in vitro API sameness studies to support waiver of an in vivo study, for post-approval change in the API source, would similar API sameness studies be required?
Ooh, another interesting post-approval changes. So in short, I would say yes. API sameness should be demonstrated if the source or if the API, there's changes in the API source. So, the short answer will be yes. Thank you.
Thank you for responding to that question. We have one more question from Dr. Algabesh, and here is the question regarding Q2 sameness. How much compositions or concentration of the proposed generics are expected to be the same as the RLD, and are any differences allowed?
Thank you, Ray. So yes, Q2 sameness, you're expected the product to be the same, but there is some differences allowed, and the general practice is 5% is an acceptable amount. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we receive a few questions for Dr. Suman Dandamudi. And here's the first question for Dr. Dandamudi. Are all bioassays required to be performed in the PSG, or would only one bioassay be sufficient?
Good morning, all. Thank you for that question. As I stated during my presentation, this is a complex drug product, and given the complexity of the mechanism of action for sucralfate, it is not practical to develop one bioassay that incorporates all the aspects of how sucralfate can exert its action. Therefore, the PSG recommendations are based on the postulated mechanism of action. As such, all the studies that are recommended in the PSG should be conducted for demonstration of bioequivalence. Thank you.
Thank you for responding to that question. We've got a few more questions for Dr. Dandamudi. And here's the next question. Will the FDA approve an ANDA application that does not contain the extra bioassays that were discussed in the slides?
Thank you for that question. I just want to remind that what I have presented in my talk today is just an example of what alternate BE approaches can be utilized if the formulation is not Q2 the same as the RLD. By stating that if ever all the criteria that is recommended in the PSG is met, that is the test formulation is Q1, Q2 the same as RLD, the API sameness between test and reference, and comparative physicochemical characterization between the products, then the bioassays that were recommended in the PSG would be sufficient to demonstrate bioequivalence. And FDA in general considers the totality of evidence approach to deem the bioequivalence. So any studies that the applicant want to propose should have sufficient scientific rationale with appropriate data to support these approaches. Thank you.
Thank you for responding to that question. A few more questions for Dr. Dandamudi. And here's the next question. Is the Sucralfate amount corresponding to dose recommended in the RLD label appropriate for conducting in vitro binding studies?
Thank you for that question. The sucralfate concentration selection typically should be based on the method development experiments that were conducted to optimize this parameter by stating that the amount that is utilized for any binding studies will typically depend on the type of adsorbate and what is the pH condition and the incubation time and so on. So as such, the sucralfate concentrations have to be optimized initially or have to provide sufficient scientific justification for to use in the in vitro binding studies. Thank you.
Thank you, Dr. Dandamudi. We do have one more question for you in this round, and here's the question. Is citing literature sufficient regarding the selection of the bile salt and its concentrations for conducting the binding studies?
That's an interesting question. So all the study conditions that are utilized for these bioassays have to be optimized so that an optimal and sensitive biorelevant in vitro conditions can be utilized. Thus, the PSG for this drug product, that's the reason, recommends providing optimization report for selection of any bioassay conditions. And these will be based, acceptance of these will be decided on case by case after the evaluation of this ANDA submission. And so as such, the citing literature alone wouldn't be enough to justify the bile salt concentrations that is utilized for conducting study. So because the inappropriate concentration selection could lead to inaccurate K2 calculations, which may have an impact on the bioequivalence assessment. Therefore, the bile salt concentrations that are typically selected for these studies have to clearly demonstrate the rising portion of the binding curve and the maximum binding region. And as such, they should have enough points in this linear portion to have appropriate estimation of this K2 Langmuir constant. Thank you.
Thank you for answering that group of questions. Moving on to our next panelist, we received a question for Dr. Alicia Hoover, and here's the question. What are some of the analytical methods that can be employed for API characterization?
Yeah, that's a really, really good question. And, you really need to think about the key characteristics that you need to think about with each individual product, because we have so many tools that we can use to characterize these complex APIs. So we've seen several examples today, and very commonly we see spectroscopic characterizations. You can use Fourier transform, infrared spectroscopy, FTIR. You can use UV spectroscopy, Raman. Really commonly, you'll need to use elemental analysis, particle size distribution, or differential scanning calorimetry, DSC. Today we saw some examples of the use of solid-state NMR to look at the degree of cross-linking. Thermogravimetric analysis, TGA, can be used to look at the degree of protonation. And you'll also see a lot of powder X-ray diffraction. But really, you need to understand what you need to characterize it and provide sufficient scientific justification and data to properly characterize those APIs. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we received a few questions for Dr. Feng Wu, and here is the first question. How can PBPK modeling help with BE assessment for GI locally acting products?
Thank you for the question. PBPK modeling to support BE assessment for GI locally acting products is an evolving area. Validated PBPK modeling can be used to predict local drug exposure For example, biopredictive dissolution data at biorelevant pH can be used as model inputs, then virtual bioequivalence simulations can be conducted for local drug amount to support BE assessment for GI locally acting products. Also, as mentioned in previous presentation, research efforts have been made on improving in vitro biorelevant dissolution methods and developing in silico models to support BE determination for GI locally acting products. Thank you.
Thank you, Dr. Wu. We've got a couple more questions that just came in for you, and here's the next question. What are the communicating pathways with FDA if I have questions regarding using modeling to support BE assessment for GI locally acting generic products?
Thank you for the question. The applicant can use pre-ANDA product development meetings, controlled correspondence, post-CRL meetings, and the to-be-launched Model Integrated Evidence MIE industry meeting pilot to discuss modeling-related questions with FDA. Regarding the MIE industry meeting pilot program, Dr. Liang Zhao will introduce it more in his talk this afternoon. Thank you.
Thank you, Dr. Wu, for answering that question. We do have one more question for you in this round, and here is the question. What is your view on the challenges of validating PBPK modeling for GI locally acting products, and what are your suggestions on overcoming these challenges?
Thank you for the question. In my view, lacking data of local concentration for drugs is the major challenge for validating PBPK modeling for GI locally acting products. To overcome these challenges, currently we are conducting research and incorporating biorelevant dissolution into PBPK model and use systemic PK data as well as data from available BE studies to support validation of PBPK model for GI locally acting products. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few questions for Dr. Hongfei Zhou. And here's the first question for Dr. Zhou. Generally speaking, if Q1, Q2, the same as recommended in the PSG, going through a non-Q2 route appears to be more challenging and requires additional efforts, then why would an applicant choose that difficult route?
Thank you for the question. This is a very good question, a very reasonable one from industry perspective. I think the best person to answer this question may not be me, but the applicant. However, I can help you sort out some potential reasons for this kind of decision. First, there's no regulatory requirement on Q2 sameness for oral suspension. Q1, Q2 is recommended on PSG, but not mandatory. This makes the non-Q2 formulation possible. And secondly, there's a possibility that the applicant did not figure out the Q1, Q2, although I think this is very unlikely. More likely, probably the applicant may purposely make some improvements on the formulation to make them stand out from the competition. And it is also possible that the applicant's drug development, they started earlier before the guidance revision as mentioned, and was finalized. And the Q1, Q2 sameness was recommended. And so if that's the case, they developed the drug product before the revision was published. It will be a combined business and science decision for the applicant to either reformulate the product or go through the alternative approach. I think that's all I have right now. Hope that helps. Thank you.
Thank you for responding to that question. We do have the next question for Dr. Zhou. Is a Sucralfate pepsin binding study sufficient to serve as the in vitro enzyme or pepsin activity study?
Thank you for the question. And I'm glad that you asked this question. The short answer to this question is no. The reason behind it is that we know sucralfate has the function to inhibit pepsin activity, and the inhibition is likely work through both adsorbing pepsin and buffering hydrogen ions. So therefore, I believe the activity assay is the one that can more accurately determine the pepsin inhibition capacity by sucralfate, as that will take into consideration of both mechanisms. And another reason is that we know enzyme inhibition by binding really depends on the binding site. Not all the binding works the same way. Some bindings just do not inhibit the enzyme activity. So to tell the real inhibition results, the activity assay is the way to go. Hope this helps, and thank you.
Thanks, Dr. Zhang. We do have one more question for you that came in, and here's the question. Are there any special considerations for the statistical analysis of an in vitro binding data?
Thank you. This is a good question. Just as Dr. Suman has already mentioned in her talk, the BE acceptance criteria for this in vitro binding is 80 to 120%. That's different from the plasma data, the acceptance criteria of 80 to 125%. The reason behind is that the data for this in vitro binding do not need to be log transformed for the analysis. Thank you.
Thank you for responding to that group of questions. Got about a little over 3 minutes left. We're going to try to get in some more questions. And we just got some that came in for Dr. Benar Algavish. And here's the next question for Dr. Algavish. And it's another question about Q2. Are the difference in the percent based on the product weight, or how are they calculated?
That's a good question. Thank you. So the difference is all related or going back to in relation to the RLD. So usually it's based on the amount, the amount of tests, versus the amount in the RLD over the amount in RLD. So it's all based on the amount or the weight. So it's not on the percent. Thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Algavish. Here's the next question, and it's regarding a sucralfate suspension. Can industry develop a product with a different color?
Thank you for your question. So yes, even the recommendation for sucralfate that the product need to be Q1 and Q2. Some changes are permitted, including the color and the flavor. So the short answer is yes. Thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Algavish. Here's the next question, still dealing with sucralfate. The PSG for sucralfate suspension recommends API sameness and another in vitro performance-based testing. One of the recommendations also indicated that the product should be Q1 and Q2 same as the RLD.
As probably indicated by my colleagues in their talks and with my talk as well, the BE demonstration for sucralfate is based on the totality of evidence. So this is why API sameness, and then going to the excipients Q1, Q2 same, and then the performance and the characterization of the product are among all these recommendations. Thank you.
Thank you for responding to that question. We're going to try to, it's just less than a minute. This will be our final question to Dr. Algavish, and it's referring to slide 6. Linaclotide, which is a peptide of BCS class 3, is listed under GI products whose PSGs recommend Q1, Q2 sameness. For this product, can the principles of ICH M9BCS based waiver be applied here to achieve the sameness?
Wow, that's a very specific question. So trying to give my recommendations over here, I would say first, the PCS classification are made for oral product that being absorbed, under pharmacokinetic studies. So trying to apply them to locally acting GI, you need to be very careful on what need to be recommended here or not. For example, the linklotide one is actually, it's minimally absorbed, so there's no pharmacokinetic studies are recommended for this to start with. And speaking about the sameness is for this product, it should be depending, okay, I'm trying to take this a step back. So I would say, no, you cannot probably apply the waivers based on this, because this one, the classification or the waivers is, or the recommendation for linklotide is not a pharmacokinetic even studies. I think it's a clinical dependent clinical endpoint studies. So I hope I'm trying to hopefully I answered that question. Thank you.
A huge thank you to our speakers and panelists for answering numerous questions that came in from the audience. We'll now enter into our lunch break until 1:35 p.m. Eastern Time. Enjoy your lunch break.
Okay, sorry.
Welcome back from the lunch break. We do have a few housekeeping notes and reminders to get us started. This activity is eligible for an attendance certificate which supports continuing education by SOCA reps, SQA, and ACRP. Please refer to our website at fda.gov/CEDAR-SBIE for more details. You'll be able to obtain the attendance certificate only upon completion of the survey, which remain open for 2 weeks. Remember, that is September the 20th. Please remember to download the certificate immediately upon completing the survey. It cannot be retrieved after the fact. Let us know your comments and feedback via the survey. This will help guide the design of our future events. To summarize, for our physicians, pharmacists, and nurses claiming the 13.25 continuing education credits, attendees have 14 days from the last day of the activity to log in, complete the required CE evaluations, and attest to attendance to claim credit. So be sure and complete the CE session evaluation on or before September the 28th. As a reminder, the email we send to attendees later today will have information on both the CE claim code and the survey link. We'd like to welcome you to Session 7, Enhanced Processes, Research, and Assessment Tools to Support Generic Drug Product Development. Our first presenter is Dr. Sam Rainey, Associate Director for Sciences, Office of Research and Standards in OGD. Then we'll be joined by Dr. Xiaoming Zhu, Division Director, Division of Product Quality Research, Office of Testing and Research in OPQ. And our final presentation in this session will be provided by Dr. Liang Zhao, Division Director, Division of Quantitative Methods and Modeling, Office of Research and Standards in OGD. Please join me to welcome our first presenter in this session, Dr. Sam Rainey.
Hello, my name is Sam Rainey, and I'm the Associate Director for Science in OGD's Office of Research and Standards. It's my pleasure to be with you here today to speak a little bit about the GDUFA research program and some of the enhanced processes that we've put in place under GDUFA III to establish research priorities that can support generic drug development. One of the things I hope you'll come away from this with is an ability to describe the generic drug science and research programs and FDA's commitments under GDUFA. Also, to be able to explain the processes that have been established and enhanced for prioritizing scientific initiatives for generic drug development, for conducting research, and for orchestrating the research program. And perhaps during the panel discussion, we can discuss some of the science and research priority initiatives for FY 2023, and perhaps discuss some research outcomes as well. Now, in terms of FDA's commitments under GDUFA III, one is to conduct 2 meetings per year with industry. We refer to these as biannual meetings, and they are held with an industry FDA working group that contains representatives from industry as well as the FDA. And through these meetings, we are able to discuss current and emerging scientific challenges that are really impacting generic product development and or assessment. And these are issues that are not necessarily regulatory or legal, but they are issues that arise because of some knowledge gaps, some scientific issues that can be addressed, remedied by research, by GDUFA-funded research. And then as an outcome of each of these meetings, we post the minutes of the meeting on FDA's website. Another commitment is to conduct an annual public workshop. And this is to identify what research is needed to address those scientific knowledge gaps that are limiting generic product availability for the American public. At this public meeting, we receive feedback about a lot of the different and most current issues that are impacting, that are making it challenging to develop generics, and in many cases complex generics or complex issues with other types of generic products. In these instances, feedback is received through a public docket for the workshop, as well as during public comment periods, during presentations. And we receive and synthesize all of this feedback and interpret them in terms of what the research priorities, what the priority areas for research should be in the next fiscal year. And then we use that as our guide for the areas in which we are advancing research initiatives. A third commitment is then to conduct that research, that internal and external research that addresses the priorities, the research priorities that we've heard are priorities for the American public and for the generic drug industry. And we do this by initiating within FDA, internal research collaborations in a lot of the priority areas where FDA has relevant scientific expertise and capabilities. We also then issue broad agency announcement, or BAAs, inviting contract research proposals that are aligned with those priority areas, as well as issuing requests for our applications, RFAs, or sometimes called funding opportunity announcements, which essentially invite grant research proposals for specific research concepts. When those proposals then come in, we orchestrate the objective reviews that help us to rank and identify BAA and RFA proposals that can be prioritized for funding, in other words, for grants and contracts that would advance these research initiatives. And there's a lot that happens with coordinating financial and administrative logistics to award these grants and contracts. Of course, among the one aspect of this is then that we post the list of new grants and contracts that were awarded each year with brief descriptions and objectives, and we post this on FDA's website to illustrate the alignment of the actual research that moves forward in a manner that is compatible with the priorities that were established, that we heard from the public workshop, from the comments, the docket, and so forth. And finally, it's FDA's commitment to report annually on how the GDUFA research that's conducted supports the development, assessment, and approval of generics. And this is done in an annual GDUFA Science and Research report that's published on FDA's website that describes some of the impactful research advances. Also, the reports provide metrics on regulatory outcomes that are impacted by GDUFA science and research. This is also done on FDA's website. So these are some of the commitments that FDA has under GDUFA III. And what I'd like to talk a little bit about is that infrastructure. You heard me refer to some of that infrastructure before. What exactly is that? So especially under GDUFA III, this infrastructure has been enhanced. And it involves laboratory research experts and equipment and research facilities within FDA. This is the infrastructure within FDA, everything on this slide that I'm referring to, as well as subject matter experts on the therapeutic performance of specific products. These are, for example, experts on inhalation products or topical products or drug device combination products. Also numerous subject matter experts in quantitative methods and modeling, for example, pharmacometrics or PBPK modeling and simulation or artificial intelligence and machine learning, as well as specialized expertise in clinical safety and human subject research, because much of the research that we do is clinical research. There's also specialized expertise in operations management for research administration and information systems, or IT support. This is because in order to be able to orchestrate, to implement a research program on this scale, it does take a lot of specialized infrastructure and research operations management to be able to make that happen. Now, one other aspect is that internal FDA research collaborators and subject matter experts exist within some of our advanced laboratory facilities in multiple FDA offices and centers. And we also have ANDA assessors in OGD and in OPQ who play a big role in the program. And in fact, the GDUFA III Implementation Research Program Working Group includes members from OGD, and OPQ. And these individuals play a variety of roles. They may, in fact, serve as reviewers for some of the for the grant or contract proposals that come in. They may be playing a seminal role, a central role in identifying the specific types of research that are needed in order to be able to address those priority areas that we've heard about. And in terms of some of the new enhanced processes, implementation of our GDUFA commitments under GDUFA III was very systematically and kind of painstakingly mapped out to be as effective as possible by including many of these representatives within FDA from multiple operating divisions within FDA itself. There's also the infrastructure for grant and contract administration, which includes budget finance partners in multiple FDA offices, as well as research governance with the CEDAR Research Governance Council with whom we coordinate. And finally, a key part of implementing the research program is our external partners. This naturally includes the industry FDA working group, who gives us a lot of guidance, as well as the Center for Research on Complex Generics, which works very closely with academia and industry to be able to understand some of the specific scientific challenges and to be able to orchestrate the dialogue that happens during the GDUFA public workshop, or that may happen in terms of feedback, public feedback to the docket or in other mechanisms, where ultimately we're able to get high quality information about what research is needed to address those scientific knowledge gaps that are limiting generic product availability. And so it's through these external partners that we're able to get high quality comments provided to FDA, again, whether it's through the dockets, whether it's through public workshops, whether it's through the biannual meetings with the industry FDA working group, whether it's feedback from the CRCG itself. Through all of these areas, we're able to ensure that the GDUFA science and research priorities are established appropriately and updated on an annual basis. Now, in terms of the flow through the course of the year, one can think of the research program as progressing through a sequential series of activities on an annual cycle that, perhaps begins, you might say, with the public workshop that happens every year in the summer. So, for example, for the FY23 public workshop, that occurred, and then based upon what FDA heard during that workshop, and in discussion with the biannual industry FDA working group, we draft the FY24 research priorities and publish those, as well as the meeting minutes from the biannual meeting. Now, with our marching orders for FY24, as it were, we can move through the rest of the cycle, but around this time, before we get started on that, we are also tending to wrap up the previous year and publishing our reporting and on the previous FY23 cycle. However, as soon as that's done, or sometimes in parallel with that, we're now announcing the BAAs and RFAs for the contract research and grants that are going to align with the new research priorities that have just been established. Internally, now that we know what those priorities are, we're beginning to conduct some of that research. And then once the external research proposals have come in and being reviewed, we initiate the external research. We meet again with the biannual meeting to start to now think about what some of the priorities are going forward in FY24. We then hold the FY24 GDUFA public workshop, and the cycle begins again with then updating the priorities for FY24. So using this process, we've been able to establish what the priority initiative areas are for GDUFA III, for FY23. And this includes developing methods for generics to address impurities such as nitrosamines, as well as several priority initiatives relating to enhancing the efficiency of bioequivalence approaches, for example, for complex active ingredients, for complex dosage forms and formulations, for complex routes of delivery, as well as for complex drug-device combination products, and to improve the efficiency of BE approaches for oral and parenteral generics, as well as facilitating the utility of model-integrated evidence to support demonstrations bioequivalence and of course, to expand the use of AI and ML tools. So the outcome from the GDUFA research is scientific advancements that go out to the field in terms of publications and posters and presentations, numerous scientific workshops that are organized, including webinars and training. The research directly supports recommendations in guidances for industry, including product-specific guidances, and it prepares FDA to be able to provide the advice that's needed in pre-ANDA meetings or to be responsive to control correspondences, and in this way really affects ANDA assessment readiness within FDA. Ultimately, the research program directly helps FDA and industry to develop these novel and efficient bioequivalence pathways, even for complex generics. It ensures that FDA can provide that timely and specific scientific advice, again, whether in pre-ANDA meetings or control correspondences, and ultimately the research program facilitates patient access to safe, effective, and high-quality, affordable generic products, particularly for complex generics. So I hope you've come away with a good understanding of why the GDUFA Science and Research Program is essential. Is it essential to A, develop novel and efficient BE pathways for generic product development, including complex generics? Is it essential to ensure that FDA can provide timely and specific scientific advice in pre-ANDA meetings and controlled correspondences? Is it essential to facilitate patient access to safe, effective, high-quality, and affordable generic products, particularly complex generics? Or is it D, all of the above? And I hope you would say it's D, all of the above. Similarly, I hope it's evident how GDUFA science and research priorities are established and updated on an annual basis. Are they based on public feedback about what research is needed to address scientific knowledge gaps? Is it based upon comments provided to the FDA through public dockets, during public workshops, biannual meetings, and industry working groups, and via the CRCG? Or is it both A and B? And I hope you'd say it's both A and B. Thank you so much.
Good afternoon. I'm Dr. Xiaoming Xu. I lead a group of researchers in the Division of Product Quality Research in OPQ. In the next few minutes, I will continue the discussion on GDUFA research and focus more on the translation of the research findings to support the development of product-specific guidances. As the title indicates, I will limit the scope of the discussion to complex products, as they are often more challenging to develop and hence more likely of needing research. In this talk, I will provide an overview of the Generic Drug User Fee Amendment Regulatory Science Program, or GDUFA program, as well as the Product-Specific Guidance Program. And I will share some recent development in GDUFA research, and specifically the approach that we have taken to accelerate the identification of knowledge gaps and how we initiate research to fill those gaps, which ultimately translate to more timely development of product-specific guidances for complex products. The GDUFA program was first enacted in 2012, then reauthorized twice in 2017, and more recently in 2022 under the GDUFA III. And in addition to aligning the user fees and review timelines, the GDUFA program also allocates resources to the FDA to fund research to facilitate generic development and review. Since 2013, FDA has awarded over 200 contracts and grants, as well as conducted numerous internal research projects led by FDA staff. This research facilitated the development of new tools and methods which helped FDA and industry to evaluate the generic drug equivalence. A key outcome of this GDUFA research is to enable more efficient development and assessment of generic drugs, as well as improve resources for the development of PSG recommendations. And a key aim of the GDUFA research is to disseminate the research findings to improve the general understanding of these products and to aid the generic industry in their development program. Therefore, results from these researches are shared through scientific meetings as well as peer-reviewed scientific publications. The Product-Specific Guidance Program, which began in 2007, aims at facilitating the development and approval of safe, effective, and high-quality generic drug product by providing FDA's current thinking and the recommended approach for demonstrating equivalence for a specific reference-listed drug product. PSGs are published on a quarterly basis on the PSG website, which also include information related to upcoming new and revised PSG for complex generic drug products. One of the key outcomes of the GDUFA research is the development of these PSGs. It's important to know that PSGs are FDA's current thinking, and the applicant can propose alternative approaches that are deviating from those recommended in the PSG, but they should provide justification for the proposed approach as well as including data and appropriate references. All this information can be found in the link below to the PSG website. Product-specific guidance commonly recommend a type of study or a property of the drug product to measure. It is up to the generic applicant to select, develop, and justify the proposed approach. This includes the justification of the development methods and the evaluation criteria used to compare the generic and reference-listed drug products. GDUFA research can help the FDA and the industry in develop and stay informed of new analytical tools for assessing a specific product or a class of products. This is important because some of the properties of the complex products are interrelated and not straightforward to measure or compare. GDUFA research aims at reducing potential scientific and regulatory hurdle by providing more insight into these properties and approaches, and providing the industry with a starting point to their generic product development. To further help the industry developing complex generics, as negotiated in GDUFA III, the FDA has agreed on priorities and goal dates on the development of these PSGs. Specifically, starting October 1, 2022, a PSG will be issued for 50% of the complex new drug applications within 2 years after the approval, and for 75% of the complex new drug applications within 3 years of the approval. A few other commitments related to complex PSGs are also included on the current slide. To meet this new goal, we will need a new process that is more efficient in identifying and addressing any potential complexities and hopefully initiate any needed research early. Now let's look at some of the challenges in developing PSG, especially for complex products. On average, there are close to 100 NDA approved per year that may warrant the development of a PSG. Of those newly approved NDAs, about 26% are considered to be a complex product. These products include varying route of administration and dosage forms, and can meet more than one definition of the complexity as outlined in GDUFA II letter, such as complex active ingredient, complex drug-device combination, complex route of delivery, or complex dosage forms. As we have seen with some recent NDA approvals, complexities can vary greatly between products and across disciplinary areas, and can benefit greatly from a close collaboration across multiple FDA teams. The use of new materials, technologies, and processes in these new drug applications also highlight potential scientific gaps and the need for new and reliable analytical methods, which needs new research and innovation. Furthermore, in GDUFA III, the new goal dates on the PSG development of complex products may pose additional time constraints, especially if additional GDUFA research is needed. So, early engagement of research and review will be crucial. Lastly, taking a life cycle approach towards the development of generic necessitates better communication and collaboration across teams and with the industry. I will use one example to illustrate the importance of starting research early. As you may have heard yesterday, cyclosporine ophthalmic emulsion approval in early 2022 was a very notable example of the impact of the GDUFA research. Shown on this slide is the timeline of the approval overlapping with research as well as the PSG development. Research generated crucial supporting data which allowed for assessment of product-specific quality attributes, best measurement practices, and assessment criteria to support the demonstration of the bioequivalence. However, it took nearly 10 years from the original NDA approval to develop scientific recommendations, and another 9 years to translate that to first generic approval. Needing such a long time to approve one drug are rare, but it does highlight the challenges with research, especially the time it takes, as well as the need to timely translating research findings into regulatory actions. The question that we have asked is how we can speed things up and doing it more efficiently. Prior to the GDUFA III rollout, FDA has established a new internal program to identify and direct research to support the complex PSG development. There are 10 subject matter expert teams specializing in various complex areas, with members scattered across 9 CDER offices. Within the last 2 years, this pilot program has conducted 59 triage meetings on newly approved complex NDAs, and already identified 8 that need additional research before a PSG can be developed. Now, briefly, a few more details about this program. It is established specifically to support the complex product PSG development. It includes relevant subject matter experts from both OGD and OPQ. And most notably, the program connects research staff directly with regulatory assessors as well as policy groups to discuss challenges and the needs for each newly approved complex NDA. The teams are created based on complexity areas such as ophthalmic, inhalation, and dermatological, so each team is very familiar with the topic area. Each SME team is tasked with making decisions on complexity and gaps to the PSG development and approval. If a gap is identified, the team also decides on the objective of the research. Afterwards, a dedicated research project is created to generate necessary evidence and to fill the knowledge gaps. And this could be a research project conducted internally within the OPQ lab, or could also be awarded externally through the GDUFA research program. We started piloting this program in 2021, and it became fully operational from the start of the GDUFA III. As mentioned earlier, including the pilot phase, we've already completed 59 triages and already initiated 8 research projects. Here is another view of the program in terms of the timeline. As we know, research can take time, so it is important that we start triaging the NDA product soon after the approval, and through the triage process to narrow down the products, especially the complex ones that can benefit from targeted research. After we complete the research, knowledge gained during the research will need to be translated to recommendations, which will then be put into the PSG. And the example of the recommendations may include new methods or new approaches to demonstrating bioequivalence. I want to provide 2 examples of the SME triage. One is an ophthalmic emulsion of cyclosporine, and this product was approved in June of 2021. And the triage meeting was conducted in October of 2021. At the triage meeting, the SME team collectively decided that there is sufficient understanding from the previous research and the precedents, and therefore recommended immediate development of the PSG. As you can see, the PSG for this product was published in August of 2022, just 14 months since the NDA approval. Another example is a little bit more complex. This product is an ocular insert of dexamethasone with sustained release properties. The NDA was approved in November of 2018, and the decision from the triage team was that research is needed to better understand the product before we can recommend a BE recommendation. The identified gaps are primarily in the area of material characterization and in vitro measurements, and this work is currently ongoing, and we hope to complete it soon and share the findings with the public. As part of the GDUFA III commitment, the agency aims to further improve the forecasting of the PSG publication, which includes both the non-complex and complex products. And this helps the agency communicating the priorities as well as the timelines of the PSG development. which allows the industry to better plan their generic drug product development program. This forecasting list is updated every quarter, and the link is provided below. As you can see, GDUFA research has a very significant impact not only on the ability of the FDA to develop product-specific guidances and to support the pre-ANDA program as well as the ANDA assessment, but is also a valuable resource for the generic industry. In closing, Product-Specific Guidance Program provides FDA current thinking on the type of studies and information to support the development and approval of safe, effective, and high-quality generic drug product. GDUFA research plays a critical role in the generation of evidence and knowledge and supports the timely development of the PSGs. I want to thank a few people who helped me putting together the slides, and also my colleagues at OPQ and OGD in contributing to the success of the PSG program. And thank you for your time and attention. For the challenge question, GDUFA research can facilitate A. PSG development, B. ANDA assessment, C. generic product development, or D. all of the above. And the correct answer is D, all of the above. Thank you.
Good afternoon, everyone. This is Liang Zhao. I'm the director in the Division of Quantitative Methods and Modeling, Office Research Standards, OGD, CDER FDA. To enhance the communication in using modeling approaches in ANDAs, today I'm going to share an exciting new initiative from us, the Model Integrated Evidence Industry Meeting Pilot between FDA and the generic drug applicants. As you can tell, quantitative method and modeling is an indispensable part for all the key components in terms of generic drug development and approval. MIE refer to using model-generated information, such as the virtual BE study result, not just to plan a pivotal study, but to serve as critical evidence. In terms of a tool set, there are many three classes modeling simulation techniques residing in OGD. The PBPK models for orally administered drug and non-oral drugs. The pharmacometrics tool sets for PK-PD relationship and a population-based model. The big data tool set include machine learning, analytics for complex mixtures, systems pharmacology, risk-based model, and models for business process. Before we proceed further, let's take a quick look on the modeling simulation impact in regulatory activities in the new drug side. Here we want to pay attention to the MIDD paired meeting pilot program. This program is a joint venture between CDER Office of Clinical Pharmacology and the CBER Office of Biostats and Epidemiology. It's a dedicated forum for regulatory interaction on MID applications in specific drug development programs. So it's mainly focused on three areas, including dose selection and optimization, clinical trial simulation, and mechanistic safety evaluation. Usually, they have received 2 to 4 proposals per quarter, and it takes about 4 months to have a meeting into completion. Under this pilot program, you can see in GDUFA 6, there have been a total of 66 meeting requests. The meeting requests plateaued around the third year of GDUFA 6, and you can appreciate in the last 2 consecutive years, there have been about 16 meeting requests annually. The MIDD paired meeting program is welcomed by industry as reflected by the paper with combined input from different companies. The benefit here include time and investment savings, alignment, and clarity. I would want to stress that modeling and simulation methodologies are usually associated with complex techniques that entails high demand on alignment and clarity between industry and the agency. From the generic side, we have also witnessed an increase in number of pre-ANDA interactions with model-focused topics since the start of GDUFA II period in 2018. It also peaked at the third year of this GDUFA period. During years 2020 to 2022, we've seen the number of interactions ranged from 13 to 16 per year. On average, we have been conducting 1 to 2 modeling simulation workshops for generic drug development per year since GDUFA II. The latest one was held on October 2022 on utilizing modeling approaches to support generic drug development. More than 1,700 people registered for the workshop, with approximately more than 800 people attended different sessions on the day of the workshop. The workshop audience profile ranged from product development, clinical study, and regulatory affairs. We have also conducted post-workshop surveys regarding using MIE for generic drug application. Most of the survey questions are regarding feedbacks toward the workshop itself. For example, how valuable did you find the content in the presentations during the workshop? Did the workshop directly address issues or challenges you're facing? Did the workshop enhance your understanding of regulatory expectations for MIE approaches? How has this workshop impacted your organization's preparedness to submit a modeling component in the ANDA? You can see the majority of the responses are very positive. Of note, one of the most important questions is, how valuable would you find a communication channel with the FDA to discuss MIE approaches relevant to the use of the same model or modeling strategy across multiple submissions for complex drug products? There's no respondents that know that we don't welcome an additional communication channel, and the majority of the responses stated yes for both. With the increasing number of pre-ANDA interactions over the past 5 years, and a constant calling for more dedicated discussions on model-focused approaches received through FDA, CRC workshop, and other venues, I'm happy to announce the launch of a new meeting mechanism, the MIE Industry Meeting Pilot, to be launched in October. This new pilot program is to provide industry with meetings or opportunities for early interaction for science-driven topics using MIE approaches for BE establishment to facilitate generic drug development and regulatory decision-making. The vision of the pilot program is to enhance scientific communication between generic drug industry and FDA on using a broad range of quantitative methods and modeling techniques to address complex generic drug development issues or questions that are either out of the scope or cannot be sufficiently addressed by the existing pre-ANDA and ANDA scientific meetings. Specifically, the pilot MIE meetings will focus on discussing scientific and technical topics of using MIE strategies for BE establishment. For example, the feasibility of using a modeling approach, the details in model building, and/or model verification and validation strategies. While pre-ANDA meetings will be focused on more general scientific and regulatory issues. Currently, we see there could be 3 major classes of topics that can be discussed in the MIE pilot meetings. First, the MIE-focused approaches that are innovative in nature for BE establishment that are not eligible or appropriate under the existing pre-ANDA and scientific meeting programs to address complex issues pertinent to a singular product or multiple products. For example, common strategies for validating a computational fluid dynamics model platform towards predicting regional depletion of inhaled aerosol from a variety of orally inhaled drug products. Secondly, non-complex products with complex modeling approaches supporting BCS-based biowaivers and other BE study waivers that are outside the current recommendations. Thirdly, novel data analytics tools such as modeling methodology advancements or new applications of modeling approach. For example, the equivalence assessment of complex particle size distributions, new quantitative approaches for sameness assessment, including API sameness, and application of novel data analytics approaches, for example, machine learning methodology for equivalence assessment. Since a new meeting type to guarantee a high-quality outcome and a meaningful discussion between FDA and the industry, we are not going to grant more than 5 meetings per year. The target pilot meeting launch time will be October 1st, 2023. Based on our past experience and understanding of the pain point from industry, we are foreseeing 2 areas of focus that are the complex products and non-complex products. For complex products, they can include locally acting products where comparative clinical endpoint studies are usually not sensitive and very costly. Long-acting injectables where the study duration and PK variability will pose a challenge. And oncology products where the patient recruitment and the state crossover study design will make the study super complicated, sometimes impossible. With modeling simulation, the tool side can critically contribute to counter all those challenges that cannot be easily addressed by conducting actual experiments. For non-complex products, we are foreseeing novel biowaiver requests supported by mechanistic modeling for approaches that are not currently recommended by PSGs. We hope, in close collaboration with industry colleagues, we can generate tremendous impacts and benefit out of this pilot program. For industry, we can enjoy the benefit we've seen from the new drug development in terms of time and cost cut, and in terms of alignment and clarity in terms of using modeling approaches to abbreviate complex bioequivalence development programs. Those programs can critically benefit the early market entry for long-acting injectables, locally acting products, including already inhaled drug products, topical dermatological products, and oncology products. From the agency side, we can use this program to increase the efficiency to handle multiple products in a single meeting, reducing number of cycles for drug approval, establishing an ecosystem with industry to develop effective BE approaches, where industry can serve as the equal partner with FDA to roll out novelties and innovations in using modeling approaches to counter all those challenges posed for various products from different angles. This pilot would not have been possible without the collective efforts of exceptional professionals. First and foremost, I want to thank the MIE pilot working group, including colleagues from ORS OGD, OB OGD, OGD communication staff and policy, OCP OTS, and OPQ. Special thanks to ORS and OB leaderships, Dr. Robert Weinberger, Lei Zhang, and Patha Roy for your guidance, vision, and instrumental support. Thanks to our exceptional product managers, Maria, Shivangi, and Sarah, for your organizational skills, meticulous planning that have kept us on track. Moreover, I wish to acknowledge the individual contributions of our modelers in TQMM. Your expertise, dedication, and innovative thinking have enhanced the quality and depth of the program. At the end, I would also like to thank colleagues in industry, consulting, and academia for your contributions and effort to make it happen. With that, I will see you again in the panel discussion.
Thank you all for the great presentations. As a reminder to our audience, if you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. In addition to our presenters, we'd like to welcome the following panelists: Dr. Darby Kozak, Deputy Division Director, Division Therapeutic Performance 1, Office of Research and Standards OGD, Dr. Robert Leinberger, Director, Office of Research and Standards OGD, and Dr. Zhen Zhang, Master Pharmacologist, Division of Bioequivalence 1, Office of Bioequivalence, and OGD. We have some questions coming in right now. The first group of questions are directed to Dr. Sam Ronnie, and here's the first question for Dr. Ronnie. Why was it necessary for GDUFA to have a science and research program?
Thank you, Ray. That's a good question. Well, the GDUFA Science and Research Program is particularly important for complex products, which are harder to develop as generics for several reasons. One of the main reasons is because pharmaceutical scientists and clinicians may not have an adequate understanding of all the factors that can impact the therapeutic performance of these products. And without that knowledge, generic product developers may only be able to demonstrate the bioequivalence of their prospective complex generic product using approaches that are quite challenging. And this represents a barrier to the availability of complex generics. But the GDUFA Science and Research Program enhances our understanding of all those factors and for numerous different types of dosage forms, actually, especially complex products. And so it supports the development of innovative methodologies and more efficient bioequivalence approaches. So in this way, the research program reduces the hurdles for complex generic product development while maintaining really high standards for product quality and ensuring that all generics are as safe and effective as their reference brand name products.
Thank you, Dr. Ronnie, for responding to that question regarding what happens with the GDUFA research program and how transparent is it to the public? Dr. Ronnie, we're not hearing any sound right now, so what we'll do is we'll give another chance. I saw some reconnections on the mics, and we'll see if you can hear now, and we'll read the question again. What happens with the GDUFA research program regarding its transparency to the public?
So this is Rob. I can answer that question. And so, a lot of the transparency are things that Sam mentioned in his talk. So we publish every year the newly awarded grants and contracts, right? That gives a view into all of the external collaborations. But also every year we publish a science and research report on the GDUFA program that goes into detail, and it includes every research project across CEDAR that's supported by the GDUFA program. So this gives a deep and clear understanding of all the scientific activities that the research program is presenting. So, there's no other research activities that are going on. That's a comprehensive report about our research activities. So I think those are two places. If you go to the GDUFA Science and Research webpage, you'll see links to all of those different reports. So it's very clear, both things at the beginning and as we get outcomes at the end of each year for the projects that are accomplished.
Thank you, Dr. Leinberger, for giving us a deep dive and answering that question in regards to transparency. We're going to check back in with Dr. Ronnie for another question, and here's the question. What has the work performed by the GDUFA research program achieved?
Okay, thanks, Ray. So the program has brought together and focused the world's foremost experts in their fields, including scientists and clinicians from industry and academia and the FDA. And together, the program works to build scientific bridges across knowledge gaps that really held back the development of many generics, particularly complex generics. And it does so in a very focused way to establish practical and efficient pathways for pharmaceutical manufacturers to develop generic drugs that were, previously challenging or even unfeasible to develop, and then to make those available for patients. And these scientific advances, they establish the new bioequivalence standards I talked about before. They make it possible for generic drug developers to meet with FDA during product development phase to receive really clear, hopefully reliable, evidence-based recommendations about how to approach their product development. And then we also, as Rob was speaking about, the outcomes provided in our scientific presentations or in our literature publications, really, or that we talk about during workshops, including this one, all of these provide tools to industry. So these have all been tremendous achievements, I think, of the program. And this means that applicants receive basically free scientific and technical advice that helps them to prepare their submissions in a manner that's compatible with the most current scientific insights and regulatory expectations. And it works. We just in the last several, even during this fiscal year, we've approved products including the first generic budesonide and formoterol furoate dihydrate inhalation, referencing Symbicort. We've approved the first generic cyclosporin ophthalmic emulsion, referencing Restasis, and several other products that would simply not have been possible for American patients to have access to had it not been for the research under this program.
Thank you, Dr. Ronnie, for answering those questions. Moving on to our next panelist. Got a few questions that came in from Dr. Xiaoming Xu. And here's the first question for Dr. Xu. For the new process you described for triaging new NDAs, does it also cover products approved prior to GDUFA III, or is it only for GDUFA III?
Yeah, thank you for the question. So as I indicated in the talk, this internal program was designed to accelerate the translation and identification of the gap, really to speed up the kind of knowledge transfer and to facilitate the development of the PSGs for so those products covered under the GDUFA III, because we have a more shorter timelines to publishing the PSG, so we need an improved process for those identification and conducting the research. However, as also laid out in the GDUFA III commitment letter, the agency will continue to develop the PSGs for those products that approved prior to GDUFA III. But we were prioritizing the development for GDUFA III because of the goal dates. But again, we will be continue to work on the product without PSG that are complex and continue to make them the guidance available. They will be published on the PSG website. Hopefully that answered the question. Thank you.
Thank you, Dr. Xu, for the response. We do have a couple more questions. And here's the next question. How do we know if FDA decides research is needed before a PSG can be developed?
Thank you again for the question. In general, we internally, when we identify research that to support the PSG development, sometimes it's not only for one product. Sometimes it can be for a class of product because they share common challenges or difficulties. So we may construct a project to support sometimes more than one NDAs. So it's very difficult to pinpoint exactly which NDA supporting the PSG, or even to disclose the initiate of the project to the public at that time. However, as Dr. Leinberg mentioned to the previous question, that the annual GDUFA research report is actually a very valuable resource, has a lot of information about what has been completed, the research project, as well as those that are newly initiated or ongoing research project, whether it's internal or whether it's through the funding through the external. So you may find some of the information in the annual report, for example, on a new project, that a new area of research that we're looking at. So I think that can provide some information that can be beneficial to make it transparent. And also, one last point is that the gap that we identified internally could be different from the one that you may identify during your product development. So it's certainly beneficial if you also provide input and recommendation through different channels, communicate to the agency about what do you think would be the gap or the need that may be beneficial for your development program. So through a channel like annual GDUFA workshop or other ways that you can bring those also recommendations to the FDA as well. So thank you.
Thank you for responding to that question. We do have one more question for Dr. Xu, with a combination question of asking you to respond first and then passing the mic over to Dr. Darby Kozak for a follow-up response. And here's the question. Is there any channel for external stakeholders to contribute to the research as well as to support future PSG development?
Yeah, thank you. So I will as indicated, I will comment on part, and I will ask my colleague Darby to chime in here. So we always welcome input and contribution, especially research is a collective endeavor. It's not something that easy. We recognize that, so we always welcome collaboration and input from outside stakeholders. So one example I want to give is a Center of Research on Complex Generics, or CRCG. It's really a great resource that can, through the different workshops that they organize, some of the research projects that conducted by CRCG members, and lab demonstration or other interviews. So it really provide a channel to provide that feedback and also obtain some of those what is available through that resource. I think that will be a valuable, at least a channel that I would suggest to recognize and utilize, and that could be really provide input to us, guide us also what may be of interest or of importance, and also we will share information through that CRCGs through different workshops to provide FDA thinking on those topics as well. But I will let my colleague Darby Kozak also maybe to chime in some other possible channels. Thank you.
Yeah, thank you, Xiaoming. So yeah, I think this is actually a really important sort of session for us because there is several ways, and we always are interested in feedback from both industry and the public and academia in general about what PSGs we're doing and what research we're doing and sort of the impact and how to engage with us. So there's actually several ways that everyone can engage, the public can engage with us on the development of PSGs in our research program. Dr. Rainey covered a few of those in his talk. But in particular for the PSGs, if there's like a request or there's not a parts of guidance already posted, industry or public can actually request those through a controlled correspondence. And that can help FDA better know what PSGs are of interest to industry. As well as if there's already a PSG posted and you feel that there's some comments that would be better or recommendations that may be more appropriate or research that can be done in that sort of space, you can actually submit what's comments to the docket at regulations.gov specific to that PSG that gets to FDA sort of reviewers that can then look at that sort of aspect. And that's kind of how you can engage with us specifically on the product-specific guidance program. In terms of the research, like I said, as I really piggyback on Dr. Rainey's talk on some of the avenues that can be done to help us focus the research program. That's that annual public workshop, in addition to the CRGC, like that Xiaoming mentioned, but also a little bit more specific is some of the other routes. You can look at our generic drug science and research website, which is very valuable. It provides a lot of background on upcoming RFAs or grants of interest in research areas that we're doing, and these will be sometimes specific to a PSG or a general area of products that we feel research is needed to help us develop those PSGs. And also there's that broad agency agreement or announcement process. So if you've got a specific research program and you want to then propose that to FDA, you can do that through the BAA process. What's really nice about that is it can be a very specific or tailored research project that you think will be impactful for either that product or that class of products and I think that FDA would be interested in. And those proposals then go to us. We review them, and then depending upon the research priorities and our funding sort of needs, we can then fund those projects. Funded projects then become a part of our collaborative research. So as part of that collaborative research, we would engage with you on generally a monthly basis or a bi-monthly basis to get updates on that research program. So I think this is a real sort of great component about the GDUFA research program is this engagement with our stakeholders and how you guys can really help tailor the program. So thank you for that question.
Thank you for responding to that question. We're going to move on to our next panelist. Next group of questions came in for Dr. Lian Zhou. And here is the next question the first question for Dr. Zhou. Should MIE industry meeting pilots be before or after a pre-A&D meeting?
Thanks, Jeff. Do you hear me okay?
Yes, please go ahead.
Okay. Yeah, before I address this question, I just wanted to make an announcement that the MIE industry meeting pilot has just been announced in the FDA website. It includes the guiding principle detailing the scope, qualification, and the process of the new meeting type. You can certainly get it easily just by using Google search for MIE industry meeting pilot, and it will lead to the page. Regarding the question, the MIE industry meetings can be requested either before or after the pre-ANDA meetings. That means the applicant can have a technical discussion with FDA in the pre-ANDA meeting and can consolidate it into an overall plan, discuss with us in the pilot meeting, then consolidate it into an overall plan to be discussed further in the pre-ANDA meeting, pre-ANDA development meeting. Or after getting the overall modeling pathway discussed in the pre-ANDA development meeting, the applicant can follow up with an MIE industry meeting pilot with technical and data details. Having said that, applicants should not submit multiple meeting requests or controlled correspondence at or around the same time with the same or similar questions. If FDA receives multiple meeting requests or controlled correspondences that contain the same or similar questions, FDA will determine which meeting to grant or controlled correspondence to answer and may deny the others. However, the pilot MIE meetings are a potential venue for discussion on model-related topics during a product development cycle. Thank you.
Thank you, Dr. Zhou, for answering that question. We do have another question came in for MIE meetings, and here's the question. Can I have multiple MIE meetings for the same products or multiple products in the same meeting?
Thanks, Jeff. The short answer to both of them is yes, if it helps your drug development program. It absolutely helps when you have a change in the modeling strategy and analysis plan after communicating with us in the form of a MIE meeting. And if you have a changing in thought and a new development plan, you are more than welcome to schedule another meeting with us. While some approaches can be eligible for discussion with the existing GDUFA scientific meeting processes, such like pre and product development meeting, the pilot MIE meeting is valuable that will focus on discussing scientific and technical topics of using MIE strategies for BE establishment. For example, the feasibility of MIE approach, details in model building and or model verification validation, data strategies for the intended purpose. Certainly can use a single meeting to discuss multiple products if all the products taking the same MIE strategy, and certainly that can gain the industry's efficiency and productivity and in terms of product entry. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we got a question just came in for Dr. Darby Kozak, and here is the question. How can we find out if FDA has done research for a PSG and the results of that research?
Thanks for that question. It's an question we often get from industry about what types of research we've done and how to best locate that. I do want to reemphasize that as with any of our GDUFA research, this is really a starting sort of point, gives a general idea. It's meant to facilitate the generic drug program development of those products. But really it's a starting point, gives you an understanding of what sort of things that we are taking into consideration, and as well as things to start your product development program. But a key thing also with the GDUFA research program is the fact that we try to make and aim to make all the research findings publicly available. And so a lot of you'll find that our research gets publicly disseminated through peer-reviewed scientific research articles. And that helps to get it all into the public domain, as well as to help certify and to reevaluate our research sort of findings to the public at large. We also present at meetings such as this on the research findings, as well as at other events and public sort of scientific sort of workshops and meetings and conferences. But one place that you can always find that sort of resources and things that FDA has published and out there is there's a bibliography of also the publications and presentations we have on our GDUFA Science and Research website. And that also includes at each end of each year, we do the GDUFA Science report. That'll actually tell you what publications and presentations were published that year into those different research priority areas. So I highly recommend checking those components out. And like I said, other aspect is you can always contact the FDA author and they can send you copies of those things. But usually we have everything out in the public domain as much as possible and as quickly as possible. Thank you.
Thank you for responding to that question. Moving on to our next panelist. We just received a question in for Dr. Zhen Zhang. And here's the question for Dr. Zhang. Do review divisions actively participate in the research?
Yes, thanks for the question. Yes, review divisions do actively participate in the research. For example, I'm in the Office of Bioequivalence. Our primary work is to assess bioequivalence studies in ANDA submissions. But our involvement extends beyond the routine review tasks. We also dedicate our efforts to investigating emerging bioequivalence issues and exploring novel bioequivalence techniques that arise during our assessment of ANDA submissions. We actively collaborate with many disciplines on research within FDA, including Office of Research and Standards, Office of Testing and Research, Office of Biostatistics, and many others. Moreover, we place a strong emphasis on transparency in our research activities. We make an effort to share our findings with wider community through various public forums, including this workshop. Our commitment to research allows bioequivalence reviewers to stay at the forefront of the bioequivalence field and also contribute to advancement in bioequivalence assessment. Thank you.
Thank you for responding to that question. Moving back up to the beginning of our panel, receive your question in for Dr. Sam Ronnie, and here's the question. Is FDA planning to update ANDA checklist, especially the clinical section, which is not updated since for long, for at least complex drug products?
Thank you, Ray. I interpret that question to be referring to updating FDA's bioequivalence recommendations for many PSGs, product-specific guidances, especially complex products. The comparative clinical endpoint bioequivalence studies are kind of a traditional way to approach it, although they can be very challenging and sometimes not very attractive to product developers. So I interpret this to be a question about is FDA planning to update more product-specific guidances with alternatives to the comparative clinical endpoint bioequivalence studies, with more efficient bioequivalence approaches like those that involve in vitro bioequivalence approaches, product characterization-based bioequivalence approaches? And the answer is yes, that is one aspect of the program, is to evaluate alternatives to comparative clinical endpoint B studies, or to evaluate more efficient ways to conduct these clinical endpoint BE studies. And the alternatives may include characterization-based approaches, it may include modeling and simulation, it may include a combination of different approaches that collectively make it more efficient or even more feasible to conduct these. And in fact, that was one of the themes of the recent GDUFA public workshop earlier this year, was that integration of evidence from modeling and simulation and from product characterization and perhaps some in vivo data as well in certain cases, to be able to find more feasible and more efficient ways to develop bioequivalence. And certainly that research would eventually be translated into product-specific guidances. There is a forecast list, if people are interested, on FDA's website of upcoming product-specific guidances for generic product development, and that indicates what the forecast is for different PSGs that will be developed in the years ahead, in the year ahead.
Thank you for responding to that question. We have time for one more question. This will be our final question in the session for Dr. Lian Zhou. And here is the question. While recommending the PSGs, which may recommend patient-based BE studies with clinical endpoints, do you also consider the high cost of such trials as one of the inputs?
Thanks, Jeff. I'm glad you asked the question. Also thanks to whoever posed that question to us. As I presented early, that's exact the benefit of using modeling approach. The benefit include cut the cost and the time for the product development program. As Sam just probably mentioned, the utility of modeling simulation in the regulatory setting, the high cost and in terms of conducting comparative clinical endpoint study can be mitigated by virtual trial or study simulations. Basically, if the model being sufficiently validated by the data set, you can conduct a virtual clinical study to assess the formulation effect on the therapeutic performance. Having said that, I think the state of the art is on mainly what the minimal data you would use to validate, verify the model to gain the regulatory confidence. The other thing is how would you evaluate the type of error, meaning if the product are not equivalent, what the chance to conclude bioequivalence with your proposed modeling approach. With that, I really encourage all the stakeholders to discuss those kind of modeling analysis strategies plans with us in the MIE pilot industry meeting pilot program or pre-under development meeting venue. Thank you very much.
Well, that's all the time we have for questions in this panel. Another huge thank you to our speakers and panelists for answering numerous questions that came in from our audience. We'll now enter into our final break until 3 p.m. Eastern Time. Enjoy your break. Welcome back from the break. We're on the home stretch now, right into session 8, global collaboration to support efficient generic product development and regulatory assessment. We do have a slight change on the agenda of presenters with Dr. Sarah Ibrahim presenting first. Dr. Ibrahim is Associate Director for Global Affairs and OGD. Our next presenter is Dr. Nullifer Tample, Associate Director for Scientific Quality, Office of Bioequivalence and OGD, and we're welcoming back Dr. Leizan to present on FDA-EMA Parallel Scientific Advice Pilot Program for complex generic and hybrid drug products. And finally, we welcome Brian Folian, Deputy Director, Office of Study Integrity and Surveillance in the Office of Translational Science for CEDAR. Please join me to welcome our first presenter, Dr. Ibrahim.
Thank you for the kind introduction. I'm Sarah Ibrahim, the Associate Director for Stakeholder and Global Engagement in the Office of Generic Drugs, Center for Drug Evaluation and Research at the US FDA. And today I'm delighted to share with you experience from the generic drug cluster. In 2022, the worldwide market for generic drugs achieved a value of $343 billion. Projections indicate that this market will grow to reach $460 billion by 2028, with a compound annual growth rate of 5% during the period from 2022 to 2028. This growth is largely driven by an aging population and an increase in chronic diseases. Now, in an effort to manage healthcare costs, governments in developed nations are actively promoting the use of generic drugs. These medications provide the same therapeutic benefits as their brand name counterparts, but at a significantly reduced cost. Meanwhile, in developing countries, generic drugs offer a potential solution to the challenges of affordability and accessibility in the healthcare sector. By providing cost-effective alternatives to brand name medications, generics can help overcome financial barriers and improve access to essential treatments. In the past, the United States was the primary source of medication production. However, over time, the manufacturing of drugs, especially active pharmaceutical ingredients, has shifted overseas. The cost benefits offered by countries outside the U.S., such as lower labor costs, cannot be matched by traditional pharmaceutical manufacturing technology in the U.S. Now, to illustrate how global the generic drug supply chain has become, we analyzed the location of API manufacturers for marketed ANDAs with active and pending manufacturers. From 2012 onwards, there has been a steady increase in the percentage of ANDAs where an API manufacturer is based in India. Conversely, the percentage of ANDAs where an API manufacturer is located in the United States has been on a decline since 2011, with only a few exceptions. Despite the global shift in drug manufacturing, the United States continues to hold the largest portion of ANDA finished drug formulation manufacturers for marketed ANDAs. However, there has been a consistent increase since 2012 in the percentage of ANDAs where finished drug formulation manufacturers are based in India. On the other hand, the share of ANDAs where the finished drug formulation manufacturer is located in the United States has also been decreasing since 2011. Now, given the global and complex nature of our generic drug supply chain, the FDA's international presence and engagement play a crucial role in securing our supply chain. The FDA's regulatory system ensures the quality and safety of drugs produced globally, even before these products hit the market. The FDA stands by the rigorous standards and regulatory inspections that ensure the safety and quality of all generic drugs, affirming that they are just as safe and effective as their branded counterparts. The Generic Drug Global Affairs Program is an initiative that aims to strengthen the FDA's global collaborations. Effective communication with stakeholders is a cornerstone of this program. by maintaining open lines of communication, ensures that all regulatory parties are informed about regulatory requirements, changes in policies, and emerging trends and challenges. The program also focuses on identifying opportunities for early regulatory alignment. This involves working closely with international regulatory agencies to discuss standards and procedures for generic drug approval under development. Such alignments can pave the path for harmonization that may expedite the approval process, reduce duplication of efforts, and ensure consistent quality of generic drugs across different markets. Proactive engagement with regulatory counterparts is another key aspect of the Generic Drug Global Affairs Program. The FDA actively collaborates with other regulatory agencies worldwide to share knowledge, exchange best practices, and coordinate efforts. The Global Affairs Program has a number of engagements and initiatives focused on international dialogue that promotes consistency, bringing efficiency in the harmonization process. Strategically taking a collective approach to establish more effective, consistent channels of information sharing and communication. In its inaugural year, the Office of Generic Drugs established the Generic Drug Cluster, a critical initiative aimed at fostering global partnership with leading regulatory agencies worldwide. This initiative is designed to address global challenges and opportunities in the generic drug industry. The Global Affairs Program focuses on establishing targeted outreach to support FDA regulatory needs, involving communicating effectively with global stakeholders. In addition to targeted outreach, the program also emphasizes on regulatory strengthening and capacity building of regulatory systems. This involves providing technical assistance to regulatory agencies worldwide with the aim of enhancing their ability to oversee the safety, efficacy, and quality of generic drugs. The harmonization of regulations plays a crucial role in the global supply chain of generic drugs, as it helps to eliminate redundancies and inefficiencies. By aligning regulations and standards across different countries, we can ensure that generic drugs are manufactured to consistent quality and safety standards. This not only protects patients from substandard or counterfeit drugs, but also reduces the time and cost associated with bringing new generic drugs to market, thereby increasing patient access to affordable medications. Harmonized regulations enhance transparency and traceability in the supply chain for generic drugs. This improvement aids in monitoring and reporting adverse events related to generic drugs, allowing for early identification of potential safety concerns and the implementation of appropriate measures to safeguard patients. The intricacy of global harmonization is often attributed to the variety of legal systems, political values, and economic structures that exist across different countries and regions. Achieving alignment in regulations and policies often necessitates substantial efforts to reconcile these differences and balance various interests. The formal ICH procedure is a structured process comprising of 5 steps, ranging from consensus building, which involves regulatory consultation, discussion, and finalization, to committee adoption and regulatory implementation. Now, the journey towards global harmonization commences once regulations have been established. This rigorous process involves assessing regulatory differences, conducting scientific and technical negotiations, and bridging the gap between existing regulatory requirements and expectations. The Office of Generic Drugs Global Affairs acknowledges that prospective harmonization efforts, particularly for complex generics, can strategically bridge our regulations while they're being established. Now, global harmonization is a multifaceted and an ongoing process that requires cooperation among various stakeholders. The Generic Drug Cluster serves as a unique platform for maintaining continuous dialogue by fostering a shared vision and promoting discussions as regulations are being established. Now, as I mentioned in its first year, the Global Affairs Program launched two significant initiatives, the Generic Drug Cluster and the Parallel Scientific Advice. I'd like to use this opportunity to not only update you on these programs, but also share the insights gained from both. Globalization profoundly changed the pharmaceutical sector, creating unique regulatory challenges that require successful collaboration and continuous dialogue. Regulators agree in principle of the importance of global harmonization of pharmaceutical regulations, aligning divergent regulations that create hurdles that can be mitigated by leveraging ongoing dialogue and collaboration, where harmonization could also take the form of a slower convergence, with regulatory bodies gradually aligning their standards and requirements with the vision of regulatory capacity building of the agencies that will be responsible for implementing the harmonized regulations. Achieving harmonization of pharmaceutical regulations requires a collaborative effort among stakeholders, convergence of best practices, regulatory capacity building, effective implementation, and an ongoing monitoring and evaluation. Addressing the complexities involved in the development of complex generic drug products requires a collaborative approach among industry stakeholders, academic institutions, and regulatory authorities. The creation of a robust regulatory framework that facilitates market access while ensuring patient safety is contingent upon dedicated research and investment. Such advancement in research could potentially enable generic drug product applicants to rely more on alternative methods, thereby minimizing unnecessary exposure of human subjects. This can be achieved through the extensive exchange of scientific information, streamlining regulatory requirements, and clarifying divergent regulations with our regulatory global partners. Providing developers and manufacturers with the necessary channels can expedite product development and enhance patient access to essential medications. Now, as an agency, we place great value on global collaboration. We acknowledge that for certain products, such as complex generics, that undergo a more intricate development process, access to multiple markets can incentivize companies to proceed with a development program. Now, one of the key benefits of the cluster that has been identified in its role in facilitating a global response, and we actively monitor and analyze emerging trends and developments on the global stage that could potentially impact our regulatory agencies and its policies. This allows us to review existing or new policies to address any gaps, challenges, opportunities, and risks posed by these global changes. We collectively discuss and assess the impacts and implications of these global changes, and the process may involve scenario analysis, risk assessment to ensure we're prepared for various outcomes, ensuring no disruption to medication access to patients. We evaluate and report on the performance and outcomes of our policies in relation to the goals and objectives of our regulatory agencies, as well as the global changes. These ongoing evaluations ensure that our strategies remain effective and responsive to the ever-evolving global landscape. With a globalized generic drug supply chain, global events, international commitments, and policies that arise have repercussions on the drug supply chain and can differ based on the nature, intensity, duration, as well as the extent of event or change. However, these impacts can have drug shortage consequences that affect the availability or accessibility of drugs for patients who require them. Having a quality or safety or efficacy concerns could potentially lead to adverse events and recalls. Compliance challenges that arise in adhering to these regulatory requirements or standards of different countries or regions involved in the drug supply chain. Global changes in policies can significantly impact the regulations of generic drugs, affecting aspects such as innovation and development. With responsive changes in regulatory policies can either encourage or hinder innovation, impacting the development process due to the possibility of arising divergent regulatory responses from our jurisdictions, thereby affecting access to generics globally. As I mentioned, the FDA has established a multi-country forum, or the generic drug cluster, to foster a shared understanding of generic drug regulatory requirements amongst what is currently 7 regulatory agencies and to promote scientific alignment. The generic drug cluster serves as a platform for the world's leading regulatory agencies to facilitate harmonization through early communication and collaboration related to generic drug development. The cluster aims to establish proactive alignment on specific proposed generic drug product review challenges or broader drug class issues through open communication. The cluster meetings have been instrumental in comparing and evaluating regulatory approaches, assessing similarities, and identifying differences. These regular discussions include current review challenges and have led to the resolution of several regulatory obstacles to approvability. Discussions also extend to regulations and guidances under development with regular updates on shared various topics. Now, one significant outcome of these discussions has been progress made in model integrated bioequivalence for long-acting injectable products. Agencies have also committed to sharing data integrity concerns at early stages, allowing for collective awareness of ongoing investigations within respective agencies. Another crucial aspect of the generic drug cluster is its role in anticipating and collectively discussing approaches and challenges due to emerging global regulatory challenges. These could be attributed to global and environmental policy changes and their impact on the generic drug supply chain, prompting the identification of and coordination of multilateral responses to emerging global issues. The generic drug cluster, as I mentioned, is a unique forum underpinned by the confidentiality commitments that promotes the leveraging of collective resources among regulatory agencies. This includes sharing knowledge, expertise, and experiences. The cluster enables our agencies to transition from being data-rich to fostering shared knowledge that enhances our individual capabilities in both depth and breadth. While the cluster typically hosts quarterly meetings, the need for more frequent communications had led to significant increase in ad hoc meetings. And in these meetings, we have recurring discussions that are held to compare and evaluate regulatory approaches, expectations, similarities, and differences. The cluster participants also share data on human testing of certain drugs where discrepancies exist in the testing population. The assessment of that combined data could support converging recommended testing populations. So if no statistically significant difference is found between patient and healthy volunteer populations, it opens up the possibility for a wider pool of testing participants for these classes of drugs. Now, we published a one-year cluster accomplishments and contributions, and over the past year that represents the cluster's second year, we continue to make significant accomplishments and contributions, and we continue to build upon these efforts. Some of the topics that we have been able to discuss include evaluating the sameness of active pharmaceutical ingredients, particularly focusing on peptide functionality using in vitro and in vivo assays, ensuring that the APIs in generic drugs are equivalent to those in their brand name counterparts. We also continue to discuss complex products that currently have no generic competition, a critical issue that impacts the availability, access, and affordability of these drugs for patients. We have also been working to understand other generic cluster member agencies' approval standards and rationales behind them, as well as their review experiences. This includes specific discussions on partial AUC recommendations for certain products. Member agencies also share their perspective and experience with use of foreign comparator. We also compared narrow therapeutic index classification and bioequivalence approaches among different agencies, discussing challenges with assessing data from alcohol dose dumping studies, as well as share safety data where we compared and investigated bioequivalence studies conducted in healthy subjects versus patient population, especially for complex products where bioequivalence subject variation exists among our agencies. We also conducted investigations on the pharmacokinetic profiles of parenteral nanotechnology products, amongst a number of other recurring topics. Regulatory harmonization and aligning regulations and standards across different countries or regions create a more efficient and effective global supply chain for medications. Regulatory authorities must have the necessary competencies, capacity, resources, and scientific knowledge to deliver their harmonization mandates effectively. And the goal of regulatory strengthening is to improve the ability of regulatory authorities to ensure the safety, quality, and effectiveness of generic drugs. Targeted outreach plays a crucial role in reducing the regulatory burden on the Office of Generic Drugs and ensuring a more efficient review process. It involves communicating effectively with regional stakeholders, helping clarify regulatory policies and procedures that are responsive, targeted, and can help streamline the review process by clarifying regulatory requirements and expectations upfront, helping reduce the number of review cycles for a generic drug application to be approved, saving time and resources for both OGD as well as patients, fostering a more collaborative relationship between OGD and regional stakeholders can enhance transparency in the regulatory process. And our first question, the generic drug cluster 1 consists of 7 regulatory agencies. 2 is a confidential forum for information and knowledge sharing. 3 allows for a dialogue that builds a more robust, coherent regulatory infrastructure for generic drugs globally. Or 4, all of the above. And the correct answer is all of the above. Challenge question number 2. Cluster members focus the discussion on 1. Guidances under development. 2. Agency actions to address global supply chain and development challenges. 3. Current and past ANDA review challenges. Or 4. All of the above. And the correct answer is all of the above. On behalf of OGD, thank you.
Thank you for the introduction. Good afternoon. It's my pleasure to join this webinar to talk about the first harmonized bioequivalence guideline for immediate release solid oral dosage forms, M13A, from an implementation perspective. The ICH Expert Working Group is currently working to finalize the draft M13A guideline. Simultaneously, the FDA is also proactively considering measures to ensure the smooth implementation when the guideline is finalized. And we anticipate that the industry would also be eager for the FDA to implement the guideline without long delays. In that regards, FDA has started to identify some topic areas that would benefit from a proactive approach, and I'll talk about them. I'll also share the ongoing research at the FDA that will help to translate some of the more challenging recommendations in the guideline into meaningful regulatory approaches. The learning objectives are to provide an overview of the M13 guideline series and the progress made today, discuss the next steps by the expert working group, the EWG, for advancing M13A towards adoption by the ICH Assembly, and to discuss the strategic planning for future implementation of M13A. That includes understanding the research areas of interest to the FDA to support the regulatory decisions. Now, EWG is developing the M13 guideline as a 3-part series, each with their respective scopes. M13A is the first guideline in the series to harmonize the scientific and technical aspects of the study design and data analysis to support bioequivalence of IR products. 13B, the next guideline in the series, will harmonize the bioequivalence requirements for additional strengths of a product line, including the biowaivers of additional strengths. And then 13C will cover replicate study design and other complicated data analysis with specific focus on highly variable drugs, narrow therapeutic index drugs, and other complex BE study design and data analysis, for example, the adaptive study design. Now, the 3 series of the guideline, they are planned for development in a sequential order, following the ICH process for harmonization. So here, the 5 steps of the process are shown here, starting with drafting of the technical document and going up to the implementation of the final guideline. So let's look at the progress of the M13 series. 13A is currently at step 3 of the process, which is the regulatory consultation period. This guideline was subjected to public consultation by the member parties in the first quarter of the year, and the comment periods for all the members have closed, and the expert working group is currently working through those comments. 13B, it is currently at step 1 of the process, which means the technical document is under development. And the 13B expert working group is working towards building consensus on different aspects of the guideline through rigorous scientific discussions. And then 13C, it will start after the 13B reaches step 2, when the draft is endorsed by the Assembly. Now let's see how the EWG is working to finalize the guideline for adoption, which is the step 4 of the ICH process. First, the expert working group members will be reviewing and addressing the comments through consensus, and they'll be making necessary revisions and edits to the guideline to support the finalization. The pie chart here provides an overview of the comments that were received for the 5 sections of the guideline. In all, there were 789 unique comments we received, and about 77% of those comments are for Section 2. That's not surprising, since the crux of the guideline is in this section that includes study design and data analysis for establishing bioequivalence. The next highest number of comments, about 10%, are on the specific topics which are covered in Section 3. In addition, the working group is considering to develop a Q&A document after the guideline is finalized. We acknowledge that the guideline provides broad strokes on certain aspects. Therefore, a Q&A document to clarify the scientific thinking and the rationale, and perhaps using some examples to further explain certain recommendations can be helpful. Other item under consideration is developing the training materials to facilitate the regional implementation and ensuring consistency for the member regions. I'd like to share that we also received a couple of positive comments, like the one that I'm showing here. Harmonizing on different scientific viewpoints takes a lot of time and efforts. And on behalf of the expert working group, I'd like to take this opportunity to thank you for the support. So coming to implementation, once a guideline is adopted, the ICH expectation is for the regulators to implement the guideline within 6 months. So anticipating the needs and preparing for the implementation is necessary to avoid the delays. Here are some of the tasks that FDA anticipates will need to be addressed. First is developing the training materials. And that includes internal training for our assessors and also training for external stakeholders. updating the relevant PSGs to align them with the recommendations in the M13A. We also have to update the general bioequivalence PK endpoint guidance for ANDAs. This guidance currently includes recommendations for both IR and MR products. Then we would need to assess if the BE summary tables need to be updated so that they align with the recommendations in the document section of the guideline. And then lastly, conducting research on select topics so we can apply good science in translating some of the more challenging recommendations into meaningful regulatory approaches and decision making. In the next few slides, I'll cover the ongoing research at the FDA. Our research focuses mainly on topics that present either significant change from the current FDA practice, such as the fasting and fed study, or where the agency has limited experience, for example, demonstrating bioequivalence in presence of the acid-modifying agents, which is a recommendation in Section 3.4 of the guideline under the pH dependency, or utilizing the modeling and simulation approach to support the deviations from the recommendations in the guideline. All this research is expected to feed directly and inform our scientific thinking and the regulatory decisions. So first, I'll talk about the bioequivalence under fasting and fed conditions. The draft guideline notes that food can have a differential formulation-dependent impact on the absorption of certain drugs. Applicants should have a thorough understanding of the comparator and the test formulations to determine if testing under fasting and fed is warranted. And the guideline also mentions that the rationale for selection of the study design with regard to fasting and fed may be supported by appropriately validated and qualified either PBPK or semi-mechanistic absorption models. Here I want to share a survey that the generic industry had conducted earlier to evaluate if testing under both fasting and fed conditions is necessary for IR products. The results were shared at the AAM meeting in 2019, and there is a link on the slide for that. And they also shared the results with the ICH EWG. Based on their assessments of the BE study outcome, the industry concluded that 97% of the time, fasting was predictive or more discriminatory than the fed. And only for about 3% of the time, it was not predictive. And that was mainly for low solubility drugs. Now, around the same time, the FDA had also separately performed a retrospective analysis of the ENDAs to look into the BE outcomes under fasting and fed conditions for IR products containing highly soluble drug substances. And what we found was that in some cases, the formulations with excipients such as the swellable disintegrants like the MCC, microcrystalline cellulose, or fillers could affect the rate of absorption of highly soluble drugs in the fed state. So currently, FDA has 2 ongoing research projects for fed BE studies. The first is a grant to develop the PBPK model to predict the impact of food on oral drug absorption of certain poorly soluble drugs. And the second is a contract that was granted to a university in Germany to understand the findings from the FDA's retrospective analysis that I mentioned in the previous slide. This research will be focusing on developing an evaluation algorithm to determine if a fed study can be waived based on the disintegration and dissolution methodologies that can mimic the food-induced viscosity. Now, the results from these research projects will be presented at the public workshop on October 12 of this year, and you can access the details from the link on this slide. We also have internal research projects on the topic of FED-BE studies. So you can tell this topic is of great interest to the FDA. So the first project is evaluating the application of the Bayesian dynamic borrowing approach for FED-PK-BE studies by borrowing the results from the fasting PK studies as informative prior. The second project is investigating how the food-induced increase in the viscosity would impact the disintegration and dissolution of oral IR products. And the results from this project will complement the external contract, which I just mentioned earlier. And the next one is using the PBPK absorption model to evaluate the effect of food on bioequivalence. The work on all this tree is ongoing at the FDA, and the results may be shared at a later time, perhaps sometimes next year. The second area of research is the bioequivalence determination under altered gastric conditions. So here, we know that the recommendations for an additional BE study with concomitant treatment of pH-modifying drugs is not explicitly mentioned in most guidance documents from the different regulatory agencies. However, it is noted in the PSGs of few drug products from the FDA and the EMA. For example, FDA's PSG for palbociclib tablets. At this time, the regulators seem to have limited experience and knowledge for the optimum study design for such BE studies. Also, the guideline notes that sponsors may justify not conducting the in vivo studies by assessing the risk of bioequivalence based on totality of the evidence, which could include certain factors which are mentioned in the guideline. So research in this area can definitely better inform the agency about our recommendations. FDA has two research projects in this area. One is an extramural project, which will extend the framework of the PBPK model for FED studies to predict the effect on drug absorption in hypochlorhydric population. The other is an internal FDA research project using modeling and simulation to support bioequivalence study design and evaluate the impact of gastric pH on bioequivalence. This brings me to my last slide. I'll summarize the key takeaways on M13A implementation. 1, we need to remember the draft M13A is an ICH guideline under finalization, and FDA needs to follow the ICH process. So this guideline will not be implemented until it is finalized and adopted by FDA. And that would be the step 5 of the ICH process. 2, FDA has ongoing research to facilitate implementation of some of the key recommendations in the guideline. 3, we intend to update the relevant guidance documents to align with the recommendations in M13A when it is finalized. And then lastly, both the FDA's current practice and draft M13A offer the flexibility of providing appropriate scientific justifications in case prospective applicants want to propose alternate approaches and deviate from the guidance recommendations, including recommendations in the PSGs. This concludes my presentation. Thank you for your attention. I'll be happy to answer questions later in the Q&A session, so please send them in. Before I leave, I have 2 polling questions. The first one, FDA will implement the draft ICH M13A guideline, true or false? This is false FDA will not implement the guideline until it is finalized. Question 2. ICH M13 guideline is being developed in 3 series. True or false? That is true. I did talk about the 3 series, M13A, B, and C. Thank you again.
Thank you for the introduction. Good afternoon. My name is Lei Zhang. I'm the Deputy Director of the Office of Research and Standards, ORS, in the Office of Generic Drugs at the FDA. Today, I will talk about the FDA-EMA Parallel Scientific Advice Pilot Program for complex generic and hybrid drug products. This is one of the initiatives under the FDA Generic Global Program to support efficient generic product development and regulatory assessment. Before my presentation, I would like to have 2 polling questions for the audience. Polling question number 1. Have you heard about the FDA-EMA Parallel Scientific Advice Pilot Program for complex generic and hybrid drug products? A, yes. B, no. C, not sure. Please take a moment to choose your answer. Polling question number 2. If you are aware of this pilot program, has your company considered participating in this program for complex generic and hybrid drug products? A, yes. B, no. C, maybe. D, we do not develop complex generics or hybrid drug products. Please take a moment to choose your answer. This poll will help to understand how many of you may be aware of this program and whether you are considering of participating in the program. Later in the Q&A panel, we will look at the results of these 2 polls. Now let's get back to the presentation. The objectives of my presentation today are to describe the scope of the FDA-EMA Parallel Scientific Advice, PSA, pilot program for complex generic and hybrid drug products, to describe what products are defined as complex products by FDA and as hybrid products by EMA, to delineate the goals of the PSA pilot program, and also to provide highlights of the PSA pilot program and its processes. In addition, I would like to share what FDA has learned through the PSA pilot. Finally, I also will share a few tips for industry to participate in the PSA pilot. The FDA-EMA PSA pilot program for complex generics and hybrid products was launched exactly 2 years ago on September 15, 2021. It is an expansion to the existing PSA programs between FDA and EMA for new drugs and vaccines or gene therapies. Similar to the existing PSA programs, this new PSA pilot program allows applicants to engage in concurrent scientific conversation with both agencies on key issues during the development phase, with a focus on complex generic products and hybrid products. The reason that it is a mouthful to say what products are under the scope of this pilot program is because there are differences in how FDA and EMA define and regulate those products under their jurisdictions. The goal of the PSA program is to support global development of these products that are harder to develop as generics or hybrid products under either jurisdiction. FDA's definition of complex products, which was since GDUFA II, are those products that include complex active pharmaceutical ingredient, complex routes of delivery, complex dosage forms or formulations, complex drug-device combinations, and other products where complexity or uncertainty concerning the approval pathway or possible alternative approaches would benefit from early scientific engagement. You can see a few examples of complex products as defined by FDA on this slide. For a full description of what is considered a complex product, you will refer to GDUFA III commitment letter or FDA map on the classification of complex products. Complex generic drug products are in general harder to develop with traditional bioequivalence methods, and many of them may require additional studies, including comparative clinical endpoint bioequivalence studies. Complex products in the U.S. have a significant overlap with hybrid products under EMA. Under EMA, complex hybrid products are those whose authorization depends partly on the results of tests on the reference product and partly on new data. For these products, although they considered generics by US FDA, they are not generics under EMA, and they are hybrid products. They are submitted under Market Authorization Application MAA Article 10.3 to EMA, not Article 10.1, which is for generics. On the other hand, some complex generics under FDA's definition could be generics under EMA through MAA Article 10.1 if BE studies are sufficient to support approval. For example, those long-acting injectable products. For the purpose of the PSA pilot program, the products do not have to be both complex generics through FDA and hybrid products through EMA to be eligible. The program is mainly to focus on the products with complexity and can benefit by concurrent scientific discussion with both agencies early in drug development. So what are the main purposes of the PSA pilot? The main goals of this program include to increase dialogue between the two agencies and also use the program to optimize the applicant's global product development by enabling them to discuss specific questions concurrently with both agencies, and also to further provide applicants with a deeper understanding of the basis for regulatory decisions from both agencies. The process can help drive convergency to help applicants avoid redundant replication of work and unnecessary testing replication or unnecessary diverse testing methodologies. And it also will help shorten the time to drug development and approval in both regions or globally. The PSA General Principles document that can be accessed through the link shown at the bottom of this slide gives some high-level overview of the program and its processes. The agencies conduct PSA meetings under the auspices of the confidentiality arrangement between the European Commission, the EMA, and the FDA. It provides a mechanism for EMA and FDA assessors to concurrently exchange with applicants their views on the scientific issues during the development of complex generic product and hybrid products. Alignment between the agencies can lead to more efficient and linear global development. And this program is voluntary for applicants. What may be a good candidate for the PSA processes? Some example could include those products that may have different recommendations in the PSGs from FDA and the EMA. Then the applicant may use the PSA program to propose a study design that may be acceptable to both regular agencies, which may be deviate from either agency's product-specific guidances. And this PSA process could drive convergences so that applicant may not need to conduct multiple studies to satisfy both agencies' requirements. Other example can include that the process can be used to discuss the alternative B approaches, and the use of modeling and simulation. After the PSA process, each agency will retain its individual regulatory decision-making regarding drug development and the marketing applications. Both agencies will strive to provide PSA responses that are convergent or explain the differences between the agencies. Any EMA fees applicable for the scientific advice are unaffected by the PSA status. And from the FDA side, PSA meetings are not considered generic drug user fee amendments or GDUFA meetings and are not subject to the performance goals for scheduling and conducting those meetings. The PSA process is designed to align the process and timeline of the pre and the product development meeting at the FDA as much as possible with the process and the timeline mandated by the EMA Scientific Advice Working Party for their scientific advice process. Similar to the FDA's product development meeting, PDAP meeting, PSA has 3 stages. At stage 1, applicants submit one single request for PSA letter or justification letter to both EMA and FDA at the 2 different mailboxes. And at this stage, no full package is needed. This is unlike the pre-ENDA meeting package. It takes 14 days for EMA and FDA to decide if accept the meeting request. FDA and the EMA will coordinate and communicate on their decisions. If both agencies agree to accept the meeting package, then it will move to stage 2, where it takes the most time of this meeting process, including the meeting package preparation and conduct of the meeting. During stage 2, there is an optional step that the applicant can send the draft full meeting package to EMA for validation review before they submit the full meeting package. At this stage, FDA will get a copy but will not review the draft meeting package. And I put a star here as this timeline at this stage varies. It will pretty much depend on the quality of the meeting package and how quickly the applicant can address EMA's comments at the draft meeting package. Once the full meeting package is submitted to both agencies, the clock starts as day 1. And the bilateral meeting between EMA and FDA will occur at day 90, approximate day 90, and also the trilateral meeting, which involves applicant, EMA, and FDA, will occur on day 120. As noted here is that those dates, 90 days or 120 days, are an estimate, and it will be largely determined by the annual EMA Scientific Advice Working Party meeting schedule, which normally occurs in a 1-week window per month, and typically in the non-summer month. So the dates can vary depending on when the full meeting package was submitted to both agencies. After the trilateral meeting is held, the FDA will issue the final meeting minutes, and EMA will issue their CHMP final advice letter within 30 days after the trilateral meeting. And both agencies will issue the final communication independently. This slide compares a few key features of the PSA and FDA's PDAP meetings, which most of you are familiar with for your reference. One thing I would like to highlight is that PSA meeting will be conducted as a video conference for 90 minutes, longer than the typical product development meeting, which is for 60 minutes. In addition, there's no in-person option for the PSA meetings because it involves both agencies. And also, as I mentioned earlier, the timeline to conduct a PSA meeting can vary depend on the meeting schedule. We have run this pilot for 2 years now. There are several things that we have learned through the pilot. We did not receive a large number of PSA meeting requests. So far, we have 2 PSA meeting requests that has been granted and have gone through the PSA process. This low number may be because the number of the applicants who submit generic drug applications to the FDA and centralize the EMA process is limited. We think that the PSA program can be the opportunity to expand the number of generic drug applicants that do submit applications to both jurisdictions. We also hear concerns from some of the applicants that the PSA program would require additional testing beyond what would be expected if the applicant sought individual advice from each regulatory agency. However, I would like to say that this has not been the experience with the pilot applications. FDA monitored and evaluated the pilot program, and one feedback received was that this is a learning experience for both regulators and applicants to understand the differences in the process and the meeting expectations from 2 agencies. The process also has been used to help convergence on science to address specific challenges in the program. For example, what scientific bridge and/or data can be used to support the use of a common comparator in one study that can support applications to both agencies. In general, the pilot was implemented as intended, demonstrating the long-term potential. The immediate benefits at this point were more visible to regulators than applicants. One lesson learned was that the designed process can be further clarified and improved to encourage applicant participation. Some recommendations based on the program's preliminary assessment are we needed to have more procedural clarifications, and also the timeline and expectation of the PSA can be further clarified because they have similarity and the differences between the PSA and the current either FDA or EMA process. And also we need to develop best practices for meeting package preparation and participation and share that with the applicant. All these recommendations and lessons learned have been communicated with the EMA, and also some of the key recommendations has already been implemented to further improve the process along the way. Having shared all these, what we have learned, I also would like to share a few tips for the industry to participate. First, you need to know the process. I hope this presentation can help you understanding better the design of the PSA program, its goal, and its benefit and its process. Second, you need to consider and be aware of the jurisdiction differences that can pose some challenge for your specific drug development program, and understand how the PSA program may help you to reduce the redundancy in your development program as one of the possible benefits. Third, you need to define the purpose in order to optimize the trilateral meeting with FDA and EMA, and also what are the meeting expectations. Finally, it is very important for the applicant to work closely with the project managers along the way from both agencies, as there are many coordination needed in order to navigate and keep the communications across all involved parties, both within and outside of the both agencies. Before I conclude, I have 2 challenge questions for you. Challenge question number 1. The Parallel Scientific Advice PSA meeting is part of GDUFA meetings and will follow GDUFA meeting process and timeline. A, true. B, false. The correct answer is B, false. PSA meetings is not part of the GDUFA meetings, and it will not follow GDUFA meeting process and timeline, although we did try to align with the pre-ENDA meeting process as much as possible. Challenge question number 2. What are the following products that may be eligible for the PSA pilot? Select all that apply. A, long-acting injectable products. B, immediate release oral products. C, modified release oral products. D, orally inhaled products. The correct answer is A and D. Long-acting injectable products and orally inhaled products may be eligible for the PSA pilot. You may refer to my presentation earlier that explains what are considered complex generics. I would like to conclude with 3 key take-home messages. First, ENDA applicants can gain an understanding of both agencies' recommendations on specific questions regarding the global development of complex generic products or hybrid products through the PSA process. Second, the PSA pilot is still ongoing to accept more meeting requests so that areas of improvement can be identified to support future recommendations. Third, we highly encourage applicants to participate in the PSA program by understanding the potential benefit to your specific global drug development program. FDA is mostly interested in submissions that have the potential to reduce duplicative studies for complex generic products that I also provide a few examples at my presentation. Questions about the PSA program can be directed to preenda@fda.hhs.gov mailbox. You can also refer to the PSA and also OGD Global Generic Drug Affairs website as shown on this slide for more information. Thank you for your attention. You may send your questions through the Q&A box. I will be back to address your questions during the Q&A panel later. Thank you.
Hello, my name is Brian Foreign. I'm the Deputy Office Director in the Office of Study Integrity and Surveillance, the Office of Translational Sciences, CEDAR. Today I'll be providing you a presentation regarding data reliability and integrity for BAB study inspections and global collaborations regarding those inspections. As a disclaimer, the contents of this presentation are my own and should not be construed as expressing the views or policies of the U.S. Food and Drug Administration. The objectives of today's presentation will be to provide an overview of the CEDAR bioavailability and bioequivalence compliance programs, oversight of testing facilities that conduct BAB studies, as well as provide a high-level and brief description of how international collaboration occurs in the BAB study conduct paradigm. The agenda for today will be to provide an overview of bioresearch monitoring. It specifically describe CEDAR OSIS's BioMO programs, describe how the conduct expectations for BAB studies play out, give you a brief description of how data integrity works in the BAB study arena, as well as a brief case example regarding data integrity for BAB studies, and a description of the complexity and criticality of international collaboration for BAB study oversight, including regard to data integrity. CDER's BIMA program objectives, to start off, the BIMA program is an agency-wide program administered across FDA. In particular for CDER, our primary focus is on ensuring the right safety and welfare of human research subjects, verifying the accuracy and reliability of the clinical and non-clinical trials that are submitted to the agency, and ensuring those studies are conducted in compliance with our regulatory and statutory framework, including ethical treatment of the subjects enrolled in any of those studies. OSIS's BIMA program is effectuated through the Office of Study Integrity and Surveillance, OSIS's vision, and our mission. Our vision in particular is to improve the public health by protecting the study subjects enrolled in clinical baby studies and promoting the proper conduct of studies under our regulatory oversight. Regarding our specific mission to effectuate our vision, we promote the public health by ensuring the welfare of study subjects enrolled in our trials, as well as verifying the quality, study integrity, and regulatory compliance of bioavailability and bioequivalence studies, non-clinical studies, also known as good laboratory practice studies, and animal studies, AR studies. Here on slide 7, you'll see a high-level overview of CDER, Center of Drug Evaluation and Research's overall structure. Within that, in the orange boxes, you'll see the offices of Study Integrity and Surveillance nestled in there. We have 2 primary divisions within OSIS, one being the Division of New Drug Study Integrity, and the other one being the Division of Generic Drug Study Integrity. Our primary mission, as we just described, is effectuated by supporting our customers and primary stakeholders, one being the Office of Regulatory Affairs, who we work with directly and collaboratively to ensure that we have appropriate inspection oversight of the programs that we oversee, as well as stewarding work with the Office of Compliance, Office of Scientific Investigations, and the Office of the New Drug and Generic Drugs to make sure we are providing information necessary for purposes of application decision making. To effectuate our mission, OSIS has directed our work into 4 discrete domains. The first is we use a risk-based model to ensure that we're appropriately selecting sites that are necessary to inspect or conduct a remote regulatory assessment to ensure we are providing information to allow marketing application determinations by the agency. We evaluate the sites via the inspections or remote regulatory reviews, RRAs, to ensure the study conduct is appropriate and the integrity of those study data. We review the evidence collected via the inspection or RRA and provide reports to the review division regarding the outcomes of those inspections. Those recommendations that OSIS provides are the support function that we operate to inform the review divisions of what is needed, as well as to provide compliance evaluations. In terms of globally BAB study conduct inspections, we go on inspections or RAs to determine that the studies supporting applications to the agency are appropriately conducted to ensure that the data and subjects enrolled in those data are protected and were adequately and consistently overseen by the testing sites and the sponsors that contracted with those testing sites in most cases to conduct the studies. What is essential there in order to do that is that the records maintained by those sites be sufficient to allow us to confirm each aspect of the study conduct and reconstruct the totality of that. So in order to do that, we review all subject documents, analytical runs from those subjects in terms of the evaluation of the biological samples from clinical BAB studies in order to ensure that the data submitted to the agency is reliable and verifiable. Oftentimes, you hear an interesting phrase bantered about the absence of evidence isn't the evidence of absence. The question always for us in terms of the agency and the BAB program in general is that type of language and thinking relevant or over applied to what we do? As you see, study eligibility in terms of enrolling subjects requires certain elements of the protocol be demonstrated that a subject has certain conditions or has certain values in their laboratory results. But where an issue is identified, there's some abnormal range, the expectation isn't that silence means that nothing, that the subject must have been deemed to be eligible for enrollment. If there's an abnormal value in that subject's records that raise a question on eligibility, is the expectation for the site and investigator to resolve that discrepant issue. If there isn't a resolution, then you're raising up a study conduct and data integrity issue whereby we can't determine the accuracy of that information. Data integrity here, in the context of, in particular, the BAB program, is that we recognize data is fundamental to what the agency does in order to make regulatory determinations on application provability and other arenas. Data integrity to us is considered regarding the completeness, consistency, accuracy, and reliability of the information. We look at that throughout the life cycle of the data. It's not just an isolated one-time incident at the end of the study where the study report is produced. There's a design component of that where the sponsor should be determining, considering what data controls are needed to make sure that the records being produced by a testing site conducting the study are complete and consistent and accurate and can be relied on. That should be done throughout the course of the study. It should be concurrent during, and there should be a periodic review. BAB studies are short, so there is an expectation that that isn't exhaustive, but there needs to be some consideration. It's a shared responsibility across the sponsor, their contracted entities, the testing sites that conduct the studies, as well as the agency via our compliance and oversight to ensure that we are getting the cleanest, best data to make sure that the products that are being submitted to the agency and the data supporting those products is reliable. I want to provide a quick case example. The study DIRE, which stands for Data Integrity and Reliability example, oddly enough. Here we have a situation where the agency in the BAB program would have conducted an inspection. We'd look at all the documentation regarding the clinical analytical site conduct, look at runs, look at how the subjects were dosed, et cetera. During the course of the analytical inspection and how they're analyzing the biological samples from the subjects in the clinical, we asked the firm to provide their PK data sets, their pharmacokinetic data sets, and ask them to plot that data so we can look at the PK trends, look at what is going on for purposes of the totality of the population of subjects enrolled in that study. One signal that may have popped out there, right, as you have on the screen on slide 11, you have where subject A period 1 and subject B period 2 seem to be overlapping entirely. One being subject A is on the test and subject B in period 2 is on the reference, right? So how could the test period of subject A and the reference for subject B be mirror images? And then the converse occurs when you look at subject A's period 2, which is the reference, and subject B's period 1, which is the test. You get the comparable mirror images, but you have the reference and test now being reverted, but comparable curves, which raises significant concerns. If you see this once, maybe aberrant, maybe just something that occurs, by chance in a population of subjects. But when you start seeing it more than once across one study, across multiple studies, or within one study, it starts to raise a question on data integrity. And further example here that provided is where you have a population of subjects when the site actually plots out their curves in totality, and the PK data, looking at the TR ratios, the Cmax, you have a distinct separation of populations into two distinct cohorts. It looks like for subjects 1 to 25, they seem to be well below, the TR ratio of 1, but then in the latter cohort, subjects 26 to 50, it's well above. And overall, the study appears to be meeting bioequivalence. So that 90% confidence interval between 80 and 120. What do you do when you see that type of data that's abnormal? The site has no data, no information indicate how they could have and what they did when they would have observed such a divergent physiological trend. That in and of itself is something that raises significant concerns and questions. If your study records don't allow you to explain the information or how you could have significant overlap between subjects across different studies, or a divergent patient population or subject population, pardon me, where you have two distinct cohorts within a subject population, how within normal variance would you have two distinct populations? It's unexpected unless there's a significant examination, right, by the firm to do that. In these cases, that did not happen, which results in what is the outcome, right? What do you have to do? The unexplainable, anomalous data to the agency in the BAB context, let me say that, equates to data integrity issue that undermines our ability to verify that that site is conducting the studies appropriately. The end result there for the agency is we must make a determination regarding the reliability of the studies generated from that site. What is the impact on the submissions to the agency, whether that's pending approved or tentatively approved applications, and whether or not the BAB studies, if pivotal, need to be totally redone. And in the course of this, and overall for these data integrity cases that we're seeing it for BABA that we might see, there are significant discussions with the foreign regulatory counterparts, which is the latter part. On slide 15, you'll see a really complicated diagram, and it's intended to indicate that our collaborations for data integrity in the BAB context with our international counterparts is complicated. There are a lot of reasons for that complexity in the international collaboration for the BAB studies that we oversee. In part, we are having significant amount of conversation with our foreign regulatory counterparts. That is without question. Part of the reason for that is that for the BAB studies overseen by OSIS, a significant number of those are conducted outside of the U.S. in whole or in part. We also know that oftentimes the same products are being submitted to other countries for marketing authorizations, and those countries are also having to do comparable inspections of the studies supporting their applications. We know that all regulators, no matter where they're located in the world, do not have unlimited resources. So collaboration to ensure a full, to the extent possible, oversight of the BAB study complex is essential. So there is significant communication with our regulatory counterparts. A key piece here to keep in mind, though, is even with that collaboration and even with, the intended, comparability, FDA and international regulators' actions in the final setting may differ. We do have similar study conduct expectations. We collaborate in ICH, in ICMRA, in OECD on the GLP side. There's a significant amount of work that OSIS does on a daily basis and weekly, monthly to work with our regulatory counterparts, EMA, MHRA, WHO, Health Canada. But we have to always recognize that our authorizing authorities, our legal frameworks are different, which can lead, even if we're doing comparable inspections and sharing information, to different outcomes, different ability to take actions. Whether or not FDA is taking a strong action, EMA is issuing an Article 31, or WHO has a notice of concern, we might in the end have a slightly different external facing result, but that isn't because there isn't comparable support for the other regulatory agencies taking their action, but in certain contexts, the action being able to be proven by any given regulator may differ from each other, which is a key point to keep in mind. So on to the challenge questions. The OSIS BAB inspection program is focused on A, verifying reliability and integrity of BAB study data submitted to FDA, B, reviewing and approving submissions to FDA by verifying the reliability and integrity of BAB study data, C, verifying the reliability, integrity, and regulatory compliance of BAB studies submitted to FDA, or D, none of the above. The correct answer here is C. Challenge question, the last one is for comparable inspections, same site and same product. FDA BIMO inspection program actions will mirror the regulatory actions of our foreign regulatory counterparts. True or false? Correct answer is false. And thank you very much. Look forward to talking with everyone shortly.
Thanks so much for all the great presentations in this final session. As a reminder to our audience, if you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. In addition to our presenters, we'd like to welcome the following panelists: Dr. Wenlei Zhang, Senior Advisor for Innovation and Strategic Outreach, Office of Research and Standards, OGD, and Dr. Jiazhen Zhang, Deputy Division Director, Division of Bioequivalence 2, Office of Bioequivalence, and OGD. We have some questions coming in right now, and the first group of questions will be directed to Dr. Sarah Ibrahim. And here's the first question for Dr. Ibrahim. Who are the 7 regulatory members of the generic drug cluster?
The question. The current members are, of course, FDA, and then the regulatory authorities that represent the EU, which is EMA. We have United Kingdom, Swiss Medic, Health Canada, and representatives from Israel, and the latest addition was Australia, TGA.
Thank you for responding to that question. We've got a couple more questions for Dr. Ibrahim, and here's the next question. How can other regulators collaborate with OGD outside the members of the generic drugs cluster?
Thank you for the question. As I mentioned, the generic drug cluster consists of the 7 regulatory agencies that, based on the confidentiality commitments, have a broader range of topics that can be discussed. However, the Global Affairs Program and a number of the international offices within the centers, as well as the Office of Global Policy and Strategy, have numerous methods by which we interact and share technical information with other agencies. And then, of course, we have our collaborations through external organizations such as IPRP and ICH, and those allow for venues with further engagement. The best answer would be, if you don't know, then reach out to the Office of Global Policy and Strategy, and they will assist in navigating to the level of collaboration that we are able to have with those regulatory agencies.
Thank you for responding to that question. We do have one more question for Dr. Ibrahim, and here's the question. How frequent is the international collaboration and engagement outside the scope of the generic drug cluster?
Thank you for the question. I think we've seen through all the presentations that our international engagements span across a number of engagements. And as I mentioned, whether it's IPRP, GBHI, ICH, and of course the cluster collaborations, the parallel scientific advice, I would say that our international engagements are on a daily basis. In fact, in a recent poll, when we asked the public, are you aware of how often our engagements are with EMA? The reality is it is daily. So there are significant international engagements and collaborations amongst our regulatory agencies.
Thank you for responding to that question. Moving on to our next panelist, we've got a few questions that came in for Dr. Nullifer Temple. And here's the first question for Dr. Temple. Will the guidance document cover all dosage forms, as an example, injectable emulsions?
Hi, Ray. Thank you for the question. The scope of this M13 is limited to orally administered IR products, which are solid oral dosage forms, and which are also designed to deliver the drug to the systemic circulation. So it would include dosage forms like the tablets, capsules, the granules, powders for oral suspensions. It does not cover the injectable emulsions. But also at the same time, the principles for other dosage forms, such as for the rectal or inhalation or the nasal products, the principles of the in the M13 may also apply to these non-oral dosage products in case the action is supposed to be for an immediate release and also meant for systemic exposure. Thank you.
Thank you for responding to that question. We've got a few more questions for Dr. Tampel, and here's the next question. You mentioned the FDA guidance on BE studies with PK endpoints for drugs submitted under an ANDA will be updated. Why is it necessary?
Sure. So FDA will move towards adopting the ICH guideline and issuing it as the FDA guidance for the industry once the M13 is adopted by the regulatory members of the ICH assembly. When this occurs, we recognize that the scope and the subject matter of the current PK endpoint guidance for the ANDAs will not entirely be consistent with the M13A guidance. And this can create confusion for the industry because applicants may question whether they need to follow the FDA's PSG or the general PK guidance or the M13A. And also remember, this FDA's PK guidance covers both the MR as well as the IR products. So FDA will not be able to just simply decommission or withdraw the existing guidance. And therefore, FDA will need to make the necessary updates to the guidance to eliminate the discrepancies between the recommendations for the IR products, between the two guidance documents, the ICH and the FDA guidance documents. And also, we want to retain the relevant portions of the MR products, as well as certain aspects of the IR products that don't fall into the scope of the M13A. So that's why we would need to revise that guidance. And for the same reason, FDA will also need to update some of the PSGs. Thank you.
Thank you for responding to that question. We do have another question, another two questions for you, Dr. Tampel. And here's the next question. Can you elaborate on the research with the Bayesian approach for Fed-BE studies?
Okay. Sure. Thank you for raising that question. The Bayesian statistics, it's a particular approach for applying the probability to a statistical problem. So this approach starts with a summary of our prior beliefs, and these beliefs are based on relevant available information. So for this particular project, our idea is to assess the feasibility of performing a second study, which generally would be a Fed study with a smaller sample size, by borrowing the results of the first study as informative prior using the Bayesian dynamic borrowing approaches. And in our case here, the first study would often be the fasting BE study. So we are seeing this approach being used in pediatric trials, and we want to investigate its applicability for the Fed-BE studies. And we hope that this approach may offer some advantage of reducing the human exposure to the study drugs and reduce the burden on the generic drug development programs, as well as it would help us to improve the efficiency of our drug approval process. Thank you.
Thank you for responding to that question. We do have one more question for Dr. Tampel, and here is the question. How could high-risk products be determined, and are there some guiding rules for that?
Thank you for the question. I think although it's not directly related to the scope of today's talk, I'll address it because this question may be on many people's minds. So first off, I would want to emphasize that the high risk here, the term high risk, is not referring to any specific risk to the patients. It is referring to the risk of incorrectly concluding bioequivalence of a product if it is tested only under one condition. Generally, that would be the fasting condition. So there are certain drug products containing low solubility drug substances which may be formulated using specialized technology or manufacturing methods to enhance their solubility or to optimize the food effect. And M13 mentions some of these examples, like the microemulsions, the solid amorphous solid dispersions, lipid-based formulations, and nanotechnology, where the food can have a differential formulation-related effect. And in that case, it may not be appropriate for us to extrapolate the results of a fasting BE study to conclude bioequivalence under fed conditions. And here we may have some less experience and knowledge, and there may be more challenges that are involved with this type of products. And then there are other examples which are also mentioned in the M13A, such as the differences in the particle size of the API between the test and the reference products, or certain excipients that can affect the absorption profile of the drug substance differently under the fasting and fed conditions. So I think the M13A nicely covers the guiding principles for the high-risk products, along with the examples that also I just mentioned. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few questions for Dr. Lei Zhan. And here's the first question. What would be the fees for the pilot PSA?
Thank you, Ray, for the question. So for the PSA, as you know, the PSA involves both EMA and FDA. And from EMA side, they follow more like the existing scientific advice process. And for the FDA is more like our GDUFA meeting process. As you may see in the principal document, there will be the EMA fee, which whatever the fee applicable for the scientific advice process, it will be applicable to PSA meeting. But from the FDA side, there will be no fee to participate in the PSA program. So if you're not sure what that fee is for the EMA side, please contact EMA directly for that question. Thank you. And also, Ray, I did do a poll in my presentation. I think maybe can we take a few moments to just quickly review the poll to everyone. Thank you.
Thank you for responding to that question. We do have a few more questions. You certainly can review the poll. And here's the next question.
So do I review the poll now?
Please,
I saw the poll was, thank you. So the first question, I want to know how many people before this webinar or SBI workshop heard about the program. It seems that we have about 50% people know about, which is great, but for another half, people who never heard about or not sure. So hopefully you learned from my presentation what this FDA-EMA parallel scientific advice pilot program is, what's the goal, what's the benefit. Next question.
Thank you for that review, Dr.
Oh, thank you. Okay, next question. I just want to see for those who are aware of the pilot program, are you considering your company considering to participating? It seems like about half people that their company do not develop complex generics or hybrid products, so they may be out of scope. However, we do have at least 15% consider will be participating and also have another 28% thinking about participating. So hopefully for those who are interested, today's presentation will help you to navigate through this new pilot program and consider using it to facilitate your drug development program. Thank you.
Thank you for that review, Dr. Zhan, and we'll go on to the next question, and here is the question. As per the pilot PSA guidance, the information regarding RPI, research product identifier, is required to be included in the meeting request. Can you please elaborate more on the RPI?
Okay, thank you for the question. First, I want to clarify that it is not a PSA guidance. It's more like a general principle document to lay out the process for this pilot program. And in the process, the person did notice there's a research product identified is required, but that's a requirement from the EMA side, because this is really their identified for the active substances. And also they require people to register RPIs through the IRS platform for this number. So I would like to, since this is the EMA requirement, I would refer the person to the EMA guidance on the IPI for further information.
Thank you, Dr. Zahn, for answering that question. We do have a couple more questions, and here's the next question. What would be the timeline for submission of the meeting package for validation after the meeting request is accepted?
Yeah, thank you for the question. And this is really depend on how quickly the applicant to get the full package ready. And also this is mainly an optional step from EMA side. You can submit to EMA to review your draft full package for validation before you submit the full meeting package. So typically I learned from EMA is the company can take as short as 2 weeks to like 2 months or even longer to submit that full package. So this is really the timeline controlled by the applicant. Thank you.
Thank you for responding to that question. We just have one more question for Dr. Zahn in this round, and here's the question. In past experience, FDA did not allow us to use the EURLD for a BA study of a complex generic product, even though the EURLD and US RLD is essentially the same. Will this PSA program help the applicant to reduce duplicative BE studies?
Yeah, thank you for the question. I cannot comment on what this person who gave the question, the experience is. I cannot comment on that, but through my presentation, you do see this PSA program. The goal is to help the applicant to reduce the duplicate BE study. If you found there's a challenging in your case, please submit a package and pose a question to both EMA and FDA, and we can see whether there's a path forward to design a study that can satisfy both regulatory agencies' needs or requirement. Thank you.
Thank you for answering that group of questions. Moving on to our next panelist, we have a few questions that came in for Dr. Brian Folian, and here's the first question for Dr. Folian. Understanding that CDER's Office of Study Integrity and Surveillance, or OSIS, conducts many remote regulatory assessments, do you see potential data integrity concerns as being suitable for examination via an RRA?
Thank you very much. That's a really interesting question and very timely. I think, as I discussed in the presentation, when we look at data integrity, right, we're considering, whether or not the data in front of the agency submitted is complete, consistent, accurate, and reliable. So for purposes of a remote regulatory assessment or an inspection, the crux there is really determining what we can actually see, what the actual issue is at hand that might be raising the data integrity concern. A lot of times we might identify something during an RRA that would actually be indicative of some concern with regard to the study sites, study conduct, and the data they produce via that actual conduct, right? So if there's something submitted to the agency and we go to a firm and we're remotely interacting with them and we notice that their records don't support something that's submitted to the agency, you start to open questions. So whether or not that's via remote or on site, you're doing the same thing, somewhat that antenna goes up, right? You start actually looking into it and you start, doing a deep dive and trying to figure out what you can see. There is some intricacy to that in a remote setting, obviously, but, over the past 3 years and even prior to that, we've been learning a lot about how these interactions can occur and, getting more comfortable with, the technology and how we engage. So
whether or not we have a data integrity signal identified, prior to determining what interaction is appropriate with a firm, or if we're in the midst of an RRA with an entity. There, it's all case specific, and in terms of whether or not, we can start digging into that and seeing what the entity has, asking questions, reviewing the data on site and the data that can be shown when we're remotely looking at with the firm, what their study files look like, what's been submitted. So generally, there is the potential. You can never say carte blanche that everything can be looked at comparably with the remote versus inspection, because they are, as everyone knows, different things. They're both fit for purpose tools. So we're still continuing to evaluate and work with RAs to determine the extent to which they can support the agency's inspection related activities, so we can make sure that the products we're approving have reliable data to support them. Thank you very much.
Thank you for responding to that question. We do have a couple more questions for Brian Folin, and there's one for Dr. Folin, right? The collaboration and complexity of FDA and international regulatory is really interesting. So, to the extent possible, can you provide more discussion on how, practically speaking, different outcomes may be reached?
Thank you again for this question. I think, as Dr. Ibrahim's talk earlier in the presentation, as well as mine, it highlights the uniqueness, the shared interest in trying to reach that harmonization and commonality in terms of approaches, but also recognizing where we have differences in the way our regulatory oversight is effectuated. So it's an interesting period where you'd have, you would see a notice of concern from the World Health Organization, or an action being taken by the European Medical Association. And what does FDA do with seeing and through our collaborative efforts with those entities? It's a really interesting dynamic scenario, right? Because as we've heard in some of the questions, it's interesting where people think that there's duplicativeness or the harmonization is possible, recognizing even if that is the shared goal, the underlying basis for why WHO might find an action supported might not necessarily be the same issue that the FDA may have in front of us, right? There's always differences in terms of study conduct that's submitted to WHO, or study conduct that's submitted in support of data that's submitted to FDA. So we always have to do that very specific consideration of the information in front of us that can lead to different outcomes. You might have comparable drug products being examined, but overall the reality is that we have to do our due diligence to consider the information in front of each of the regulatory agencies in effectuating our regulatory oversight, which shouldn't be construed to say that we aren't supportive or otherwise constantly considering what each of us are doing and trying to the extent possible to be in the same area, but that isn't always going to be the case. So further work, as Sarah mentioned, is ongoing endeavors across the universe of what FDA does. We continue to collaborate and seeking that commonality where we can. Thank you.
Thank you for responding to those questions. Moving on to our next panelist, we do have a question came in for Dr. Wenli Zhang. And here's the question for Dr. Zhang. I know there is an International Pharmaceutical Regulator Program Nanomedicine Working Group. And this working group, is there any activity ongoing related to generic products?
Thank you, Ray, for this question. Actually, the simple answer is yes. I can elaborate a little bit more. The International Pharmaceutical Regulatory Program Nanomedicine Working Group, abbreviated as IPRP-NWG, works on the exchange of non-confidential information on nanomedicines among IPRP Nanomedicine Working Group members and the regulatory harmonization on nanomedicines. So this working group discussed topics related to innovator drug, generic drug, or hybrid products involving nanotechnologies. Currently, there are 15 regulatory members in IPRP Nanomedicine Working Group. In 2020, the working group conducted a survey on liposome and the lipid complex products among member agencies as well as non-regulatory stakeholders to provide a comprehensive analysis of the regulatory landscape for liposomal products to identify the needs of both research and standards development in order to enhance the potential for harmonization of regulatory requirements. Based on the survey response by regulatory agency representatives, the majority of respondents indicated that they had no review or approval experiences with any follow-on or generic liposomal products. Several agencies reported they had approved generic liposomal products, but only limited to like pegylated liposomal doxorubicin hydrochloride and liposomal amphotericin B injection. And most agencies consider it is challenging to assess generic or follow-on liposomal products. For example, they do have questions. What are the appropriate equivalence criteria to compare innovator and the generic liposomal products? For non-regulatory stakeholders, for example, we conducted a survey among academic institution, pharmaceutical industry, trade association representatives. And based on the response, the call for alignment of critical quality attributes for liposomal products among different regulatory agencies. And also they recommend guidance development to address the evaluation of generic or follow-on liposomal products. Considering these survey responses, the working group had quite some activities going on, like we have information exchange on scientific and regulatory considerations for generic and follow-on liposomal products. In the past 2 years, actually, we have organized webinar presentations given by the agency representatives who had experiences in reviewing generic liposomal products to IPRP nanomedicine working group member agency scientists. They shared their agency review criteria and also the rationales supporting such review criteria and the review experiences. So the agency, which hasn't reviewed the generic or follow-on liposomal products, can gain experiences in attending these webinar presentations. In addition, some similarity and difference among different agencies were identified during the discussion. Hopefully, some scientific consensus can be reached among IPRP nanomedicine working group member agencies regarding any specific generic liposomal product. Also, a number of liposome standards were developed by ISO and ASTM. So these standards were very useful for both innovator and the generic applicants working on liposomal product development. IPRP nanomedicine working group have been helping disseminate these standards and provide training to member agencies. Yeah, that's about it. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we do have a question came in for Dr. Xiaojun Zhang. And here is the question for Dr. Zhang. For oral extended release products intended for administration only under fasting conditions, can the agency consider waiving the requirement for a fed study?
Thank you for the question. ICH M13A guideline applies to immediate release solid dosage form. Therefore, oral extended release product is not in the scope of M13 guideline. The harmonization on modified release product will occur at a later time.
Thank you for responding to that question. We have just under a minute left. We're going to try to squeeze one last question in, and this will be directed to Dr. Sarah Ibrahim. And here is the question. Is the generic drug cluster an appropriate place for discussion of a single global comparator's development?
Thank you for the question. The generic drug cluster is a confidential forum. There really is no topic that we do not raise in the cluster if there's a public benefit for our citizens. And most of our engagements are on scientific engagement. So there's no true limit to the level of scientific dialogue that is ongoing in the cluster, so long as there is a benefit and access for our patients.
Thank you for responding to that question, and we have run out of time for questions in this final Q&A session. A huge thank you to our speakers and panelists over the last 2 days for answering numerous questions that came in from our audience. Before we provide some housekeeping reminders, we'd like to welcome back Dr. Lineberger to provide some closing remarks for advancing generic drug development. Welcome, Dr. Lineberger.
Thank you, Ray. This was a fantastic workshop. First, I want to thank the SBIA team for all of the logistics, the organizing committee for putting together the program, and finally, the speakers and the panelists for providing the fantastic comment. All of those people had to work together to have a successful event. At this workshop, you heard updates on complex science that can support the development of complex generics products. You got practical advice from FDA staff who assess your applications as to what they look for when reviewing some of these novel approaches. This is a very unique and practical insight for those of you in the industry and a perspective on what happens to your application and how FDA staff look at it. You saw examples of complex generic products that were approved, and you gained some insight into some upcoming regulatory changes or new programs such as our model-informed evidence and our global harmonization. But now you know what's possible in all of these. Let's talk about what some of the things you can do with the information you've learned about this or the insight that you saw from today's workshop. The goal of this is, in one sense, to bring generic competition to every product, even those complex products that don't currently have any generic competition. And from the industry perspective, these are huge opportunities for you and your organizations to contribute to public health, but also to, for the business opportunities as well as being the first company to provide these opportunities. And today, during the Q&A sessions, we tried to answer as many questions as possible. But we know that you have more questions. We also know that you have many questions that are product specific and specific to your particular development programs. We can't answer all of those at this forum. And answers to these questions will help fill in the gaps for your development programs. And so what can you do? The best thing you can do is to use, for complex generics, is to use the pre-NDA meeting process to get the scientific advice before submission. This is exceptionally valuable, right? Because we all want the review process to be as efficient as possible, to get timely evaluation and approval of new generic products. But the way to do this is to get things right the first time. And the way to do that is to make sure that as many of the scientific issues are resolved through the pre-application process as you can. So use the product development meetings. During GDUFA III, we have a new pre-submission meeting for complex products. We encourage people to use that meeting process as well to help share insight into what you're doing with FDA and get appropriate feedback on that, as well as to use the control correspondence as well. Today's meeting, you also saw an illustration of FDA's commitment to more meeting opportunities. You heard for the first time about our model-informed evidence pilot program. This is an expansion to the type of questions that you can bring into the meeting process. So it covers modeling and simulation related questions that might be applicable to more than one application or to non-complex products where you're supporting some novel approach. So again, our commitment is, we recognize, and the theme of this workshop is moving, how do you use the science to move products through the review process and make them available to the public? And the meetings and the product-specific engagements are such a key part of that, is that we hope that what you'll take away from this are the opportunities for developing these complex generics and the places you can go to interact with FDA to get feedback on the scientific issues facing these development products. And again, just to close out, we have a shared goal to make generic versions of complex products available. And this workshop is a very important part of that collaborative effort. So you can really see across 2 days of content, a lot of the different areas of opportunity across many different categories. And so we hope this workshop has been very helpful to you and helps you develop complex generic products and points you to many different ways that you can interact with the agency to have scientific discussions about those development programs. And so with that, I want to turn it back to Ray Ford and the SBI staff so they can close out the housekeeping for this workshop. So thank you all very much.
Thank you, Dr. Leinberger. And we do have a few closing reminders. Please remember the 14 days deadline. That's September the 28th for the certificate of attendance, which supports CEs for SOCRA, RAPS, SQA, and ACRP, and also the 14-day deadline for CE for healthcare professionals. Claim codes for both the certificate in attendance and CEs will be emailed each evening following the conclusion of today's event. On behalf of Cedar Small Business and Industry Assistance, we hope that you enjoyed today's presentations and found them helpful. We look forward to your presence at our next SBIA event. Thank you for attending, and this concludes today's workshop.